Good morning to everyone. Welcome to Patras. Welcome to University of Patras. We are really happy to be here starting our European conference, which is called ONE Bridge, ONE Bridge project, European projects that we're running for 1 year with 4 countries. And our final conference, we have done 3 previous workshops in different countries, and we are really happy to have you here. It's a hybrid conference, One Health Nexus, bridging human, animal, environmental health action across Europe. As you already know, there are several problems with One Health and all the issues that we're going to discuss these days about One Health. This is 4 days conference. And we have tried organizing this conference to cover all the problems and all the issues in One Health. You know that One Health is one of the major issues that have come up through all the European and, of course, world community, scientific community. It's a way of thought. So during our 4 days, we're going to try to analyze all the issues about One Health. So we have a very full and complete schedule. And I hope that in this hybrid conference, you are going to learn. And of course, we have the time to discuss a lot on these issues. I'm happy that I have here our rector. So I'm going to ask him to give us the first welcome. Professor Bouras is our rector from the University of Patras. Professor Bouras, thank you very much that you are here.
Okay. Good morning. First of all, I would like to welcome all of you in the University of Patras, and I would like to wish to you to have a successful conference on this interesting topic. And I would like to thank you, Professor Vandarakis, for organization of this. So, welcome again, and I hope to success for this conference. Okay.
Thank you, Professor. Thank you, Rector. I would like to ask Professor Stathopoulos, who is the Dean of Department of Medicine, to make a short welcome.
Good morning, everyone. I would like also to welcome you to Patras and our university on behalf of the School of. Also would like to thank Professor Vandarakis for taking all these important initiatives regarding global health and all these actions that give a holistic perspective health, in animal health, and also in tightly interconnected. So I wish you all the best and the best success for your conference. I'm sure that would be a very. I know that you have already organized many workshops that are better insights on these issues. Best and the best success for this.
Thank you, Professor Stathopoulos. As you already know, One Health is an issue of the community. So it would be, of course, logical to have the community here, and who better person than Dr. Mastorakou, who is the regional governor to public health region of western of Greece, and I would like to ask her to have a welcome. Dr. Mastorakou has working a lot with One Health in the community, so I would like to ask her to tell us a few words on that. Thank you.
Good morning to everybody. Much to invite me to this welcome. As a host region, I welcome to Patra. And on behalf of Governor of Western Darius Farmakis, I convey his greeting. This conference is a very important conference because dialogue about One Health allowed me to highlight the important development. By 2025, we created the One Health Network, coordinated mechanism built on 6 environmental and climate risk, our University of Patras, with public health, environmental, veterinary services. Our aim is clear: to unite science, innovation, and policy to strengthen resilience and place citizens at the center of decision-making. I'm also pleased to announce a significant milestone. Western Grid is, as an active member of WHO Regional Health Network, will host the 2026 European Annual Conference. The first time a Greek region receives this distinction. This honor reflects our commitment to public health and position Greece at the heart of the European dialogue. The 2026 conference in Patras will focus on key priorities on common health, mental health, healthy and active aging, preparedness for climate change, digital transformation, strong primary health care, and deeper cooperation about cities and regions, which is very, very important. In an area of complex crisis, no sector can act alone. We must fight all together. Best wishes for the conference. Thank you.
Thank you, Dr. Mastorakou, and I know that you are going to do the best for our future conference here from the WHO. I hope that you work with that in One Health as strong as you started to do, and I hope that we have One Health issue in the top of our agenda the next future time, because it's really, really important around Europe, and it will help us a lot to improve our life and, of course, public health in general. I have to tell you that we have invited Major. Major is not here, but anyway, I have just to tell you a few things about our city and our work in the, as a welcome in One Health. Our lab of public health epidemiology and quality of life is working in a couple of projects about One Health, and you have met Zoe Anastopoulos. She's doing a PhD about AMR in One Health. We were working on this issue the last few years, and I believe that it's really, really important it should be really important, especially for the communities. I realize that most of the communities do not really know about One Health. Of course, the scientific community works a lot in One Health, but the community does not realize the problems so far. So I think that, of course, during that conference, and in general in Europe, because we are spending a lot of money now for One Health, I believe that we'll try to make them to realize the problem, and the communities will be more sensitive on these issues. And it's, of course, one of our main responsibilities to do that, to try to make them to understand the significance of One Health issues. I hope that from this conference we are going to succeed a lot of things, and we're going to have some policy dialogue at the end of that. The project is continuing, the One Bridge project is continuing until July of 2026. So having all the data from the 3 workshops that have been organized in Lille, Dresden, and Barcelona already, and the results from this conference, then we'll sum up everything, and we're gonna produce something that will be mature and to give it to the European Union about the importance of having One Health in a priority in European countries. So I hope that you will enjoy this conference, and I welcome you officially to Patras, to my born city and my university. Thank you again that you all are here, and I was hoping that most of you will be here to travel to see Patras and our university. Now, I'm really happy, it's my pleasure to have our EU officer, Ludmila Nistor Mikhailova, who is project advisor and member of HADEA. I would like to welcome you and give us a talk about HADEA, funding opportunities in health, EU for health. Thank you, Ludmila, that you are here, and I'm really happy that you are here, honoring our conference.
I hope I'm clicking the right buttons. So I have it, okay. Okay, so today I will take you to a journey to HADEA. It's the European Health and Digital Executive Agency. And also I will take you to a journey how the EU invests in health. And more importantly, how you can be part of in what are we investing and shaping the future. So my name is Ludmila Nistor Mikhailova. I am working in HADEA for a year already now, and I will guide you through the EU health program and the opportunities it opens for the organizations. So who we are and what we do. So let's see first of all the very big picture. So European Commission is the one that sets the direction, the priorities, and the goals. The agencies like HADEA, we have other agencies you'll see, come in to do the implementation. We turn the priorities into real actions. We launch calls of proposals, we do the tenders, and we monitor the implementation. So, and first to go in more details, we will need to see the big difference, not difference, but to have a clear distinction between the European Commission and the executive agencies. And to understand how EU for health works in practice, we need to understand that these are the two key players. The Commission essentially is the strategist, and the executive agency is the implementer. The European Commission sets the policies, sets the priorities, and drafts the annual programs. And the executive agencies like HADEA, SINEA, REA, it implements the calls for proposals, procurement procedures, and manages the funded projects and the contract itself. So in the overall, we don't compete, but we complement each other. So more concrete for HADEA from the agency I'm coming from, you see the title that HADEA is delivering results through implementation. So what this would mean is that HADEA implements European programs and initiatives on behalf of the European Commission. In practical terms, this means we take policy priorities that the Commission sets, and we transform them into real projects, real partnerships, and real solutions. If you see, we cover topics like health, food, space, industry, and digital. And this is all converted into programs. And also, HADEA manages the programs related to the previous slides. It's the EU for Health program, single market dealing with food, digital, which derives from Horizon Europe Digital Industry, and space. So, as previously said, HADEA is working closely with 6 directorates of the European Commission, basically the ones that give us the annual programs inputs to be implemented, and this is HERA, the Crisis Preparedness GROW, Santé, it's EU for Health, RTD is Horizon Europe DEFIS, and Connect is the digital part. So if you see about if you want to talk about HADEA, it's very interesting to see how money talks. So if you see that from 21 until 27, the budget managed by Hadea, it's around 20 billion. This is a big amount, and this is an amount that covers health, digital capacity, industry, and food safety. And from what we are talking today, it's EU for Health, it's nearly 3.79 billion we are managing as the agency. So the EU for Health program, it was set basically, it was designed by the regulation 2021, and it was meant to encompass, first of all, what has been encountered during the COVID crisis, and then to work on 4 major sets of priorities to develop on and improve and foster health in the Union, protect people in the Union from serious cross-border threats to health, and make medicines available and affordable, and strengthen health systems, their resilience and resources efficiently. So EU for Health is not just a funding program, it's a long-term commitment to ensure that Europe's health system, its citizens and its crisis response capabilities are stronger and better prepared than before. So, as mentioned earlier, the way we work on the annual work programs. These are designed by the European Commission. The annual work program defines the topics and their indicative budget, and then the call document is the one is designed on the basis of the annual work program and provides further information as per the call for proposals. It designs objectives, eligibility, duration, expected impact, and deliverables. And it's all very transparent. Everything is online. You can look at the previous work programs to compare how the priorities are evolving, and to wait for the next work program if you wish to be part of it and to apply. It is very important. It is very important to see that to understand the differences between procurement and grants, because both are of relevance, but it depends of what are you intend to be part of. They may look similar from outside, but they work very differently, and they serve different purposes. So on the left you see procurement. This is when the EU is essentially buying something. It's buying service. It's buying a study, a platform, an IT tool. So the principle is very straightforward. The EU pays an agreed price, and the contractor delivers the product or service, which is for grants. And it's very important that it's profit-oriented, meaning companies can earn revenue from the contract. The legal form is standard contract, and the ownership of the output stays within the EU, which is not this for grants. It's on the right side, and it has a difference, and the idea is completely different. A grant is the EU giving money to support actions that contribute to EU objectives, whether that's improving health systems, increasing preparedness, or building capacity. The principle is non-profit, and deliverable stays with the beneficiaries. So now that to focus more on grants, because I believe this would be the more interesting to see for you, is to understand also the type of EU health grants we have. You see the 3 types of grants. It's the first one, it's open call for proposals, and these all are published on Funding and Tenders portal. And who can apply? It's a variety of stakeholders as defined in the EU for Health annual work program. And the other type of grant is direct grants to member states, including joint actions, and it's by invitation letter to competent authorities nominated by the member states. And the last one are direct grants to identified bodies, and it's also by invitation letters, and it's entities mentioned already in the EU for Health annual work program. And it's also very important to understand when applying, it's how EU is funding Usually, normally, as a rule, EU co-funds up to 60% of eligible costs. Exceptionally, you can co-fund up to 80% for actions with a clear EU added value, which satisfy the following exceptional utility criteria. So these are two conditions for this: when at least 30% of the budget of the proposed action is allocated to member states whose GNI per inhabitant is less than 90% of the union average, or bodies from at least 14 participating member states participate in the action, of which at least 4 are member states whose GNI per inhabitant is less than 90% of the union average. This is important to understand because to avoid any kind of misunderstanding, but also to group the way your consortia, your actions, that you can get this 80%. Okay, so it's also important to understand when applying and when waiting for the procedure. So if you see the overall procedure is from the call publication, is to understand the timeline. So from the call of publication of opening until we sign, we need to have 9 months. And it's a process that cannot be compressed. So it's a process that we need to do, and it's a process that allows you to draft maybe a better proposal, and for us, time to evaluate and to sign a good grant at the end. So it's important to remember that during the preparations, we do information sessions where you can attend. You need to check the calls, frequently asked questions, which are available on the website, and Q&A, question and answers. And as in every country, we have, member states, we have your EU for Health national focal points that can really help you, guide you through these processes. This is what is now on the menu from this year. So two of calls already closed. The rest, it's still open to be applied. So if you are interested, and you can check directly the website. Links to info sessions, we all the time organize info sessions, and when we do, we also publish them on the website, so you can go back and retroactively look at it, maybe if you have missed something or you want to clarify, so all is there. So all recordings and materials are available online. So this is basically your getaway. This is your starting point and final point to communicate with us. It's EU Funding and Tenders portal. It's really a one-stop shop, and all EU programs, including EU for Health and Horizon Europe calls and projects. And so we kindly invite you to look for calls for proposals or calls for tenders for partners, projects, and project results to see how is this done, how is this delivered, how is this placed on this platform. You can search by type of program. There is an option to search by type of program. You type in the call, you call number, so you can get the information can be filtered. You can set up your profile also there and receive personalized recommendations. So, and also you can find detailed guidance and documents, yeah. So this is how, just, I will leave this presentation so that you can see how basically to click, where to get and how to personalize your access on this EU Funding and Tenders portal. And so it is also very important for you to understand when dealing with the EU for Health action grants is that these action grants, you have to review each call strategic goals and make sure when you write the proposal, it's aligned with the scope and activities of the call for proposal, because this is then how we evaluate. So these are the criteria how we evaluate, and if it doesn't feel so, it cannot not be moved forward. And you have also to pay attention to eligibility rules. These eligibility countries, consortium composition, maximum budget, timeline, and also ensure that the proposal clearly address all the course award criteria, relevance, quality, and impact. But also be realistic. There is no need to fantasize and to write unrealistic things. You need to propose actions you can really deliver and you can really report on them. And this is links to support to applicants, as I was mentioning before. It's all on our website. So yeah, you can go and check. And this is but the menu is now on the call for tenders, as I was mentioning before, just for you for information. So you can follow us. Online to all these platforms like LinkedIn, website, and also European Commission is all the time. At least on our website, on Hadea website, it's plenty of information how to keep the hand on the pulse of what's going on in the program. So thank you very much. If you need more information, I'm here, happy to discuss and to provide you more.
Thank you, Ludmila, for this presentation. Are there any questions on that, on Hades? I just want to emphasize that Hadea has done quite a job on funding this. I have participated in several projects. I believe it's a really important funding for public health in general. So I believe that we can move forward. There are several projects that have produced very, very important outputs from their work. And I have been in several, noticing several projects that can continue and are used, their results have been used in the community. And that's really important because it's to my opinion, it's a very, how can I say, a very good work on connecting the scientific community with the community and the work that needs the community to be done. So I hope that Hadea will continue with that, especially in public health, which is my area. So, and because there are really significant outputs. So, well, a little bit ahead of schedule, but it doesn't matter. Let's go to our first keynote lecture. I have the pleasure to have Ais Chouros here, who is International Advisor Health Policy and Strategy, former director at WHO, adjunct professor in Boston School of Public Health. Professor Chouros, he's going to give us a keynote on One Health as a strategic compass for European public health. Thank you very much that you have accepted our invitation and you are here in Patras. We have with Professor Chouros, my colleague Professor Gelastopoulou has cooperated in the past, and I really admire his work all this time. Thank you very much, Professor.
Thank you. Kalimera σας. Thank you for this opportunity. You know, what I would like to do today is to give you a guided tour into the evolution of key public health concepts in the last 30 years, which are highly related to what we wish to address today, which is the strategic importance of One Health approach in our days. The reason why I want to do this is because in the past 30 years, the narrative of health has changed dramatically. It basically started in the early 80s, and since then, especially the field of public health has been radically changed to what it was before. And many of the ingredients of One Health can be found in this evolution that I will describe. You may think, why are you telling us this? Why is it relevant? It is very relevant for one main reason. The key change of paradigm in health in the past 30, 40 years has been the need to stop working in silos and to work horizontally. I was director of policy and governance in WHO Europe, and one of my main jobs was to get our member states to learn to work together, breaking down the walls of different sectors, because all the evidence supported the idea that health is the result of a wide range of factors that, or determinants, which is the correct term, that need to be addressed at the same time. But you know, it's easy to say, but it's very difficult to apply, even to our own organizations. And I will explain why. Why still, today, in our days, we are struggling with this concept of working across sectors. One day in my office at the WHO, I discovered on a dusty shelf a book. And that book had this title, Intersectoral Action for Health, and underneath it, on the cover, you could see the logos of 4 United Nations organizations, including WHO and UNICEF and World Bank. I can't remember the 4th one. I was very surprised. And when I looked inside, I realized that if I had just changed the date, the book was dated 1986. If you change the date of that book to today, it would have been extremely valid and true. So, we agree and we know that everything is connected, that the real shift is acting across human, animal, environmental systems, because no sector can succeed alone. So we use the key words integrated. One Health is an integrated and unifying approach that optimizes the health of people, animals, and ecosystems. That's good. It was very good, if I can say that the COVID pandemic actually provided us, that's the positive side of the drama of the pandemic, provided us with the opportunity to understand 2 things. One was to understand the complexity and the interconnection of issues that matter for the successful pursuit of health development. What I said before, no one issue, but a range of issues that need to be addressed. The second one, which is very important, is we learned, but we really learned, what it means to be vulnerable. Who paid the heavier price in this pandemic? The poor, the excluded, the most vulnerable populations. And One Health, in many ways, has to be linked to an understanding of the value of equity and an understanding of what makes us vulnerable. We have made a mistake in the past years. We love to talk about concepts and new issues, and I'm going to mention a few, systems thinking, ecoepidemiology, socio-ecological frameworks, social determinants of health, holistic, multisectoral action, planetary health. There are Agenda 21. There are so many terms that are very similar. They have been evolving over the years because since 1992, when we had the first global summit on the environment, since then, we have been struggling with those issues, integrated, horizontal, intersectoral, multisectoral, but we made a mistake to over-intellectualize those terms. It's very difficult very often to explain to ordinary people, and even more difficult to explain to politicians and to decision makers what it takes to actually shift, and what is the key word here? To shift the governance paradigm. Because, dear colleagues, you don't understand governance, you better stop talking about One Health. Then One Health is just a slogan. It's just a slogan, or it's just an interesting topic for academics in non-real world environments to try out different approaches. But we are talking today about sustainable development. We have the 17 goals, the UN agenda. We have a wide range of issues that need to be addressed with One Health approaches, but we are not ready. We are far from ready. Now, it is true that there are a number of global strategies that give prominence to One Health. The G7, G20 agendas, the European Green Deal, EU for Health. You, of course, are familiar with the pan-European, the so-called Monti Commission. You probably have heard of that. There was an effort to try to operationalize this concept. You probably know that WHO Europe developed a roadmap for One Health, and of course organizations like FAO, UNEP, and so on, they're all working on those issues. And there are recommendations, and if you look at the WHO Euro recommendations, of course, we talk about One Health becoming a policy compass, shifting from the biomedical views to systemic resilience and equity, operationalizing, listen to terms, to upstream cross-sectoral action on climate ecosystems. Upstream, have you heard of this term? These are all terms being repeatedly used to make us move from addressing a problem to addressing the root causes of that problem. So we are talking about issues such as, of course, AMR, zoonotic spillover and emerging infectious diseases, climate change as a health multiplier, urbanization, housing, and the urban environment ecology, highly relevant on the ground to the One Health approach. We have new areas of science, like exposomics, if you heard about them, which is an area which, in my view, is going to revolutionize the way we practice medicine, the way we practice precision public health, the way we practice now the policies that need to address in the widest possible way health and well-being. But for me, having underlined the fact that the reality is interconnectedness, attention to vulnerabilities, and the need to be strategic, the preconditions for change should start from being extremely clear about our values. There is an intrinsic value of all living things within the ecosystem, of all living things. The right to health, equity. One Health shows how social disadvantage and environmental conditions combined to create layered vulnerabilities to heat, pollution, zoonotic risks, and other health threats. We need to use approaches that are inclusive and engaging communities. We need to address behavioral changes as part of this One Health approach. And we need to think about current and future generations. So let's come to the key issue of governance. Governance is the missing link between One Health theory and practice. It doesn't mean that we change, we create new structures. It means that we rethink and transform existing structures. So here is the menu. One Health requires a strategic vision. It requires high-level political authority. It requires leadership which cuts across sectors. The need to overcome fragmented mandates, to enforce joint priorities. We need regulatory coherence, aligned legislation, interoperable surveillance across human, veterinary, wildlife, and environmental domains, joint financing, institutional mechanisms, pooled budgets, national One Health platforms, and joint risk assessments. We need to be very careful about who we work with, which means who are the stakeholders, which are the key ministries, the key institutions, the private sector, local authorities, regional and municipal authorities, civil society. There is a professional groups. One Health, like the whole field of the social determinants of health, which is economic, social, environmental, commercial, cultural, political. Health is a political choice. It's a political choice because it is the decisions we make through our policies that affect inequalities in health, that affect whether the policies of different sectors are going to do good or bad on the health of populations. We need health diplomacy. Diplomacy at global, national, and subnational level. Diplomacy for health is critical to be able to negotiate this interface of different and sometimes conflicting interests. You want to make different sectors work together, but different sectors have different interests. They may be coming from different perspectives. You need to be able to negotiate, to understand what it is that makes them tick, what it is that they should be aiming for. So I want to make it clear that siloed approaches are no good in the domain we speak, but not only. And this is, if you like, one way to make you understand and think that when you talk about One Health and your mind goes directly to AMR and zoonotic diseases and so on, and you think, well, animals, rodents, mosquitoes, and that's all good. And AMR, you need to work with vets and antibiotics, and this is good. But please remember that the One Health approach is part of the universe of the new public health domain, which calls in every aspect of public health for the same things: intersectoral action, equity, understanding vulnerability, working closely with communities, building resilience, introducing systems that talk to each other, which means platforms, which means surveillance, which means budgets, which means planning, that they talk to each other. You can no longer address any public health issue without thinking the same way. I give you an example, which is not AMR. Take one of the biggest problems we have in Greece, but throughout the world. It's part of the chronic disease epidemic, obesity. There are still those who spend millions on addressing such a super complex issue like obesity in a traditional way. Where should we spend our money? Campaigns, talking to parents in schools, and generally doing things that may have worked in the 70s or the 80s, but hello, we are in 2025, and we know very well that addressing obesity, for example, would need you to engage at least 8 ministries, starting from the beginning, which is from the agricultural land and the production of food, to marketing, to packaging, to labeling, to promoting, until it goes to your child that is going to be addicted to ultra-processed foods. By just focusing here, as we say in Greek, you are making a hole in the water. You do not really have the results you should have. One Health is the same thing. You need to think. And some people may say, we do One Health. But they think they do. Because by working separately on the 3, for example, sectors that relate to your problem is not One Health approach. It's still a fragmented approach. One Health means that you work jointly. Joint platforms, joint surveillance system, joint evaluations, joint accountability. Now, a country has to develop a way to interpret these global strategies into something that will guide the nation to these approaches. If you go to Africa, you will find that many countries have national One Health strategies. Namibia, and Kenya, and several countries. Because these countries primarily have huge problems with zoonosis. In Europe, you will find mostly, like Denmark, for example, nations that have strategies for specific problems, like a One Health approach to AMR, for example. But I want to underline, as I will be closing my presentation, to one thing. If we understand that the new paradigm for public health requires sectors to truly work together, if we understand that we have to have as a guiding light the right to health, equity, and a good understanding on what makes us vulnerable, then we need to have a hierarchy, to think strategically of what and how we will make it happen. Some make the mistake to mix all this, to embark on One Health projects which will live just for as long as the funding will be available. You know, you apply for different European and other monies, you do a nice project, beginning to end, finish the project, finish the whole thing. We have the bad habit of financing and implementing out of honest academic or otherwise interest projects that are not sustainable, that leave nothing behind, that do not even leave a change of habits or a change of mechanisms that will allow us to work together. So, think of 4 levels. If you are serious about these issues, start political. You need to have high political leverage. You need to have high political and strong leadership to be able to enable all these sectors to work together, including, remember that, regions and local authorities. It's nice to have a nice national strategy, but until this strategy is applied on the ground, it doesn't help. Second, think strategically. Your strategic level is to choose your correct partners. Have we left anybody outside? Second, try to find which will be the best interventions that will have optimal impact. Third, evidence. What you do should be evidence-based, evidence-informed. It has to have the credibility that what you do is supported by science and is supported by experience. It's not only academic papers that tell us what is important and true, but also the experience. And lastly, the last level is the operational level. Where and how, what mechanisms, what systems do we need to be able to take down all this narrative and translate it into action? So I believe that that's all I wanted to mention to you and to go back to where I started from. Take the opportunity of thinking about One Health as an opportunity to be proselytized, to be able to embrace and understand and advocate for a new public health practice that will have to make us be able and capable to work horizontally and also to be much more conscious of the huge inequalities and the huge vulnerabilities that we need to address in our world for better health, better well-being, and a sustainable future. Thank you.
Thank you for this amazing talk, Professor Chouros. I'm really happy for everything that you said, because you criticize and, of course, mention a lot of things that we are not doing correctly. Any questions? I have questions for you, but let's ask for the audience first, or from the internet, if there are any questions. I have a question, very, very basic question. Are you optimistic with One Health activities that you use? Are you optimistic with the One Health activities that you are running now, around Europe, I mean, from your experience?
No.
Okay, very.
I'm not, and I think we are hiding behind our, it's, I don't see it happening. And what makes me very sad, and it's not only in the field of One Health, enormous money, funds are spent and used by people who have all the qualifications and the academic credentials, but this connection with the political and governance system, it's still the biggest wall we have to pull down to be able to begin to be a society that works in a different way, not only in this field. As I said before, this paradigm is the paradigm of the new public health, but we don't do it.
Thank you for this. Second question I have, I really would like to ask you, because I have heard you long years ago that you mentioned the same term, precision public health. I have heard you in Athens saying that in a conference. So I'm not quite satisfied how we work on precision public health. I don't think that the people understand the term precision public health, because we know precision medicine, for example, which is a very different term. But what do you think about precision public health, maybe in One Health, but in general as well? As a term, can it move a little bit forward? How can we do the people to understand about that, especially the governments, because they don't really understand?
Well, the term precision, again, is being used in so many different ways. Precision medicine, precision prevention, precision pharmacogenetics, precision public health. Of course, what's behind all this is that today we can match the rhetoric of what we want to do in medicine, in public health, with hard data. I make a parenthesis, though, before I continue this, to say this. When I started working in the 80s in WHO, it was the time when the global strategy for health for all had been launched. And target number one of that strategy, which is fantastic for that era, was equity. Equity was the number one. But do you know what was happening at that time? There were member states of ours who were saying, "Oh, come on, WHO is becoming too socialist. That's a good rhetoric, but can you back it up with data?" Now, we all thought that when we will have the data, then the world will change. Well, I have to tell you, we are drowning in good evidence on the social determinants of health and the root causes of equity, of inequalities. But this has not followed into serious action, which means that data alone will not change what we wish to do. Nevertheless, precision means today that it becomes more individualized. So precision prevention and public health allows you to be able to identify parameters in a person, parameters in the environment. I mentioned exposomics before. You can think of genomics and epigenetic influences that give us today the possibility to measure and to follow the health of populations and individuals in a way we could not have done it before. So, if you like, the word precision, you know, we are colleagues in the same field. Our problem is that we're not neurosurgeons. And we're not specialists in cardiosurgery. I mean, the glamorous specialties. Public health does not have the status that other specialties have. So today, there is a wealth of new information that is brought into the domain of public health that will elevate also the potential of public health to convince and to make a difference which goes beyond the rhetoric of changing lifestyles, changing policies, creating healthy environments, and so on. And of course, this applies to this domain of One Health in every respect, especially when it comes to individuals and their vulnerabilities.
Okay. But I just want to emphasize that I can see that if governments understand the precision of public health, then they will specify the measures that they need to take in a local way or in a regional way, because they don't specify. They just copy measures from other countries or other areas. I can see that in smaller countries that they do it. So when you want to solve one health issue, for example, in Patras, it's different than when you want to solve it in Denmark or somewhere else. So they don't specify, they don't work at a local or regional level, so to work. They just take ingredients, guidelines from others. And I don't really understand how it can work in an issue like One Health, for example, or even, you said obesity, for example. We are not talking about obesogenic environment, which is specialized in an area, for example. So that's why I was asking you about precision public health in a very local or regional level.
Well, just to summarize, precision has strengthened the hand of everybody who cares about both the health and the promotion of the health of individuals, but also whole populations. The drama in our country is, for example, we have never measured inequalities in health. I mean, you mentioned Denmark, I will mention Britain or so many countries, Sweden and so on, the Netherlands. They measure inequalities. They know what is going on. And they have a basis which is strong evidence to then design appropriate policies. We don't. I mean, after all these years, we do not have any idea of inequalities in health in Greece.
Yeah.
And who pays the heaviest burden of that?
Yeah. I have a question from internet, last question I hope, that is Matthew Sotieno. It says, how can we strengthen collaboration and information sharing between human health, animal health, and environmental sectors to improve early detection, prevention, and response to emerging public health threats? Your opinion on that?
Well, an important step, which goes back to what I said before, also starting from the awareness building, sensitization, and another key word is consensus building amongst those who you expect to become the supporters of what you are promoting. I have, and this is not a criticism per se, but even amongst people who work in the public health field, there is no common understanding of the practical application of the terms we are talking about. So, in my view, you cannot mobilize a society, you cannot convince a politician to change the way they are working. Or, very often, do you know what happens? You may convince someone to do a project, and you may get them to work, because especially if there is funding, then it's all nice, let's do it together. But this doesn't change the situation. We need to get into the same room, not only you who probably you are working in the field of public health, but I would like to see in this case, public health, medics, communities, vets, environmental people, architects, urban planners. We need to create environments and platforms to have this common understanding of what we are talking about. I believe strongly that the big umbrella is the Sustainable Development Goals. It's the best point of reference for everybody. And through that, and through good diplomacy, create mechanisms and policies that can support this work, not in individual projects, but in a holistic way for a country, for a region, for a city.
Thank you very much, Professor Juros. No other questions. Thank you very much. Thank you for your presence. Okay, let's move to next speaker, who is Panos Kalagis, Assistant Professor, Faculty of Health and Medical Sciences, Globe Institute Section for Evolutionary Hologenomics, University of Copenhagen, Denmark. And he has a keynote lecture about double-edged viruses, bacteriophages as promising antibacterials with a hidden side. Thank you, Professor Kalagis.
Okay. Thank you very much, Professor, for the introduction. First of all, I would like to thank you and the organizers for this invitation. It's a great pleasure for me to be here. And today we're going to talk about viruses, but probably not the viruses you have in mind. We're going to talk about bacterial viruses. Before starting, I would like to highlight the importance of collaboration also in our field, in microbiology work. Having myself an experimental microbiology, a wet lab background, I believe that there is a big value of utilizing data in what we do. And I'm talking about big data. At the moment, I'm working at a computational biodiscovery group with a lot of bioinformatics involved. And I'm mentioning that as an example. This is a case in point example of our latest study, which is going to be soon published by Nature Microbiology. So we had the opportunity to screen over 3.5 million bacterial assemblies of over 1,000 species, and we detected over 100,000 unknown viruses. So there's definitely a high potential there, and that would not have been possible by doing only experimental work. So having said that, I would like to set the scene for today's talk, and I'm going to mention the antimicrobial resistance problem, and we're going to see how bacteriophages, bacterial viruses, can actually help us in that approach, mentioning some examples in animals, in humans, and also what we need to be aware of while we try to apply these bacteriophage therapy strategies. And before summarizing, a few words on the regulatory framework that's in place at the moment in the European Union. So, without further ado, we are all in this room quite familiar with the One Health approach. It was said very nicely before also by Professor Chouros. Here we can see that it is of utmost importance to collaborate and bring together all these principles and work in an integrated manner in order to combat these challenges, these health challenges that go from animal, plants, humans, and also their environment. So talking about antimicrobial resistance today, we have an idea about antibiotics, of course, and how they work. So in fact, they are actually disrupting some sort of specific cellular component, like the cell wall, for instance, or some function, some biosynthetic pathway of the bacteria. And this antimicrobial resistance, the infamous AMR, is developed upon the administration of antibiotics in a bacterial population, and some of them manage to survive due to some AMR genes that they carry, and after proliferating, they can spread them horizontally or vertically, and then, of course, create these AMR populations and AMR clinical problems. So, having said that, if we take a look at some numbers, unfortunately, they are not very nice. We can see that the lower respiratory infections and the bloodstream infections rank quite high, and there's a high death toll in these problems at the moment. And based on the One Health approach again, we know that there is a lot of antibiotics that at the moment are administered in livestock, and I'm mentioning here some examples. And of course, it can vary across different countries. But if we take a look at the future perspectives, by 2050, unfortunately, we can foresee, and according to the models, over 10 million annual deaths caused by antimicrobial resistance pathogens, even more than anticipated deaths by cancer. So there are alternative approaches, and bacteriophage therapy, the application of bacteriophages or bacterial viruses, is one of them. Having said that, let's go and see what are bacteriophages. So in fact, bacteriophages are viruses that infect bacteria. You can see them here landed on the surface on the bacterium in this graphic. And if we want to be a little bit more realistic here, you can have a Vibrio pathogen, and the virus is sitting on top over there. So these are basically prevalent and available all over. I mean, in the nature, in any part that in any niche that we can imagine. How do we find them and how do we isolate them? For instance, for clinical pathogens, we first need to establish a biobank, a bank of clinical pathogens. And then we can sample in different areas in order to detect them and find them, the phages that are able to infect the pathogens of interest. So starting for the hospitals, for instance, we can collect bacteria from clinical cases, purify them, store them, and then create the digital files, and then go sample in, for instance, wastewater treatment plants or hospital waste, enrich these samples with the appropriate nutrients for the bacteria to grow, and then add the bacteria of interest, the target bacteria, in order to detect if there is any activity, any bacteriophages, any bacterial viruses activity against this bacteria. On a good day, a result should look like that. Here, the spots that you can see, the small transparent spots, signify the presence of bacteriophages. But I think visual is always a little bit more helpful. So let's have a look at the life cycle of a T4 phage. This is a very well-known and well-studied bacteriophage that infects E. coli. So we can see the phage that is landed on the surface of the bacterial cell, and then it actually recognizes properly the proper host using the receptors of the bacteria with its tail spikes. And after it's landed, then it injects its genome inside the bacterial cell. And when that process happens, the bacteria is taken over by the bacteriophage. So the metabolic machinery of the bacteria is now doing what the bacteriophage wants to do. After replicating the genome of the bacteriophage, and after producing the proteins that are necessary for the new virions to be assembled, for the new phages, then all this is put together, the new virions are assembled, and when this process is completed, then the entire cell is going to burst, it's going to disrupt, and the new virions will be released to the environment, and they are now ready to infect more cells. So on a more laboratory-based perspective, if you take a look at this video, you will see a glass slide, and these dots are the bacterial cells, and you will see them disappearing slowly because we have added a high concentration bacteriophage. So they are kind of popping and leaving the frame after, this is a time lapse of 4 hours. And if we want to take a closer look on one of these events, we will see here that this particular bacterium over here is going to pop, is going to explode and it's going to release the new virions in the environment. So let me push the play again. Yeah. So it kind of puffs and disappears. And from a Petri dish, from a more macroscopic perspective, we can see here a Petri dish which contains a bacterial loan, and then you can see that we have added 2 spots of high concentration bacteriophages, and after 24 hours, you can see the clearing zones, which actually mean the inhibition of the bacterial growth on this particular Petri dish. So this is actually the visual representation of how phages work. So having said that, I need to mention that phages is not something that we found yesterday. Phages were discovered in the beginning of the previous century by Frederick Twort and Felix Derel, and actually Derel, in his first paper, he mentioned them as some invisible microbe, which is antagonistic to dysentery bacilli. At that time, phages were very much in play against bacterial diseases until the discovery of the antibiotics. And especially after the Nobel Prize was awarded to Fleming and his colleagues for penicillin, the Western world kind of abandoned the idea of bacteriophages. Not the same happened to the eastern states of Europe, though, and we're going to mention something later in the presentation. Phages were still, though, in play in a lot of research, and they were also participated in some of the work that led to some other Nobels later on. But after all, let's see why phage therapy. So phage therapy is the application of phages against pathogenic bacteria, and has 4 main benefits. The first one is that we can tackle antimicrobial resistant pathogens. When antibiotics don't work, phages are able to work. Of course, this is not a generalization. It needs to be checked, but there is a good chance. The phages are extremely host-specific. They have a mechanism of detection and recognition to a species level or even to a strain level to the pathogen that they want to tackle and infect. Additionally, they can actually control their own population based on the availability of the host. So they can be self-propagated or self-limited according to the availability of the host. And last but not least, it's an environmental approach since phages are already part of nature. Of course, they can be genetically modified, but also there are many natural phages that we can find in all niches in the environment. So having said that, let's see how phages are applied in animals. By animals, I will focus on fish, on aquaculture. I've been working a lot on bacterial pathogens in aquaculture. And I must say that aquaculture is at the moment the fastest growing animal production industry in the world. It has been growing over the last decades steadily. And today, on a global level, each of us consumes, on average, more than 20 kilograms of aquatic animal products. This is expected to increase. Aquaculture has taken over captured fisheries. It's more than half, more than 50% at the moment. And it's expected to grow more because by 2050, our world is anticipated to reach about 10 billions. So there's a lot of extra need that needs to be fulfilled. However, there are a lot of diseases in aquaculture that can hamper productivity and biomass. So the intensification of the production to meet these increasing demands, the diversification of the production, there are a lot of new species that are introduced whose biology is unknown, and there are a lot of new pathogens that might come into play. And also the increase of temperature that we have noticed across different aquatic bodies favors the outbreaks of pathogens. In marine aquaculture, especially Mediterranean aquaculture, the majority of losses happens in the larval stages where they are more fragile. So the point here is that the small larvae of the fish are fed with live feeds because they are highly nutritional food. But the problem is that these live feeds can harbor inside them potential opportunistic pathogens, such as Vibrios. So while these live feeds are fed to the larvae, we typically can expect in future stages some outbreaks which have originated from that point. So having said that, bacteriophages can be used as smart disinfectants in the live feeds. So in this particular research, we have used bacteriophages, a cocktail of 2, to combat the bacterial populations, the native Vibrio, in live feeds of Artemia salina. And we actually saw that it was possible to decrease them by 1.3 log after a few hours of treatment. In a different stage of aquaculture production, we're going to fish larvae. We have also treated fish larvae while they were still feeding on their own yolk sac with bacteriophages against some Vibrio pathogens, Vibrio anguillarum. Then we used a broad host range bacteriophage. It's called KVP40. Here, you can see, although the overall mortality is quite high due to the bad quality of the water, you can see still that the phage-treated condition, which is this one down here in all the graphs, showed the highest survival, and that's because they were able to significantly remove the native Vibrios that were actually able to cause potential losses later on. And in a different system, here we have used rainbow trout and the pathogenic strain of Flauobacterium sacrophyllum, which is a common pathogen in trout. The cocktail of phages was D22 and FPV4, again in larvae. Different routes of administration were used, phages in the feed, phages intraperitoneally, and phages in the water, in the tank where the fish were swimming. The intraperitoneal application was able actually to give an additional 25% of survival after a few days of control. And this is further enforced on an additional experiment that was done later, where again some larvae a little bit later in their growth stage were used, and then the significant reduction was much higher. That's why the survival reached about 85% in the high doses. So we have seen that in animals. There are a lot of studies also in poultry and swine, and quite extensive literature on that. So let's have a look at the humans as well. So that's much closer to us. First of all, when we're talking about phage application or any intervention in human health, we need to check safety and efficacy of that procedures. So the PICO approach, which stands for population, like who's going to receive the treatment, typically that are people who suffer from AMR infections, or even not AMR infections, but definitely the standard of care treatments have failed. Intervention is phage therapy. The comparator can be a placebo or an antibiotic or anything like that. And then, of course, the outcome can be, of course, the safety of the application, and then the efficacy, what was the difference in the symptoms, in the quality of life, or maybe in the biomass or bacterial eradication in the patient. So the bad news about phage therapy is that, unfortunately, we do not have randomized clinical trials, a lot of them, that produce good data. We have a few of them, and those few didn't give promising data so far. The problem is that bacteriophages have been used in these trials as a one-size-fits-all treatment. And that's not how phages should be treated, because it's not a chemical agent, it's a living form. The good news is that we see an increasing number of individual successful phage therapy cases, what's called compassionate application, and it's generally considered to be a safe approach via different routes of administration. To show a little bit more evidence on that, we're going to go through a study which was published last year by some researchers in Belgium in collaboration with the Eliava Institute of Bacteriophages in Tbilisi, in Georgia. And these people, coming from the Queen Astrid Military Hospital, University of Leuven, and Cienzano, did a retrospective analysis over the past, over about 100 cases of bacteriophage therapy, and conducted some conclusions. So the research was focused on European cases. We are talking about 35 hospitals from 29 cities in 12 countries over a period of about 15 years. So the retrospective analysis actually wanted to respond to how often personalized phage therapy produced a positive clinical outcome, and what are the functional relationship in these cases. So before moving to that, as I promised before, one of the partners of this study is the Eliava Phage Institute of Research. So I don't know how many of you have heard of this, but this is a center the oldest center in the world that does phage therapy, and it does it today as well. These are a few phage products that are produced by these scientists over there, and they even have a local treatment package. So some people from Europe or United States or the Americas have actually visited the center, and thousands of people, whenever they have a very highly risk or high risk AMR that is impossible to cure in the local countries, and they have been there to receive the proper treatment. And according to the information that these people publish, a lot of the results have been positive. But back to the study, what it was conducted after the analysis of the phage therapy cases was that we do not aim to replace antibiotics. We actually aim to enforce the action of antibiotics. A lot of these treatments were done together with antibiotics, about 70%. The top pathogens that were targeted were Pseudomonas aeruginosa and Staphylococcus aureus, about 90% of the cases. The most important finding of this study is that about 77% of the studied cases showed improvement, statistical improvement, according to the medical doctors, in at least one of their symptoms. And about 61% of these studies showed complete eradication of the pathogen that caused the problem. So there were about 43 batches of 26 phages for the treatment of these patients. And it was mostly synergistic activity that was observed between phages and antibiotics rather than antagonism. There was only one case of antagonistic behavior. Additionally, phages could be also neutralized after some days by the serum. There were cases that this could be observed. But this actually depends a lot on the phage and on the patient. And then, of course, there is resistance against bacteriophages. And it also occurred in some of the cases of this study. We're going to talk about that in a bit. And the adverse effect, fortunately, were not so important or were not so significant, at least the ones that they observed in these particular studies. But the question still stands. We do not have randomized clinical trials, and we need them quite a bit. On that note, I would like to mention that the European Commission earlier this year announced a very spot-on action on this particular case. They announced a 45 million grant dedicated to the safety and efficacy of randomized clinical trials for phage therapy in humans. So we are expecting over the next years to hear more about that, and it's really an exciting development. So phage therapy might have sounded quite nice, but it's definitely not a silver bullet. There are certain things that we should be very careful, especially due to the fact that they are living forms. Viruses are there. So in the video that I showed you before, you probably didn't notice because they're very small, but if you see these points over here, these are bacteriophage-resistant colonies. That means that we have development of resistance when we have together phages and bacteria. And what I mean by that is that the bacteria can actually summon a very versatile suite of different ways to circumvent the infection. It could be by preventing the viral attachment. It could be by preventing the DNA injection. It could even be to destroy the DNA after it's injected inside the bacterial cells. This probably you have heard the infamous CRISPR-Cas systems. That's how it works. It's coming from the bacteria. And also, it can actually commit an altruistic action. It can actually commit suicide, the bacteria, the infected bacterium, in order to protect the rest of the population. So there is quite a lot of elaborate activities. The good news for us is that it doesn't come for free. Bacterial resistance against bacteriophages is closely connected with virulence cost and fitness cost. We have seen that for fish pathogens, not only fish pathogens, it's the literature is full of such references, but we have seen that the resistant to bacteriophage bacteria are not as virulent as their wild-type bacteria. And also, they cannot grow as fast, and they cannot utilize as efficiently the same metabolic substrates in order for them to grow. Even in the study that I mentioned before, in the humans, the scientists observed that there was a decreased virulence of phage-resistant colonies in specific patients, and there was even the unexpected event of resensitization to some antibiotics in one case upon administration of antibiotics. So what we have here is that when a phage infects the bacterium, it will lead to the death of the bacterium. But fortunately or unfortunately, that's nature, this is one scenario. The dynamic interactions between the phages and the bacteria don't stop here, because the lysis of the bacterial cell is what we want for phage therapy. But of course, there is another pathway that's called temperate or lysogenic pathway that is employed by the temperate phages. So in this lysogenic pathway, the phage injects the DNA in the cell but doesn't kill it. It actually integrates its DNA to the bacterial chromosome. And when that happens, we have, let's say, some unexpected results. So that stops being a parasitic relationship and it starts becoming a symbiotic relationship. The example of Streptococcus pyogenes, for instance, it's a project that we are currently working a lot. So this pathogen is full of mobile genetic elements, meaning mostly phages. These phages are responsible for the very high homologous recombination that takes place among different Streptococcus pyogenes, and also a lot of accessory genes that are present inside these bacterial genomes. So when they are present there, they actually exist in the chromosome and they render virulence and fitness properties to the host. An example here, we have constructed two large databases. One is based on RefSec, it's very analytical, about 2,000 genomes. The other one is it's more unprocessed, let's say, about 50,000 genomes based on sequence read archives. And we have seen a very high diversity and very high amount of mobile genetic elements. And when we did an analysis on the metadata that are available from these genomic sequences, we found out that the prophages, when they are at least one present in the genome, and you can see this is 0 and this is at least 1, in the cases that we have at least one, the clinical pictures are more connected with invasive manifestations, invasive Streptococcus pyogenes clinical pictures. So we found ourselves some phages for Streptococcus pyogenes. That's two of them, SPI1 and SPI5. This is the genome, 40 kb, more or less, and these are the guys over here in electron microscopy. These are two more, different. They look similar in morphology, but they are different. Their genome also quite different. Are they good for phage therapy? The answer is no. We have developed in the lab, in the group, some tools to help us understand if it's a good candidate for phage therapy or not. So in this particular case, when you have the genome of the bacteriophage, you can get a prediction on the suitability of the phage as a phage therapy candidate. So if there are AMR genes present, if there are virulence genes present, is a negative response. And another tool on the same lines is this phage clouds, which actually can connect our newly discovered phage genomes with the existing diversity of phage genomes. And then if we zoom in, we get the networks very clearly. And then if we click on this dot, which represents actually a phage genome, we can get more information about the date, the accession number, the species, etc. So having said that, let's have a look at the regulatory framework and where we are today on this matter. As I mentioned before, there is already a project, or a call, sorry, by the EU, which is announced, it's closed, and we're expecting to see more on that on the coming years. At the moment, phages can be faced as non-drugs or drugs. And the non-drugs are managed by the EFSA, European Food Safety Agency. The drugs are managed by the European Medical Agency. There's no specific framework for the EFSA, but we know that phages there are considered additives. The point is that there are no health claims in this case. So we're talking about health. We're talking about European Medicines Agency. A couple of years ago, end of 23, there was a framework announced about the veterinary application of bacteriophages in EU. Not much have been done so far, but I think we are going to get there quite soon. For humans, there's nothing established at the moment. However, there has been a lot of discussions, a lot of open consultations, and there are a lot of countries which are individually encouraged to develop initiatives for phage therapy. And I believe that we're going to see something on that quite soon. So, at the moment, the scientific and regulatory issues that are precluding the approval of phages by EMA are mainly that the data that are generated are not coming from well-designed random clinical trials, and these are not very convincing for EMA because they have their own set of rules that needs to be followed. Then we also have a lot of small studies, but they have a big variability and nothing is very cohesive, so cannot be judged as vividly. And we know that phages are generally considered safe. However, there is this possibility of mobility, genomic mobility that I also showed before, that needs to be examined on a case-per-case basis. And these phage therapy medicinal products need to be manufactured on a GMP environment. And that's actually a very tedious process, very expensive process, and it's going to take a long time. And as long as the regulatory, legislatory framework is not well in place, prevents a lot of, let's say, stakeholders from investing a lot of time there. I need to mention that we have few news which are encouraging, and that has to do with a couple of companies at the moment. One is Biomex, has 2 products in clinical trials, one for diabetic foot and the other one for cystic fibrosis. And then there is another startup which was recently raising a good amount of money to develop phage therapy in veterinary applications. So there's a lot of activity which is quite promising for the future. And I'm going to summarize and close this presentation by the One Health approach from which we started. So bacteriophages at the moment can be used across all areas of One Health approach. Humans, we are talking about prophylaxis during outbreaks. We are talking about vectors to deliver vaccines, talking about modulating the microbiome in the microbiota, and also some biodefense approaches. For animals, we are talking about veterinary interventions, as I mentioned, and also replacement of antibiotics, or let's say enhancement of antibiotics. And last but not least, the environmental approach has to do with food safety, can be even decontaminating the slaughtered animals, disinfecting the water, and then also applications in the field in aquaculture and agriculture. So we want, preferably, to have lytic phages, as I showed before. We definitely want them to kill efficiently the bacterial hosts without leaving remnants behind. And we definitely need to fully characterize these phages before moving, because we definitely don't want any kind of safety and efficacy issues afterwards. With that, I would like to thank you all for your attention. I would like to acknowledge a lot of great colleagues that we have been working together, and I would be very happy to take any questions and comments. So, thank you.
Thank you, Professor Kaladzis. It's really extensive analysis of bacteriophages. I really love that. Any questions? Yes. Uli? Yes, first. No. Wait. Just give me a second. Please name yourself and...
Hey. Okay.
Yeah.
So my name is Uli Klumper from Technical University of Dresden. Love the talk, and I also think that phages have great potential, but it's probably not all there yet. So what I'm wondering is, I strongly believe in the applicability for like short-term treatment in the human sphere or for animal disease, but you were also considering feed additives, so basically long-term treatments of the same communities. With regards to ecology and evolution, wouldn't you expect that there you have major ecological consequences of the microbiome and by thus lose treatability rapidly and would have to redevelop new cocktails consistently?
Thank you very much for the question. The point is correct. It is much more efficient to apply in, let's say, batch cultures. So the approach that we did in smart disinfectant on live feeds was because it was very short in duration, only a few days. So that's how it's, let's say, maximizing the efficiency. On the other hand, though, cocktails are much more preferable because they actually complement the activity of many different bacteriophages, and that could actually avoid or prolong a lot development or signs of resistance. But even if that happens, what we have seen at least is that as long as the pressure of the phages is removed from the system, the bacteria have the tendency to revert to their wild types. So I wouldn't consider that as a major ecological, let's say, issue. Of course, a study needs to be done in advance because we need to know what we are doing. But at the same time, the property of bacteriophages to control their population according to the availability of the bacteria in the environment would also benefit them in their activity for this way.
Okay, thank you.
Welcome.
Ραφαήλ, πρώτα στην κυρία και μετά.
I just wanted to ask you, how long do you think that it will take to engage the the big pharmaceutical companies for treat the health problems about the lack of antibiotic efficiency and lack of new antibiotics being developed because it's not economically interesting for them? So how do you see that part?
Please, can you name yourself, please?
Sorry, Elizabeth Valério from Portugal.
Yeah, thanks for the question. Well, it is true that the net present, let's say the profitability or the return on investment for all pharmaceutical industry in antibiotics is definitely not attractive, let's say. So at the moment, what happens is that compassionate therapy kind of prevails. So it's a more, let's say, personalized approach of this particular medicine. However, I believe that the clinical trials that are in place for issues that have very broad interest to the audience, such as, for instance, the diabetic foot that I mentioned before, is sort of combined with clinical pictures that actually are particularly interesting for the pharmaceutical companies. I can mention, for instance, we are all probably heard of Novo Nordisk, which is a Danish company that developed this medicine to manage the obesity from diabetes and stuff like that. So this is a particularly interesting market, and I believe the companies might be lured into investing that if they see the results and if they see the stability and efficacy from randomized clinical trials, because there's always a market share to be claimed by them as long as there is a reliable product and a legislatory framework to follow. I think what discourages them a lot at the moment is that the unclarity is quite high. So investing a large amount of money and also like 10, 15 years to sort of go the pathway, it's rather precarious. So they prefer to go alternative pathways or more attractive areas like oncology or something like that.
Thank you. Yes.
Roberto Spurio, University of Camerino, Italy. Thank you for the talk, very, very interesting. My question is, if there is any research going on trying to genetically modify some bacteriophages, some specific bacteriophages, trying to avoid or bypass what we could expect as a response from bacteria, for instance, the CRISPR-Cas system. So since we know the basic biology behind the response of the target and the basic biology of bacteriophages, which is relatively simple, is there any project aiming to modify some of these features of the bacteriophages?
Yeah, great question. In fact, there is literally an enormous amount of work on this area. There are actually publications that have actually developed genetically modified phages that were even applied to patients to treat them against specific infections. And that happened because, for instance, when I mentioned you have a temperate phage that might integrate in the bacterial genome. So what they did is that they removed the genes that do that, and actually they produced a modified X-temperate, but now lytic phage that could actually infect and kill the bacteria. So there's a lot of activity in this, across these lines. And on top of that, what is also particularly famous and, let's say, growing a lot at the moment is to utilize specific genes of the phage, for instance, enzymes that are called endolysins, and the endolysins are responsible for basically disrupting the bacterial cell wall. You don't need the entire phage, you just need the enzyme that will do the job and disrupt the bacterial cell wall. So what's happening is that there's a lot of genetic engineering in order to isolate this, express them through vectors, produce them in high quantities, and even enhance their efficacy, and use actually these enzymes as antimicrobial agents without having to get the entire phage. And this way sort of gives you a little bit more flexibility when it comes to regulations, again, because it's not a living form anymore, it's just a protein. So yeah, definitely, it's a field that it's growing a lot.
Okay, I think we're okay. Oh, thank you very much, Professor Kaladzis.
Welcome.
Now it's a time for coffee break. Thank you very much. I will wait you at the next session, 11:30.
We will bring together expertise from human health, from veterinary science and molecular biology to explore how deeply interconnected our ecosystems are and how these connections shape both the emergence and the control of infectious diseases. So we have three distinguished speakers, and let me move to the first. It's Professor Chrysostomos Dovas. He's professor of molecular microbiology at the School of Veterinary Medicine at the Aristotle University of Thessaloniki. And he will speak on avian influenza virus, evolution, pandemic potential, One Health perspective. So please, Professor Dovas, he is not here in Patras, but he is in Thessaloniki, online. So the floor is yours.
Thank you for the invitation. I'm going to discuss regarding H5N1 avian influenza virus, about its evolution during the last 30 years, its pandemic potential now, and the One Health perspective. We have to remember a few things regarding influenza avian virus. Influenza virus A consists of 16 different antigenic types of HA and 9 antigenic types of NA. And according to the HA-NA combination, we have the name of the virus. Usually it's, for example, H5N1. And from the 144 candidate subtypes that could occur in nature, 131 have been identified, mainly in bird populations. The virus genome is segmented also. It consists of distinct segments, genomic segments. And so in this regard, he is prone to recombination. And all these subtypes, they exist in aquatic wild bird reservoirs. And AIVs in these ecosystems cause localized infections in the respiratory and gastrointestinal tracts, and they are generally innocuous. And they can maintain a fitness advantage with minimal genetic change. So they don't change genetically in these environments rapidly. They are adapted for mucosal replication and transmission through the fecal-oral route. And their dispersal in wetlands is easy due to the dispersal of the virus in water and due to the aggregation of birds. So in this regard, we have very frequent infections. And also we have very frequent recombination events via reassortments. And so we have chimeras of viral genomic segments and novel genotypes with novel adaptational fitnesses. So in this regard, we have generally a very high dispersal of these viruses in aquatic birds. These viruses, they don't show any signs, we don't have any signs of disease in these aquatic birds. And on the other hand, we have regular spillover of viruses to poultry through direct and indirect contact. And later on, we have these subtypes from the bird reservoir that they can acquire adaptive mutations through serial passages in mammalian intermediate hosts. And these passages can enable cross-species transmission and infection of novel hosts like human after adaptation and mutational changes. Genomic segment recombination via reassortment in mammals is also important because we have rapid acquisition of entire genes for adaptation to new host species. Most influenza genes contribute to host rates adaptation, and in this regard, co-infection of a cell with an AAV virus, and a mammalian-adapted virus can yield reassortant progeny with novel gene constellations that can enable cross-species transmission and immune evasion. So AAV virus and human influenza virus co-infection in a mammalian host, like pigs, for example, or fur animals, could produce virions with human-adapted segments and AIV-derived antigens like hemagglutinin and neuraminidase. These antigens can evade human immunity in the human population. We have different host specificities of these viruses. This avian influenza virus is preferentially attached to sialic acid bound to galactose via a tooth-ring linkage, whereas human adapted viruses, they attach to different sialic acid receptors in the upper respiratory human tract. On the other hand, we know that occasionally humans, they are infected by avian influenza viruses because they do possess avian receptors in the lower respiratory tract. So in this regard, human infections with AAVs is limited to individuals with intensive direct contact with the infected animals, and also the presence of heavily contaminated environments. So the virus is not sustainable regarding human-to-human transmission. And on the other hand, when in cases we have infected birds and slaughtering, defeathering, evisceration, or playing with infected birds, then we have a high probability of human infection because the virus can reach the lower respiratory tract and infect susceptible cells. And on the other hand, because the infection is in the lower respiratory tract, this is a heavy infection with high mortality in human. So in order for this virus to adapt and become a pandemic virus, it has to adapt and recognize receptors, human receptors in the upper respiratory tract. And if this becomes a reality, then this virus will transmit easily by airborne transmission, and on the other hand, its lethality will be significantly reduced because it will be an infection of the upper respiratory tract and not the lower respiratory tract as it is now regarding avian influenza viruses. Now, the big question is whether this H5 hemagglutinin can adapt, can mutate to sustain human-to-human transmission. Is it possible? And this was answered in 2012 in 3 different experiments in 3 different papers, where serial passaging of H5N1 in ferrets changed the receptor binding preference towards human sialic acid receptors and enabled airborne transmission between ferrets. So H5 can mutate and can become airborne transmitted. And this was facilitated not only by the 2 reversed engineered mutations that were performed in this virus at that time, but also after that they also made passages in ferrets and mechanical passages of the virus. And then after a few passages, the virus became able to transmit via respiratory droplets. And this is a perfect example how that passages in mammals can facilitate adaptation of the virus towards airborne transmission. So not only it was found by these experiments that not only in the viral receptor domain, binding domain, we have to have mutations for the virus to become pandemic, but also there are other regions of the HA that have to be mutated, and these are somewhere here that they are responsible for stabilization of the HA to the low pH of the human epithelium because the low pH of the human epithelium causes an irreversible change on the conformation of HA and renders the virus non-infectious. So we have to have additional mutations for the virus to become stabilized, and then this is a further adaptation towards a pandemic strain. On the other hand, we need a neuraminidase that has a functional balance with the respective HA. So this functional balance of activity between HA and NA promotes efficient viral entry, replication, and release in human airways, elevating the virus's pandemic potential. So not only HA is important, but also it has to be paired with a compatible NA for optimal infection and species barrier transmission. On the other hand, other genes also they are adapted. Most of them are the polymerase, the genes involved in the polymerase complex. So the virus polymerase has to be adapted in the human cells. And finally, last but not least, is that these viruses have to counter the cellular innate immune response, human response, by encoding interferon antagonists or masking pumps. So influenza viruses, they are adapted, having mutations in the polymerase and the NS1 and NP. So more genes have to be adapted in order to evade human innate immune response. In this regard, reassortment is very important because by reassortment between a humanized virus and an avian influenza virus, this reassortment event in a different mammal like a pig or a fur animal could rapidly produce an adapted virus that has different genes that they are adapted in human and antigens like HA and NA, that they are different antigenically and they are derived from avian sources. And then we have the perfect combination for a pandemic strain. On the other hand, this is an interesting slide that shows the different steps towards a pandemic strain. So mostly in the beginning, we see PB2 adaptations in different hosts that are infected, mammal hosts. And then we see a shift in the HA binding adaptation towards mammalian sialic acid receptors. And then we have to see an HA-NA balance through mutations of NA or recombinations towards different NA. And then virulence stability of HA and evasion of restriction factors regarding the innate immune response and further adaptation through passaging in humans, probably silent passaging in the beginning, and then finally an adapted virus that can be easily transmitted airborne from human to human. In this regard, you can see a possible scenario for these adaptations, which involves an intermediate mammal and passaging in this intermediate mammalian host, which also could involve recombinations with avian and human viruses, and then a final jump to humans and adaptation in humans through serial passaging. Most of the non-pandemic viruses have, all three of them, they are proven that they have been evolved through reassortment events from mixing avian and pre-existing human influenza genes in mammalian hosts, whereas for the pandemic strain of H1N1 in 1918, the Spanish flu, the common idea is that they have derived, this virus was derived directly from an avian source that was probably adapted in a mammalian species and then transferred to humans. Let's see the interesting issue regarding the pathogenicity of these viruses in poultry. We have low pathogenic avian influenza that causes no or little signs of disease in poultry, whereas highly pathogenic avian influenza that causes very high mortality rates in chickens. Sometimes the low pathogenic avian viruses can become established in poultry, and then they mutate in the HA gene, and only H5 and H7 subtypes can mutate into highly pathogenic viruses. So the name highly pathogenic avian influenza virus H5N1 becomes from its ability to cause very high mortality rates in chickens, 100% in a few days. So low pathogenic avian influenza viruses and high pathogenic avian influenza viruses H5 and H7 are notifiable to the OIE, and the wild birds have the potential to transmit the viruses to poultry, and then these viruses, they circulate, they mutate into high pathogenic avian viruses, and they can also sometimes go back to the pool of the wild birds and recombinate and evolve further on. These mutations through passaging in poultry involve the insertion of several lysine and arginine residues in the cleavage site of HA, and we have mutations in this site here that they change the HA cleavage for virus infectivity. The HA has to be cleaved in order for the virus to infect the cells. And in case we have mutations of insertions of these residues in the cleavage site, then the HA is cleaved intracellularly by ubiquitously expressed furin-like proteases, facilitating systemic replication in chickens and high mortality. Now, this is important because probably this phenomenon has to do with the fact that the multi-basic cleavage site of HA is associated with viral dissemination in mammals, extending to systems beyond the respiratory and digestive tracts. That means that probably the multi-basic cleavage site contributes to the capacity of these viruses, H5N1, to induce neuroinvasive disease in mammalian hosts. And we have seen this type of infections recently in mammals throughout the world. This virus emerged in 1996 in China. Well, it was effectively contained after slaughtering 1.5 million poultry. But the first time in history, we had 18 human cases and 6 fatalities. So this was a very important event. And the virus later on re-emerged in 2001 and 2004. The emergence of these reassortants were due to the fact that the initial virus of 1996 went back to the wild fauna of aquatic birds and recombined and produced new recombinants that were very adaptive and this to birds, and they were very easily dispersed with migratory birds, and this genotype G was very efficient in infecting migratory birds, and then this virus was able to be transferred throughout the Eurasia and Africa. Later on, we had novel recombination events that enabled the virus to produce another panzootic in 2010, 2012. Later on, in 2014, we had a third panzootic with the virus recombining again and producing a novel genotype that kept only the H5 from this initial virus that emerged in 1996. Only the HA was, the H5 was maintained and all the other segments were changed, including the NA gene. And finally, in 2019, we have the current panzootic, which is huge, which is a phenomenon. Again, we have the same H5 that emerged from the initial avian influenza virus that has evolved, but we have a novel constellation of genes, including a novel neuraminidase. This recombination event happened in 2020, somewhere in the Mediterranean, and then this virus, which has the H5 clade 2.3.4.4b H5, carries a novel neuraminidase, a novel N1, different than the old one, with a long stalk. Now, this long stalk is important because this long stalk can effectively cleave decoy receptors present in airway mucus and it can increase virus transmission between mammals through contact and airborne routes. So this is a further step towards a pandemic virus regarding mammals. This virus effectively infected migratory birds and was migrated towards different continents in US, where we have rapid transmission in wild birds, recombination events, novel genotypes also appearing, and these are infection events in wild fauna in 2020 and up to 2024. Regarding this new panzootic virus, and this is the recent, the last 2 months, the detections that were that have been reported in wild birds. We have a wild bird surge in Europe from 6th of September up to 14th November 25, last few weeks. We have an unprecedented high level of highly pathogenic avian influenza virus infections in wild birds in Europe. On the other hand, we have a geographic location increasing regarding mammal species affected by this virus in the current panzootic throughout the world. We have many different mammal species being infected during this current panzootic. In USA, 647 different mammals. We have neurotropic disease in different mammalian species. 3,487 dead sea lions in Peru. Different mammals, ruminants infected in USA, cats throughout the world, cat infections, lethal, and mostly we have mammal infections that are due to predation of sick individual animals or scavenging of carcasses. However, in this regard, solitary animals, they are unlikely to transmit the virus far or infect humans. The most dangerous events regarding adaptation of the virus through a panzootic virus is farms with large and dense populations that can provide conditions for viral adaptation to mammals because they permit many passages and they facilitate contact and airborne transmission. In this regard, many passages, they permit the virus to mutate and select and adapt towards these new species and cross the species barrier. So most dangerous events are those that involve transmission in farms regarding mammals. And we had an event a few years ago in Spain with 52,000 farmed minks infected and the virus transmitted effectively. We had also similar outbreaks in fur animals, in different fur animal farms in Finland, and that's why in Finland avian influenza vaccinations began to be offered to persons at increased risk of infections. On the other hand, these viruses derived from mink, they showed that transmission was effective at direct contact to 75% of exposed ferrets, and this virus was also airborne transmissible, but fortunately it was not effectively transmitted very effectively. It was transmitted airborne, but not effectively, in order to produce sustained infection cohorts. Now, fur animals, they are important because they can be infected by human influenza viruses also, so can have mutations and recombination events in these animal farms. And most importantly, in the US, we have dairy cows infected by this virus, and we had a huge the dissemination of the virus throughout the US. 1,083 dairy herds were infected in 18 states up to date. And the virus also had spillover near the environment of these flocks, where the wild fauna was infected due to the dispersal of the virus from the cows to the environment. And humans were also infected. This dispersal was highest at July 2024 after effective surveillance and measure effects taken. The dispersal is highly contained, but exists. And this was due to a certain genotype that was dispersed in the US through the flocks of animals in cattle farms, but we had also a second genotype infecting cattle. So it's an event that can be also that could be also introduced to Europe. So biosecurity in cattle farms is very important. Milk is contaminated with the virus, and the primary route of transmission within the herds is aerosol containing milk droplets and in the milking parlors. The virus is transmitted through a movement of animals and inadequate biosecurity conditions. And this virus was also transmitted between ferrets through direct contact and also via respiratory droplets, but not very efficiently, fortunately. So this virus retains receptor binding and pH fusion characteristics of avian host phenotype, fortunately. And the situation in the US regarding poultry flocks is there. We have 2,000 flocks infected, millions of birds affected during this panzootic. And on the other hand, we have also human cases in the US. Since 2024, we have 71 cases, 41 due to exposure to dairy herds and 24 due to exposure to poultry farms and culling operations, whereas on the other hand, we have 3 cases with exposure source unknown, which is highly worrying because there is an increased risk of silent adaptation of the virus to humans, and surveillance could lose this signal effectively. So, on the other hand, we have mild ocular symptoms concurrent with mild respiratory symptoms in infected workers suffered from conjunctivitis. And possibly the lower pathogenicity of this virus in these cases is due to the different site of infection. We have also avian receptors in the conjunctiva. So this is another way of infection for these avian influenza viruses. Another route in human, the conjunctiva. And probably this infection site in combination with probably pre-existing immunity from the 2009 H1N1 pandemic influenza virus due to the neuraminidase 1 has provided protection from mortality and severe clinical disease. So this is the situation, the current situation regarding human infections from this panzootic. We had a huge surge in human cases. I mean, huge surge regarding this sporadic infections. Okay, we don't have a pandemic, but we have infections of humans in 69 infections in US in 2024, and whereas after significant measures, these infections have dramatically been reduced in the US. So the most important problem with this virus is adaptation in mammals and the reassortment in vents and passages in mammalian in mammal flocks. And on the other hand, we have sporadic isolated infections in humans, and in case we have local clusters of human infections, and efficient adaptation, we could have a pandemic strain in the future. So we are somewhere here regarding the different steps towards a pandemic virus. And in this regard, it's very important that we try to address these risks through integrated surveillance that could involve wild birds, poultry, mammals, human sentinel networks, shared case definitions and reporting, also routine sequencing, metadata to track plates, reassortment, antiviral resistance, mammal adaptive markers, and also early warning triggers like unexplained poultry die-offs, neurologic disease deaths in mammals, severe respiratory clusters in exposed workers, prevention in farms regarding biosecurity, disinfection, control entry, and vaccination where appropriate, strategic poultry vaccination paired with surveillance to detect silent circulation. In these cases, it occurs routinely. And also human protection regarding healthcare IPCs and prophylaxis and etc. And also regarding the environment, safe carcass disposal, decontamination, or equipment transport. These are a few things that could be shared regarding this very interesting virus, and I hope it was interesting. Thank you.
Thank you very much, Professor Dovas, for your very comprehensive and insightful and very detailed overview of the avian influenza evolution. So I will ask if there is any question in the auditorium or online. Have we? No online. So have you any question in the auditorium? So I can ask perhaps a question. Do you think there is a surveillance gap which we must address? Is there any urgent surveillance gap? You have here many integrated surveillance, but is there a gap, or is there any single surveillance indicator we could have to be prepared for the risk of emergence of an outbreak?
Well, I think that the experience from the USA regarding this huge episodic, also involving animal farms, is very important, because this integrated surveillance has to do not only with animals, but also with humans working in these premises, and surveillance also to try and find signals regarding silent transmission in humans and possible adaptation. So surveillance should be done not only in animals, but also in humans working in these premises. And also the most important in Europe is fur animal farms. Like minks, that it seems that minks, they are susceptible to these viruses, and they also can be infected with human influenza viruses. And this is a major problem regarding possible candidate recombination events.
Thank you. Thank you very much. Other questions from the auditorium? No one, so I think we can thank you very much, Professor Dovas.
Okay. Thank you.
And we will now move to our second speaker. It's Professor Akinosoglu. She is Associate Professor at the School of Medicine here in Patras, and she is an internist and infectious disease specialist. And she will discuss the challenges of managing human and zoonotic disease outbreaks, and I think emphasizing where One Health consideration intersect with ethical decision-making. So, Professor Akinosoglu.
Thank you. So can I have my slides, please? Okay, shall I open it myself? Okay, no worries. I'll do it. This way. Sure? Yep. Yep, okay, great. So thank you everybody for being here this morning in this difficult time, and also I'd like to thank the President, the Chair, and also Professor Vandarakis for the honorable invitation. So I'll try in the next 20 minutes or so just to give a brief overview of what happens in human zoonotic disease outbreaks, and I'll try to give an aspect of the bioethic part of challenges that we face, and especially when One Health comes into the game. So, and it all starts from way back, and I think the same challenges that we used to face in the common era of 541 in the Justinian plague is the ones that we even face in the era of the commoners in 2020 and COVID-19 that we'll discuss later. So remember that what happened then, back then, the plague of Justinian was actually a big plague coming in that occurred in 541, and actually it originated from Egypt and really cost the lives of many residents of Constantinople at the moment. So Justinian I was an emperor that actually tried to recover all the glory of the Roman Empire to the east, and revived, trade practices. Hence, actually, through this intersection and moving population and trading opportunities, then actually gave a very big interaction and increased interaction from wild rodents sometimes to community and population associated rodents that actually gave birth to this plague. And things did not change much, but in the COVID-19, actually the lessons we got were the same, but we didn't get them early enough. So it all started from a very nice story of that we're not alone, and it starts the same. We're not alone, we're actually in the midst of many emerging pathogens, either that is viruses or bacteria or fungi or parasites, and whatever that is, you see that more than 50% are zoonotic, either coming from the wildlife or not the wildlife. And these are zoonotic diseases that are emerging, but actually what bothers us most is that they claim more than 2.7 billion lives worldwide each year, and they cause more than 2.5 billion cases of sickness each year, which is quite an impact to just address and not to address and just not bother with. And I think climate change, you know better than me, and we've already discussed this morning that it's a major risk factor and is a key factor, and because actually it has to do with our transmission dynamics in terms of zoonotic diseases. It actually has to do with how we form the habitat, the emerging pathogens, but also in the setting of susceptibility in the population that is emerging, that is immunocompromised within the multi-drug resistant pathogens, but also in terms of iatrogenic interventions that actually cause more harm than benefit sometimes. And in this whole crisis, we are not innocent bystanders, because actually it's the climate that we form by fossil fuel exploitation, but also through human immigration and also through the decreasing water quantity and quality that we are so good at. And I think it has by now been quite clear that actually humans, animals, and environment, but also pathogens are quite strictly interconnected, and one can do without the other. That's why we're discussing this point at the moment, and it comes to One Health, and that One Health aims to sustainably balance and optimize the health of people, of animals, of the climate, and also of pathogens, because they are useful sometimes. So One Health promotes a sustainable and healthy future through actually collaboration, because you need a collaborative setting of sectors and disciplines. It needs to be coordinated through societal arms, and it's quite important, and also to build capacity in order to provide a sustainable One Health. And as it comes to pandemics and as it comes to zoonotic diseases and outbreaks, then it's quite important, obviously, both to prevent but also to manage and treat. And in terms of prevention, the actions to identify threats and reduce spillover include all these actions that actually extend far beyond the human kingdom, but also obviously the climate, but also and the environment, but also the animals and insects and plants. And they have to do with risk of reduction activities, the understanding of infection dynamics in the natural host and environment, to address the anthropogenic drivers of disease and emergence, and also integrating surveillance that sometimes has to be quite aggressive and actually plays with the boundaries of ethics and bioethics we're going to discuss about. And remember, in terms of management, this is what we have to break, and One Health actually tends to understand, but also breaks this vicious cycle, and then go to the management. But the management actually what has to do with is to be quite prompt in our diagnosis, to implement surveillance and epidemiological investigation to build the healthcare research capacity, but also research and development, vaccines, and management in the long-term impacts of zoonotic diseases, but also the outbreaks. So rapid investigation, research, and management and intervention is quite pivotal in the management of outbreaks and zoonotic disease outbreaks in order to identify origin, to map transmission, but also to document symptoms and then to promptly intervene. And I'll start with the most simple or plain measure, not so invasive, let's say, but intrusive or invasive, but thus the one that actually starts stepping on the line of bioethics and ethics in terms of One Health. And that was the control measures. Remember that the huge lockdowns that were implemented worldwide, even in our country, that actually were quite effective, because what we saw and what we did is that actually they reduced the reproductive numbers of both transmissions in wild animals, but also in humans, hence controlling the outbreaks plus the whole epidemic. And then they had to be implemented sometimes blindly early enough so that they have some impact of at least 50 to 80% of transmission reduction, and this goes through the ages back in 1918 of the Spanish flu. However, then the problems start, and the problems start in terms of, for example, patient rights and restrictive measures. So remember that the fundamental principles of medical ethics are obviously beneficence, do no harm, but also be just, and also do have in mind the autonomy and the need of the patient and the individual to ensure its rights. And it's a very thin line where justification of limitation of human rights, individual human rights, tend to be. And remember, by the Syracuse principles, if public health is actually a threat, then maybe the line can be stepped and broken. However, how transparent that would be, it's a matter of discussion. It's still a matter of discussion. So remember that in a pandemic or a zoonotic disease, either that has to do with animals or has to do with human beings, then who is the patient and who is more patient than another patient? So in the event of the pandemic, we needed to keep surveillance, and keep surveillance quite aggressively sometimes. And remember the cases of namely giving in public, names or reporting the individuals that were infected in order to protect the others. What was in COVID-19, or unfortunately and misfortunately was in HIV, if you remember, sometimes a decade ago in our country. And we had to intervene quite aggressively. So what is a patient's right to decline either therapy or diagnosis if, the fellow individual is at risk or the society as a whole is at risk? And we had to get specimen collection quite aggressively sometimes, but can you decline getting a specimen and getting diagnosed, since you can be transmissible and thus threaten your co-fellow or your co-citizen? And we had to be separated, isolated, and implement social distancing, and how this could work within a family, for example, that has got dependable members, or within a family that has to work or keep its adherence in terms of societal level. And we had to control international borders. So what we lost, and what in the meantime we actually risked for, and unfortunately happened to us and would draw upon, was the loss of privacy and sometimes confidentiality. We got some stigma, and that is all across the centuries in terms of pandemics and zoonotic diseases, either that was Ebola or was HIV, or was back in the leopard time in Spinalonga Island that most of you can recognize south of Crete. And so the rights, the violation of the individual rights is a very, very big discussion to keep on your mind when you were talking about One Health and actually the integrated policies that this includes. And remember, did we tended to, do our compromise our individual rights for the sake of the community. However, who is more patient than patient? So did we prioritize correctly? Because then comes another dilemma, another bioethic issue of whom should we have saved first, or whom should have reallocated or allocated our resources, either that were consumables or that was personnel. And remember, we made a choice back then, but was it the right choice? Should we have saved the women and the children first, like happened with the Titanic? Should we have served first the ones that come first, as we do in the ICU beds, first come, first serve? Should we have saved the most quality life years and be most cost effective, for example, the younger ones instead of the older ones? Should we have saved the worst off, as happens with organ transplantation lists? So that's another big question. Should we have saved first the ones or allocated our resources, vaccines or drugs, to the ones that have shown reciprocity to the society, as happens to the blood transfusion lists? The one who has already donated blood should be donated first, the blood back. We have saved the ones that are most likely to fully recover and go back in the battlefield, as happened in the World War II? Or should we have saved the ones that were instrumental in making society flourish? And that's quite subjective, right? So who would make society flourish after the death is over? So that happened in our groceries, that happened in our supermarkets, and that happened when it comes to our consumables, our food, our mandibles, and then the shelves went out. And this happened in our supermarkets, but it also happened with our personnel, because we got no medical personnel that was available, and this had to be another discussion we had to make. Because many of the physicians, either from the old times of William Dafoe and the plague year, it's quite, distinctive to read that what happened to the plague. And so the plague, it said in the diaries, defied all medicines. The very physicians were seized with it. Men went about prescribing to others and telling them what to do, and they dropped down dead, destroyed by that very enemy they directed to oppose. And this was the case of several and most skillful surgeons. And I think in the beginning of the pandemic, I think you all experienced, as comes to COVID-19, this nice little gentleman, so Dr. Li Wenliang, who was a morning whistleblower, who actually, first reported that something was going on in terms of the very strange respiratory tract infection that came out perhaps from the labs, or who knows where it came out from. And actually, it was the case of many, many physicians back from the Common Era or Antichrist that fled from their environment when plagues outbreak and about broke. And that was even Galen and Sideman that were, major fathers of modern medicine. And the same happened more recently. I remember 30 to 40 years ago with HIV pandemic. That's a more chronic situation that now at least we are quite happy to be able to manage and keep a good population at health. However, physicians tend to and have the right to reserve the right to not serve everybody that is sick out there, or do they have not? The American medical ethics code that actually is at least the one that most medical ethics codes are aligned to, actually says that when pestilence prevails, it is their duty, our duty, physicians' duty, to face the danger and to continue their labor for the alleviation of the suffering, even at the jeopardy of their own lives. Do we or do we not? And should we or should we not? But a paragraph later, then the same code actually states that the supply physician is not an unlimited resource. Doctors need to balance immediate benefits to the individual patients with the ability to care for patients in the future. If I die, how am I going to care for patients in the future? And how am I going to care for the depending members of my family or the future members of my family? So as Immanuel Kant used to say, two things, or we must, or the most, and that's the starry sky above and the moral law within me, because actually nothing can define it and nothing can actually limit it. And the thing is, the same happened with, and it happens in every outbreak, either that was zoonotic or was had to do with transmission from human to human. So did we see all of us? Because actually a health crisis is within a social and a social crisis. And the greatest moral challenge posed by the pandemic is how actually to respect commitment to social justice in the face of overwhelming and entrenched inequalities. And it's very, very important. Because burden of disease to developing world or middle or low middle income classes is actually the one that's posed more, and it's more severe because we've got unequal access to interventions, emergency access, international trade, or research. See how international trade or borders were closed, and how many and how diversely, countries, low middle income countries, got affected quite diversely. How fumigating, for example, the Dominican Republic was actually different from what we did, or upper middle income countries happened, and actually what happened to the culling of our animals. See the minks, for example, in COVID-19, but actually remember the bird flu and the killing of birds in Koilang during 20 years ago during the avian influenza. And the thing is, did we also see us as a whole? Because in every pandemic and every One Health perspective, you actually have to see not only the health issues, but also the societal issues, the ethical issues, the cultural norms, and etc., the ones that actually tie us in a cohesion and hold cohesion in a society, or else they burn into the ground. Because remember, what we did is actually we had to preach alone, actually to dance alone, to exercise alone, to study alone, sometimes actually dance in the street alone, and actually either play music alone, remember the empty auditoriums, actually, have fun alone in separate circles in the countryside, actually even graduate alone, even give birth alone, but most importantly and most devastatingly, die alone. Because remember how the funerals were carried out and how these things have to do and occur during the pandemics and outbreaks of zoonotic diseases, because actually, whether within violent deaths or major disasters and armed conflict, the inability to perform rituals condemns a family to a second death. And that is the symbolic death of their loved one for the lack of the tomb that perpetuates his or her name and confers social worth to the deceased and his or her inclusion in the generational continuity of a family. And that is a cultural norm that is very important in terms of psychology and the cohesion of the society. And what we did afterwards, and what happens afterwards, is we turn ourselves to research. However, even though research is driven by a desire to understand the causes of disease in order to or dysfunction and find effective methods for prevention or treatment, this way can go severely, severely wrong. And even such humanitarian aims can be risky. Because remember what we did back then, and we had at least the flexibility to do it during the last pandemic that was not so flexible, obviously a century ago through the Spanish flu or other pandemics, is that actually could intensify our research. We used telemedicine, we did trial and recruitment and consent a different way, or we did not at all, and this is a quite big discussion. We modified intervention delivery, we standardized remote evidence adjudication, et cetera, that actually changed a bit our good research clinical practice. And we skipped the timelines. Remember how the approval of medicine or drugs or vaccines did actually go through and how long they take, similar to a timeline of a good relationship, and how they take investment of time, of sentiment, of money, or energy, then actually we went and hopped up there in the fast-track procedure. Fast-track procedure because we didn't have time, any other time, to spend losing people and losing our beloved ones. However, remember that was the start and that was the end, because it was the rapid rise and rapid fall of many prematurely approved drugs in terms of enthusiasm and need and despair, most probably, that actually after sometime later, due to lack of benefit or toxicities, they had to be withdrawn by emergency FDA or EU decisions. And we also did science by press release, and that's another bioethic line that actually we crossed, and we will, unfortunately, have to cross during the next, hopefully not, pandemics. And many of our papers, remember, that were retracted because due to they were not properly either reviewed or critically appraised or checked as comes to their results. And even though this was a knowledge that we already had through 1918 with the Spanish flu, remember that the information that was communicated, the Spanish flu, was very transparent on behalf of Spain. That's why even though it didn't originate in Spain, then it was called Spanish flu, because during the war, then it was only this nation that actually was willing to discuss about it openly. That's why the name. So we try to balance, and this is what is happening in every systematic approach of zoonotic disease or outbreaks, our epistemological ethos versus our ethical ethos. But it was not so successful, right? Because things are much, complicated, more complicated in reality than it is in theory. So what we did is we act first and we think later, because actually we didn't even, consider ourselves and thought carefully of who set the research agenda back then, who was the research for, how was the research designed, who owned the knowledge, and what is the impact of the research. So there were many, many voices back then yelling that, okay, ethics is only useful if you've got time, because now we don't have time. We're losing lives, so we have to act first and think later. However, if we did that, and we didn't only do that in the COVID-19 pandemic, but also as comes to the experience with the Nazis and back a century ago, then what about the results and the data were actually produced, not through a bioethics, but also stepping on to our bioethics? Then there are data that actually would not have been produced otherwise. What are we going to do with that? Just send them in the garbage or actually use it even though it has not been produced properly? That's another food for thought that actually the One Health will meet upon it in next pandemic or in next crisis. So preparing for the future pandemics is a big discussion, because it actually requires surveillance, coordination, research, and actually preparedness. Preparedness in not in the sober times, not in the times of crisis, because then you cannot form plans or frameworks and actually discuss with the society so that communication is transparent. And actually, you've got to make frameworks and coordination and plans that actually are sustainable, or else, you know, they are threatened, and One Health is threatened by our ethics first, and then obviously our lack of funding or whatever difficulties, technical difficulties might that implicate. And by closing and to summing up, the conduct of life by Emerson was saying that life is a succession of lessons which must be lived to be understood. We made mistakes, but we have to learn, and we wouldn't have learned if we hadn't made the mistakes. However, William Osler, which most of you know that it's the father of medicine, actually said that the best preparation for tomorrow is to do today's work superbly well. So instead of acting first and then thinking later, it's most important to think now and then act better later. So thank you very much for your attention, and I'm open to questions.
So, thank you very much, Professor Akinosoglu. It was a very rapid, waterfall-like, but very thought-provoking presentation. And I must say I'm always fascinated about the speed in your speaking. So I think your reflection on the ethical dimensions of One Health show us that we are not alone, as you said at the beginning, but that our decisions affect not only public health, but also society, animal and environmental health. And I will open for questions. Mr. Zouros, you can ask your question. Wait a moment for the microphone.
Yeah, fascinating presentation. I wanted to ask you, I put this question from my WHO perspective. We were faced with many ethical dilemmas, and we learned a lot from the pandemic. Now, if you look into our new way of thinking, how we should prepare for the future, which relates to the ethical dimension, there are two things. One is that we have to build resilience in the communities. This is key in most WHO strategies for pandemic preparedness today. No matter how many plans you may have in the country, the reality is on the ground. The second is that we should map out our vulnerability in the community and not expect to find out who needs more, who is more vulnerable, who is a chronic disease patient, who is unable to walk and go and access a service. So the concept of ethics in the new era of the post-COVID preparedness for such challenging situations that affect the whole planet has expanded now. So you described very well the ethical dilemmas you faced then, but the ethics of the post-COVID era are significantly now changed.
Yeah, that's true. Thank you for your comment. That's quite true. And actually, you know, the pandemic or the crisis actually highlights the already existing inequalities and the social crisis that actually we have to address even in sober times. And either that, you know, gets heightened up and highlighted during a pandemic, but also in the post-pandemic era that actually has got all the consequences. So I totally agree. It's something that we have to map in advance and actually support in advance so that we are more resilient when crisis comes up. Thank you.
Other questions? We have no online questions.
Thank you. Congratulations.
Can you please say your name and where are you coming
yes,
from?
Sure. Elizabeth Valério from the National Institute of Health from Portugal. So congratulations on your talk. And I wanted to ask you, do you feel that we have learned enough during COVID? Are we really going to use the supposed lessons we were supposed to take?
Okay. so thank you for your question. So that's a rhetorical question, right? It's mostly subjective. Hopefully our systems must have learned some things and implemented, and also, already make frameworks and make plans and preparedness plans for the next pandemic. However, how much we've learned this time, we'll see in the next pandemic. Then, it's going to be the final report of what we learned. Hopefully, we've learned much. I think we're most prepared than, what we had to face 30 years ago. However, I'm afraid that we still got place for improvement, right? Thank you.
Other questions? So, Professor Akinosoglu,
Thank you.
thank you very much. And we will now continue with our final presentation. Our third speaker is Mr. Rosset. He is a research scientist at the Centre National de la Recherche Scientifique at the University of Lille, and he will take us to the molecular frontline.
So hello everyone. So now I will talk about something more focused on one example about salmonella and the chicken production, and how we are trying to link in the One Health perspective these two questions. I mean the chicken poultries breeding and the salmonella carriage. So first, I'm gonna give some statistics from the EFSA. The latest one available are from 2023, and so the zoonoses among the EU are involving different type of bacteria. The three most important bacteria found involved in zoonoses are Campylobacter, Salmonella, and E. coli, and some specific strains of E. coli, followed by some bacteria like Yersinia, Listeriose, and viruses, or as well some parasites. So as you can see here, the Campylobacter remained the most reported zoonoses in 2023, just followed by Salmonellosis. And I will just go in more detail for first the Campylobacter. In 2023, 229 fundamental outbreaks have been described involving that bacteria, reporting more than 1,000 cases of illness and 80 hospitalization. And as you can see here, among all the countries of Europe, Malta is, in 2023 at least, the most important cases were coming from that country. In the outbreaks, broiler meat and related products were involved in 15 outbreaks, so it's mostly linked to that type of food. One thing interesting for, let's say, the everyday life is the outbreaks are always more important during the summertime, and it's most likely because people are cooking raw meat first in the same plates. After cooking the meat, so you're putting and after killing the bacteria on the barbecue, you're putting back in the same plates your cooked meat, which contaminate the naturally present bacteria on the raw meat. So next time, just think about that during your summertime. About the Campylobacter jejuni, so the different animals are not only chicken, but you can find pigs, most likely, and bovine animals who are carriers of that bacteria too. But if we looked closer to Salmonella, in 2023, it was the majority of multi-country outbreaks were involving that bacteria. So I'm going to go deeper in that specific pathogens. So for more than 1,000 foodborne outbreaks, more than 9,000 cases of illness, and 16 deaths. And in terms of hospitalization, it's much more important than the Campylobacter cases I just described, because we are almost reaching 1,700 hospitalization. Regarding the countries, as just mentioned, as you can see here, there are different countries who are affected by that bacteria. In the numbers of outbreaks involving eggs and egg products were 83, for the broiler meat and related products 30 outbreaks, and eggs and egg products were the highest risk then agent food pair for Salmonella. Again, exactly the same observation than Campylobacter. It's occurring most likely during the summertime. And if we look closer to the different poultries involved in Salmonella outbreaks, so we have Gallus breeding, laying hens, broilers, and two types of turkeys too. If we look at the different product again and the different type of meats, there are pig meats who are in terms of positivity of sampling are quite important after, of course, the broilers meats. And something important with Salmonella is a single species, but it encompasses more than 600 serovar that has evolved to adapt to different type of hosts. And if we look at that serovars, the most important ones is Salmonella enterica enteritidis, followed by Typhimurium and Infantis. I will explain a bit later why enteritidis is so important so far. Why poultry are such an important product. It's since few years, it's became the most consumed animal product in the world just after the pork meat or pork products related. And if we look at why, one study has described or tried to describe and understand how it became for only one serotype so worldwide found. So because most likely there are a lot of trade of live poultry, in even eggs, between different countries all over the world, involving USA, but Europe, of course, and many other countries as well. So after describing that, now it's interesting to try to understand how Salmonella can, or even other bacteria, because it could as well be exactly the same case for Campylobacter, but I will just stay focused on Salmonella here. So it's very interesting to understand how physiologically the chicken can carry such bacteria in their body. So it's been described that it's most likely through the ceca, so one specific part of the intestinal tissues, we can find that bacteria, and Salmonella can get inside the entire body as well through the ceca and affect other organs. But something very interesting to say as well is that Salmonella enterica doesn't kill chicken. There are other Salmonella species, which is Gallinarium, who kills the chicken, but here it's just affecting human health. Then, if we look closer to the intestinal organization in the chicken, it's like in many other animals. There are first different types of cells. And the most important cells here to discuss in more details, it's the global cells. You can see here, they are secreting some type of glycoproteins called mucins, who form what we call the mucus. So there are two types of mucus, the inner mucus layer, which is quite difficult for the bacteria to penetrate in. And after that, there are the outer mucus layer, which is degraded most likely or used or at least just moving with the food in the lumen of the intestines, who is interacting directly with the bacteria. So how it works, the bacteria have on their surfaces different type of molecules like adhesins through pili or flagella who are able to stick, recognize and stick on the tissues or of mucus, and they are as well able to degrade the different type of molecules on the mucin. So what type of molecules I will explain very quickly because that's a very long story. So here that's the mucin's core, so composed of amino acids, nothing special, but for the mucins, there are here in blue a lot of different glycans. The glycans can be divided in different domains with core and chain, and you can have different type, and I'm not going in that details here, different type of core, etc. Here we will just focus on the three terminal structures. So here, sialic acid. So one talk this morning just talk about sialic acid, but regarding the flu and in interaction with virus, it's pretty much something possible with Salmonella too. Sulfation, which is a non-glycosidic part on the oligosaccharides, on the mucins, and fucose, another one monosaccharide, who, for example, is able to make blood group, and so in your body, but as well in chicken body too. So why it's something difficult to characterize is that because glycosylation can be predicted, and because of different reasons. For example, one of them is the initiation. It's not well understood so far. There are different types of enzymes who are involved in the modifications, sometimes for some very similar modifications. The localization of that enzyme are hardly predictable, and the cofactors and substrate are limiting factors, of course, and they are differently distributed in the body. So that's why we need to do experimental characterization of glycosylation, which rely most likely in mass spectrometry or NMR. So our strategy first was to compare two types of lineages of chicken, one called N and the other one is 6 or 6-1, but I will say 6 to be faster. And so the lineage N is resistant to Salmonella and the other one is sensitive. So let the bacteria proliferate inside the chicken body. And we cut different parts of the intestinal tissues and focusing, of course, on the ceca and ileum. So the two important parts, as I said. We looked at the transcriptomics levels due to the collaboration with one team from France in Tours, where they did some transcriptomic on the two lineages. And they first look at very, let's say, obvious enzyme for the glycobiology world. They found, for example, FUT2, which is an enzyme who is able to add one fucose in alpha-1,2. On the oligosaccharide, like here, for example, who form different type of blood groups, like I just show here for the blood group H. It was something interesting, of course, because of the literature. It's been described in mice for Salmonella typhimurium, for example, here, that FUT2 minus mice were showing an increased proliferation of that bacteria. But if we look here, relating to the different lineages. So in lineage N, which is the resistant one, we have an upregulation of FUT2, but it doesn't fit with what we found when we were measuring the quantity of fucose in the CCOM. We see the all way round, in fact, in L6, more fucose. But by looking again in the transcriptomic data from our collaborators, we found an upregulation in the lineage 6 of FUT10, another one less described enzyme in literature, so that's a good target for future experimentation. The other aspect is about the sialylation. So as you can see here, different type of glycans modified with that. When looking at the transcriptomic analysis, many sialytransferases were upregulated. But when we looked at the global quantity of sialic acid, we couldn't see any differences in the CCOM between the two cell lines. So for us, it led us to the possibility that sialic acid are not really involved in the Salmonella enteritidis colonization. The last part, but I will go very fast for this one, is the sulfation between the two secum of the two lineages. And we see, it was a big surprise for us, many differences between the two lineages. So we need to further characterize that specific, and that's something quite challenging to study sulfation by mass spectrometry. So how we can decrease the Salmonella enteritidis in numbers in poultry products. So one strategy would be to select one very strong lineage of chicken who could boost the resistance of the proliferation of that. And for doing so, we need to think back to the genetics of chicken, because we eat chicken, but we are not thinking where they are coming from the wild initially. And surprisingly, they are worldwide available, but they are just coming from a very specific part in the world, Southeast Asia, with a repartition of different species of Gallus, or Gallus and other name of species name, sorry. If we look closer to the relation between all that species, so you can see some very fancy ones. That's my point of view, at least for Gallus varius. That's a very fancy chicken. So you can see here, the domestic chicken are very close to only one species of chicken, the red jungle fowl, found in Indonesia. And the other species are not involved in the domestic chicken selection. That's what I'm showing here. Yeah, it's a bit blurry, but anyway. That's what we call the bottleneck effect. So if we think about the jungle fowl, so there are different species of Gallus. They were selected only among one species, the red jungle fowl, which decreased the genetical variability. And if we think about the industrial production, it's even worse, because only few breeds, are used for obvious reason, of course, the rapid growth for the broilers, and that's the main things to the industrial keep in mind, is just to have a big, in a quick way, chicken. But it's have a cost, of course. And if we think about the bottleneck effect, it's then showing a very small diversity. And even a very interesting study described among 800 genomes that it led to 3% more deleterious mutation during the process of domestication. So we are trying to, in collaboration with Nagoya University, to think about the evolutionary perspective and, of course, of the glycosylation, because that's not something easy to do, and that's something we are doing. So by comparing the different sulfation, fucosylation, and sialylation, among the different type of jungle fowl we are trying to have access. And to see if during the domestication we have reduced the complexity of glycosylation too. Yes, that's something we will do with the avian glycomics facilities from Nagoya. In the same time, we are trying to have a One Health perspective, not to involve only scientific, but as well industry. And in that case, we are trying to collaborate with a big company called Hendrix Genetics through Derek. There are facilities in France who are producing and trying to select some new breed of broilers. So we are in touch with them to work on the genetics from their company, and in the meantime, because it's not only about genetics, to try to have an effect on the glycosylation through the food via probiotics. So that's something we are trying to do at the moment. So yeah, that's it for me. So I would like to thank all my collaborators from Tours, I just mentioned, and all my collaborators in my lab in Lille. Thank you for your attention.
So thank you very much, Mr. Rosset, for your very comprehensive and detailed talk, showing how the glycosylation could protect both poultry and also humans. I think I have not understood if it's also in real world poultry. You can make it in real world in poultry, or it's only in laboratory?
We are, of course, laboratory based. So our knowledge is to do the glycosylation and to develop techniques to have high throughput analysis of glycosylation. But that's what I mentioned at the end. We are in collaboration with the industrial group who are producing different breeds of chicken, and what we are trying to do is to select with them through different markers new breeds. Yeah.
Okay, so you try to translate.
Yeah, that's a long process, but yeah, that's what we are trying to do.
And are there any obstacles in translate it to the real world poultry?
The first limit is because they have a huge access to genetical information, but they keep that information for them. So we are just working on what they give us, and we will produce some data, and then we will try to match those two aspects. Yeah.
Okay, thank you. Is there any question from the auditorium for Mr. Rosset? Please wait for the microphone. And if you can say your name and where you are coming from.
Uli Klumper from TU Dresden. So I'm wondering how many genes are actually involved in encoding for these glycosylation things? How easy is it to create GMO chicken that cannot be salmonella infected?
That's a good question. It's hard to answer, but based on the transcriptomic data from our collaborators, they found more than 800 genes affected between the two lines. So yeah, it's not easy, but yeah, probably GMOs in the future, if it's something possible, yeah, it would be for sure something interesting to test, yeah.
Any other question? We have 4 minutes.
But it was a perfect timing to talk about chicken, and it's time for food.
So if there are no other questions, we can conclude this session. And I will thank you all 3 contributors, all 3 speakers, for the excellent and complementary contributions. And we see that the One Health perspective is not just a framework, but it is a necessity for understanding and preventing the complex zoonotic threats we face. So thank you also, you very much for attention and enjoy the lunch and the rest of the conference. Thank you.
Okay, everybody's ready? So thank you very much for being here. We'll start the third session of this conference. So for the third session, we will welcome 3 different speakers. The first one being Catherine Rob-Maslow from the University of Lille, from the Institute for Structural and Functional Glycobiology, and then she will discuss and present some work about the mucin O-glycans and how we can use them to prevent bacterial and viral infection. So I'll leave you for half an hour or so.
It's okay? It's better. Thank you. So let me first briefly summarize what are mucus and mucins. Mucus is a secretion that covers every epithelial surfaces of the body, and it constitutes the first physical barrier between the external environment and the underlying tissues. Secreted mucins are the main solid component of mucus. They are high molecular weight glycoproteins able to form hydrogels at the surface of the epithelium. And they are heavily O-glycosylated since more than 60 to 80% of the mass of mucins are based on O-glycans. As you can see in this picture, different types of apomucins are secreted depending on the organs. For example, you will find MUC5AC in the ears, the lung, and the stomach, and you will mainly find MUC2 in the intestine. O-glycosylation of mucins is initiated by the addition of a first residue of an acetyl-galactosamine to form the TN antigen. And this TN antigen is further elongated by the addition of galactose or an acetylglucosamine to form different types of cores. And in humans, you will mainly find core 1 to core 4. And these cores are elongated by the addition of lactosaminic chains, and they are terminated by 3 types of monosaccharides, which are fucose, sulfate, and sialic acid residues. And please keep in mind that these residues are really important for the interaction with pathogens. So O-glycans are very diverse, as you can see in this slide. You can find several different types of O-glycans carrying by the same type of apomucins. Oops. I don't know. Do I have something to do or? Yeah, I can. I'm not sure if I can close all rooms. Yeah. Okay. And O-glycans can also express different types of antigenic structures, such as the blood group H, A, and B, the Lewis determinants, but also some oncofetal antigenic structures, such as the TN antigen and its sialylated counterparts, sialyl-TN and sialyl-T antigens. Okay, but now it doesn't work. And click on remote control.
Let me check. I'm sorry. Okay, this one.
Yeah.
Yeah.
Okay, so I can't use it anymore. No problem. Okay, so expression and glycosylation of mucins vary among species, but also between individuals in the same species, and this is mainly due to the type of lifestyle, the diet, the consumption of medication or surgery. For the same individual, O-glycans can vary depending on the organs, but also during pathologies. And this is the case in pathologies such as cancers, inflammation, metabolic disorders, and of course, infections. Proglycans play fundamental roles in physiological conditions and confer rheological properties and control viscosities. They help to maintain mucosal homeostasis. They protect the epithelium against toxic and pathogenic agents, and they play a role in innate immunity by interacting with selectins. Proglycans are also implicated in the sweet dialogue with bacteria. For example, they serve as a size exclusion barriers that can selectively bind certain type of viruses and that can trap microorganisms and flush them out of the body thanks to the mucosal turnover. They also serve as nutrient sources for bacteria. Commensal or pathogens can degrade mucinoglycans thanks to their bacterial glycosidases. They can catabolize glycan-derived sugars and produce short-chain fatty acids that will, in turn, modulate host cell signaling and the activity of goblet cells. On mucinoglycans can also specially organize microbial communities by altering, for example, molecule transport, such as nutrient or host immune factors, or key signaling molecules. Glycans can help to, can favor the direct binding of microorganisms, and they can prevent bacterial aggregation. However, despite their well-known role in the interaction with pathogens and bacteria, they are still underexploited in host-driven anti-infective strategies. But many studies have demonstrated that mucinoglycans can neutralize pathogens without killing them. And I will show you now 5 types of different interaction between mucinoglycans and bacteria, and show you how we could use glycans as therapeutic adjuvants. So the first example is the interaction with Pseudomonas aeruginosa. And in this paper, they have demonstrated that if you use free mucinoglycans from MUC5AC, but not the monosaccharides that constitute glycans, you can downregulate virulence genes involved in quorum sensing, biosynthesis, the biosynthesis of siderophore, and the secretion of toxins, and in particular the type 1, type 2, 3, and 6 secretion systems. And they also demonstrated that these mucinoglycans can disperse biofilm, as you can see in this picture, and promote a planktonic lifestyle of Pseudomonas aeruginosa. 2 years later, the same team demonstrated that in a healthy mucus, if you use core-1 or core-2 glycans, but not their fucosylated or sialylated counterparts, these glycans are able to activate the sensor kinase RET-S, and RET-S is able to inhibit the two-component system, GAC-S, GAC-A, which normally drives chronic infection programs. And this is strongly correlated with a down-regulation of the type 6 secretion system, which is normally used by Pseudomonas aeruginosa for its virulence and to kill other bacteria. So core-1 and core-2 glycans can promote a kind of microbial coexistence. And they demonstrated that in an altered mucus, where there is no expression of core-1 and core-2, these altered glycans favor a pathogenic behavior of Pseudomonas aeruginosa. The second examples concern Streptococcus mutans, which is a causative agent in dental caries. And the authors have demonstrated that if you use MUC5AC or MUC5B glycans, you can downregulate expression of the quorum sensing pathways, which is normally activated by the competence-stimulating peptide and the SIGEX-inducing peptide. And you may know that Streptococcus mutans is naturally competent, which means that it is able to uptake foreign DNA and to share it across species through horizontal gene transfer. And what they have shown in this study is that if you use MUC5B mucinoglycans, you can prevent the acquisition of antimicrobial resistance through natural genetic transformation, and you can also prevent formation of biofilms. But again, if you use the monosaccharides alone or the mix of monosaccharides constituting glycans, you completely lose the effect. The third examples concern Candida albicans, which is an opportunistic fungal pathogen, which is able to asymptomatically colonize mucosal surfaces of most healthy humans. And in these studies, they demonstrated that mucinoglycans can suppress filamentation, which is a yes to high-field transition and is a major virulence factor for Candida albicans. And they demonstrated that if you use either MUC5AC or MUC5B or MUC2 glycans, you can observe this effect. And they also tried to synthesize some short glycans, and they demonstrated that COR1, COR1 plus fucose, or COR2 plus galactose are sufficient to inhibit filamentation, but if you use COR1 substituted with sialic acid residues, you completely lost this effect. They also demonstrated that mucin glycans, and in particular MUC5AC glycans, are able to suppress phenotypes relevant to infection, such as surface adhesion, as you can see here, on the formation of biofilms of Candida albicans. And they demonstrated that in absence of mucinoglycans, if you co-cultivate Candida albicans and Pseudomonas aeruginosa, Pseudomonas aeruginosa will bind to Candida albicans hyphae, and it will secrete toxins that will kill the fungal pathogens. So this indicates that mucin glycans can influence microbial population dynamics because when they add mucinoglycans, they demonstrated that there was an attenuation of the virulence of Pseudomonas aeruginosa and a delayed Candida albicans eradication. The fourth examples concern Vibrio cholerae. Vibrio cholerae, normally infection by Vibrio cholerae, arise from a toxigenic conversion by a CTX bacteriophage, a process by which a CTX bacteriophage is able to infect non-toxigenic Vibrio cholerae strains, and this bacteriophage will secrete the cholera toxin, which is responsible for the watery diarrhea observed in cholera infections. And they demonstrated that MUC2 or MUC5AC mucins, but also the mucin glycans from these two types of mucins, are able to block the ability of the CTX bacteriophage to infect susceptible Vibrio cholerae strains. But again, when they use the monosaccharides, a mix of monosaccharides alone, they didn't recapitulate this effect. And in fact, they demonstrated that it was COR2 glycans that were implicated in this process, and COR2 glycans are also able to suppress the production of cholera toxin. If you use COR2 glycans substituted by fucose or galactose, the effect is weaker, and when they try to use COR1 or COR1 plus fucose or COR1 plus sialic acid residues, they totally lose the effect. And the last examples concern Salmonella typhimurium. So you heard about this morning with Yannick. This is one of the most common foodborne pathogens, and this pathogen has evolved to develop multi-resistant genes against antibiotics, and this is a pathogen that is also able to exhibit sophisticated mechanisms such as Salmonella pathogenicity island 1, which encodes for the secretion systems, the type 3 secretion systems. And in this study, they demonstrated that MUC2 in an in vitro cell culture with HT29 cells, they demonstrated that MUC2 are able to inhibit host cell invasion by the pathogens by suppressing the expression of this SPI1. They also use synthetic glycopolypeptides carrying native mucin glycans, and they demonstrated that these synthetic glycopolypeptides were enough to recapitulate the antivirulence function of native mucins, but only when these polypeptides were substituted by the first residue of N-acetyl galactosamine, because when they tried to add some sialic acid, either in alpha-2,3 or alpha-2,6, they totally lose the effect, as you can see here. So, in conclusion, mucinoglycans can attenuate virulence of bacterial and fungal pathogens. They can help to disperse biofilms, but their effect is strongly correlated with the structure of glycan. So this is really important to be able to characterize which type of glycans are implicated in interaction with which type of pathogens. And in the future, it will be possible to synthesize glycomimetics or synthesize short glycans, or, if necessary, to purify mucinoglycans from animal sources, such as porcine or bovine, and to use these glycans for the design of precision antivirulence therapeutics. So the second part of my talk will be a short introduction of bacteriophages. So you heard a lot about this morning, which are viruses specifically targeting pathogens on bacteria. And you may know that more than 90% of the intestinal virome is constituted by these bacteriophages. And these phages are able to interact with mucins. Around 10 years ago, the bacteriophage adherent to mucus model has been proposed. And in this model, they demonstrated higher concentration of phages closer to the epithelia, where mucins are newly synthesized and secreted. And these phages are able to encode for carbohydrate-active enzymes that can cleave mucinoglycans, and this allows phages to diffuse down the mucus layer. And they also demonstrated that the diffusivity of phages is strongly correlated to mucin concentration, as you can see here in this left cartoon. And a particular protein, which is the OC protein, from the coliphages T4 are able to bind to mucins via the immunoglobulin-like domains of this OC protein. And when bacteriophages express this type of protein, they demonstrated that, for example, for the E. coli bacteriophage PNG6, this OC protein can interact with fucose residues, but not sialic acid residues, and they can promote mucin secretion. Another E. coli bacteriophage, the PNG9, is also able to adhere to the intestinal mucus and to fucose, but is not able to promote mucin secretion because it expresses a variant of protein which lacks domain 3. In this study, for example, they compared the efficacy of two phages, VAC1 and VAC3, which are morphologically and genetically similar and are both able to infect Pseudomonas aeruginosa. But what they have shown in this study is that only VAC3, which is a phage adapted to mucosal condition, as you can see in this picture, since only VAC3 was recovered in nasal washes, trachea, and lungs of mice, and only VAC3 was able to protect the mice from lethal infection with an hypervirulent strain of Pseudomonas aeruginosa, which is PA14. And in this study, they demonstrated the efficacy of the bacteriophage PNG6 to kill enterotoxigenic E. coli in vivo, and they also demonstrated that this bacteriophage can upregulate MUC2 expression, and this suggests a positive feedback loop for phage adherence to mucins. So, in conclusion, bacteriophages adherent to mucins can kill pathogens, can induce secretion of mucins, and thus can restore the integrity of mucosal barriers, and their adhesion to mucins increases their persistence and efficiency. So, in the future, it will be possible to engineer phages with tailored mucin-binding properties and to use these engineered phages in prophylactic use, for example, to prevent biofilm formation on medical devices, and to use phagotherapy to target multidrug-resistant bacteria in treatments where antibiotics alone failed. And the last part of this talk will be about some work we are currently doing in the lab about the development of cyalomimetics, which can be used as potential antimicrobial peptides. So in the past, we demonstrated an hypercellulation of mucins, of airway mucins, in response to different pathogens. For example, for the infection of Pseudomonas aeruginosa, we demonstrated using a cystic fibrosis piglet model at birth that there was an increase in the level of cellulation of mucins correlated with a disturbance in the mucociliary transport, and this favored Pseudomonas aeruginosa growth, adhesion, and colonization. And in another study, we also demonstrated that the hypercellulation of airway mucins in cystic fibrosis patients favored and drives the selection of long-adapted variants of Staphylococcus aureus, because these variants are able to consume free sialic acids that are released from cellulated mucins by sialidases produced by other pathogens. And this variant and these sialic acid residues can drive the chronic lifestyles of Staphylococcus aureus and explain why it is possible to recover some persistent pathogens in the lung of cystic fibrosis patients during years. And based on these results, we decided to develop some heptapeptides able to mimic sialic acid residues to fight pathogens, and we demonstrated that these heptapeptides were able to reduce the viral titer or SARS-CoV-2 on the influenza virus H1N1 by more than 4 to 5 log 10. And we also demonstrated that these heptapeptides were able to inhibit the bacterial growth of Pseudomonas aeruginosa and Staphylococcus aureus. By doing some transcriptomic analysis, we demonstrated that, in fact, these heptapeptides killed the bacteria by creating perforation in the cell membrane, as you can see here in this electronic microscopy figure. And these heptapeptides were, in fact, able to impact cell wall biosynthesis and metabolic pathways. And they can also activate the SOS system on bacterial defenses. And we evaluated the efficacy of these heptapeptides on more than 20 different types of bacteria. Some of them were multi-drug resistant, and we demonstrated their efficiency on gram-positive, gram-negative, and mycobacteria. And we also demonstrated that they were able in planta to prevent infection. When we incubated peptide on bacteria, we didn't observe any anti-road 6-day post-infection, as you can see here, in comparison to bacteria alone, confirming a preventive effect of these heptapeptides in planta. So, as take-home messages, mucinoglycans can act as natural decoys for pathogens and help a pathogen to bind to mucins. These glycans can weaken bacterial virulence by disarming pathogens. They can disrupt biofilm formation, and this can increase treatment efficacy because they keep bacteria in a planktonic state, so they will render bacteria more susceptible to antibiotics. And phage therapy can help also to fight mucosal pathogens because addition of bacteriophages to mucins will enhance phage concentration and persistence in mucosal environments where most of the pathogens reside. And as potential clinical application, synthetic or purified mucinoglycans could be used as therapeutic adjuvants with antibiotics, for example, to reduce required dose of antibiotics or minimize their side effect. They could be integrated in wound dressings, in catheters or medical devices to prevent adhesion and biofilm formation. Phage therapy, as I told you before, could help to eliminate mucosal pathogens, and glycomimetics could be used as anti-adhesive therapeutics, but also as antimicrobial molecules for personalized medicines. So thank you very much for your attention. And of course, I would like to thank all my collaborators and the members of my teams. Thank you very much.
Okay, thank you very much, Catherine, for the very thoughtful presentation. So is there any question from the audience? Please, one question here.
Roberto Spurio, University of Camerino, Italy. I would like to ask you a question about what you've shown in terms of regulation of gene expression mediated by these mucin glycans. You have shown that in some cases there is a shutdown or in the expression of toxin or anyway a genetic response of the bacteria. So is it known that chain of reactions in general, at least in this model organism like Pseudomonas or Salmonella, that goes from interaction of these mucin glycans to a response inside the cell and then a response at the DNA level? So which are the steps, if they are known?
So I'm not sure they are known for each type of pathogens that I showed you in this talk. So this is mainly the work of Katharina Riebeck in the United States, and she usually what she is doing is to try some transcriptomic analysis to be able to describe the signaling pathways that lead to the attenuation of virulence of the different pathogens, depending on the type of glycans that are important for the interaction.
Okay, and the second question is looking at from the side of the bacteria, since they are much smarter than anything else. You mentioned that most likely there will be no resistance mechanism. But do you think this is really true because it has never been described? It's just an hypothesis or lack of evidence? Or because I guess bacteria have evolved over millions of years with this kind of molecules, and so they are well adapted also to react to this kind of molecules. So it's not synthetic material, it's something that they have co-lived together for a long time.
So you mean no resistance if we use mucinoglycans to attenuate virulence? So I guess there is no need for the bacteria to develop resistance because it's she will just become more commensal instead of pathogens if bacteria have to live in the mucus. So yeah, we can't be sure that bacteria won't evolve to develop some kind of resistance if we just use mucinoglycans. But I guess because we don't kill the bacteria, she will not need to develop this type of resistance. If we use some glycomimetics to kill the bacteria, as in the last example I showed you with cyalomimetics, maybe bacteria will be able to evolve and to develop in the future, I guess, some type of resistance, maybe, yeah. But there is no work that has been done today to be sure that it will not happen.
Thank you very much. Is there any other question from the audience? Nope, I have a short one for you. About the glycomimetics. How can you imagine that a single glycomimetics can target gram-negative, gram-positive, and mycobacteria that have vastly different cell wall organization?
That's a good question.
Thank you.
We don't have the answer today. We are, as you have seen, we have tried to do some transcriptomic analysis, and we are still trying to compare the effect of these heptapeptides on gram-positive and gram-negative bacteria by transcriptomic to see which could be the mechanisms leading to this. But because it can kill the membrane, we imagine that these heptapeptides cover the outer layer of the bacteria, and we have to find the mechanisms.
Thank you very much. So that will be all for this session, or not this session, this talk, and thank you very much, Catherine, again.
Thank you.
Okay, so we're gonna start the second talk, so that will be made by Elizabeth Valerio from the National Institute of Health in Lisbon, in Portugal, and she will discuss about the wastewater-based surveillance from hospital setting to animal farm.
Okay, but I have to.
So thank you to her very much.
With that one.
Okay, and if you don't need to show, we can show, but you change with that one.
Yeah. Okay. Hello, good afternoon. And first of all, I thank the organization for inviting me to be here, talking a little bit about wastewater-based surveillance on the within the One Health context. So I have divided my presentation in 6 parts. So first, since we are in One Health conference, we do know that One Health relies on 4 pillars, on human health, animal health, environmental health, and also the governance and coordination between all these interconnected pillars. So we must be aware, and that's what the One Health approach brought, was know that we now have, we always had, but now we are aware of the interconnection between the human, the animal, and the environmental health. So it's a collaborative and multi-sectoral approach. Regarding environmental health, we now must be aware, especially of the impact, as some talks already mentioned today, of climate change and pollution on environmental health, the impact of environmental changes on the disease, emergence and spread, on the water quality and sanitation systems, waste management, and pollution. So, and also the disruption of some ecosystems, and also the loss of biodiversity. Regarding wastewater-based surveillance, it is a powerful One Health tool that can help link human, animal, and environmental health just by monitoring pathogens and or chemical substances in sewage. It offers a foundation for providing early warning for known and also novel health threats. When we talk about the next pandemic, we expect it to be able to act before it gets in a way where COVID did because of these new early warning systems. And it can also allow us to measure the health community status through a non-invasive approach and also anonymous community level sampling, which is important to avoid discrimination. Now connecting a little bit to the ethics that we also mentioned in the morning. So COVID catalyzed scientists and public health professionals to revisit the wastewater-based and optimize resources allocation to mitigate the disease spread and also prevent other outbreaks. However, wastewater-based surveillance is something that dates back from the 19th century. If we look at this timeline, we know that in London, John Soe was the first to be able to establish a link between a water contamination and a human disease. And this was a really important topic. And then in the 1940s, in the USA, some efforts began to detect poliovirus, not only in water, but also in sewage. So it dates back already a long time. In 1999, we started to have PCR methods that are a big revolution to use in this approach. And then we had COVID-19, and now we have other projects that started having this base into mind. So we can, like I mentioned, we can monitor municipal, hospital, and even animal farms to track down pathogens, for example, viral, bacterial, fungal, and also antimicrobial resistant genes, for example, or even chemical substances. And why is it important and relevant? It is an efficient and sensitive method. Since one wastewater sample covers a large population, the same sample can be simultaneously analyzed for many different targets. We can use sequencing, metagenomics, chemical analysis, and it's really fast, so we can have like a close to real-time snapshot of the health community, and it allows an early warning, and this is especially relevant for the public health sector. So, of course, it can provide us, we have lots of health determinants that we can select, and that is the hard part, I will mention a little bit later. And since we can explore the exposure to environmental hazards, see trends in consumption of licit and illicit substances, the spread of infectious diseases, and also, like I mentioned, have an overall idea of the community health. Now I'm going to present two case studies that we are working on in Portugal. The first case is about a zoonotic disease. Zoonotic diseases are transmission of pathogens between animals and humans. It is necessary to have a surveillance, prevention, and also try to control zoonoses. And here are just some examples of zoonoses like influenza, rabies, and hepatitis E. And I'll focus on this one. And we know that we can and should monitor pathogen circulation among domestic animals, wildlife, and livestock, understand how wildlife reservoirs contribute to the disease emergence, and also promote diversity and ecosystems resilience to reduce the health risks. So regarding hepatitis E, it is an acute disease that causes inflammation of the liver. It is caused by this type of virus, which is a small virus, single-stranded RNA, and this has this capsid and no envelope. And it can infect a wide range of mammalian species, and it is estimated that around 20 million infections occur. Most are asymptomatic cases, but there are some deaths. There are 8 genotypes known. Genotype 1 and 2 infect only humans and are responsible for most human disease, which are caused by this virus worldwide. And it has been associated with large waterborne outbreaks. Genotype 3 and 4 circulate mainly among mammalian animals, including pigs, wild deer, birds and deers, and only occasionally infect humans. They have been occasionally detected in small food outbreaks in developed countries. It's not only something that occurs in underdeveloped countries. And genotypes 5 to 8 have been also described, 5 and 6 genotype only in wild birds and never so far associated with human infections. But in the case of 7 and 8 genotype, it has been detected in camels, and genotype 7 has been reported. There is a reported case of human infection. In humans, this infection causes mild asymptomatic illness and usually resolves itself without medical intervention. However, we must be aware that there are some persons with compromised immune systems, maybe due to HIV infection, chemotherapy, immunosuppressive therapy, and also that can allow the virus to cause severe forms of infection, liver failure, and even death. In here, it's just some representation of some of the symptoms which are common to any hepatitis. The average incubation period is long, between 15 to 16 days, and the recovery time may range from 1 to 4 weeks. So regarding the transmission routes, we see that we do have this animal reservoirs part, where we can have both the feces contaminating the water, for example. Here, the wildlife, the hunters may be a vulnerable risk because they have contact with the feces. Also, the undercooked meat can be a contamination route, and in here are highlighted some more vulnerable groups to this infection. Of course, that if the water is contaminated, it can somehow, the virus can accumulate and also come into contact with us. So we have a project ongoing since 2023, which is Project CVs, and aims to develop an integrated surveillance and alert system of zoonoses in Portugal, but it belongs to an European also project. And in our case, what I'm going to show here is tracking hepatitis E in pig farms, in wastewaters, and compare it with the results from the feces, also from the pigs, from the pig farms, and understand if it was endemic in Portugal, we were not aware, and how to guide public health interventions. So what we found so far was that the wastewater was actually quite good at like having an accumulation of the contribution from the farm. We detected more cases than only from the feces alone. And so far, all the genotypes that we detected, which are here, all belong to genotype 3, to subtype 3C and 3F, which usually infects pigs, wild boars, and deer. Here was just a reminder of what the genotypes, which animals they usually infect. This is still ongoing, and it's going to keep going. And we know that this is a multidisciplinary collaboration. We need a coordinated work between the veterinarians, the physicians, the microbiologists, the environmental scientists, and also the policymakers, because they need to take actions about these possible risks regarding detecting, in the cases where they detected positive signals for hepatitis E. Now, going to the other case study case that I wanted to talk about, it's linked to fungi that are related to AMR. So we had a project that has finished, which was called Agir project. Agir in Portuguese means act, to act. And it relied on giving the technical support for industries to optimize their wastewater pretreatment processes, and also to characterize wastewater from hospital units and improve epidemiological surveillance and optimize treatment processes, among other more technical stuff dealing with the management of wastewater treatment plants. And here were the institutions that were involved in this project. So regarding the work that we have done, where we participated, we intended to understand the spread of resistant microorganisms between humans, animals, and the environment due to the misuse of antimicrobials, and also very important in the case of fungi and antifungals, the lack of efficiency removal from many wastewater treatment plants. And we selected two fungi that are considered really relevant from the WHO. One is Candida auris, which we know that can be often resistant to antifungal medications. And it's also resistant to temperature and salinity and produces biofilms. So it's really hard to be eliminated from healthcare facilities. And can also be underseen because you're not expecting it for it to show up. And also the especially resistant to azoles, Aspergillus fumigatus sensu stricto, which has been reported in Europe, and where we know that the agriculture azole use has increased the resistance of these fungal species throughout the years. So in our cases, we have sampled several wastewater treatment plants from several hospitals within the Lisbon region, and we found that most samples were negative. We tested it for using PCR and also tried some culture, and we found 5 different hospitals positive for Candida auris, and also 2 wastewater treatment plants. We're not expecting this at all, since there are no reported cases, clinical reported cases of this infection. However, it may be underlooked because it's not expected, but it's probably already in the community through clean colonization, most probably. So having these results was important because we gave this information for the hospitals, and so they are aware that this was in circulation, and they should have a closer and special attention to the cases, to difficult cases. You probably would be a case where you had more than one infection in the patient, and so they are more aware of what they might be dealing in the facilities, and also gave aware for the hospitals to try to contain possible contamination of the surfaces and so on, and so to deal and manage the situation. Regarding Aspergillus fumigatus and stricto, in this case, first we have isolated the fungi here in plates. Here is a microscopic photograph of this. And then we confirmed its identification through molecular methods. And from the 127 samples we analyzed, most were negative. However, from 4 hospital samples, we detected these fungi, and also sometimes in some wastewater treatment plants. So as conclusions for this case study, it showed us that wastewaters of non-endemic areas, because that's what we consider to be, although there are a neighboring country that is endemic for this fungi, should monitor the presence and determination of the genetic clade and AMR of Candida auris, has a community and hospital early detection warning system, so that they can manage and mitigate possible future threats. That's what we intend to using wastewater-based surveillance. In this case, we saw that PCR and culture can be used. However, for Candida auris, since we are not endemic, qPCR is much more sensitive. And we also think that monitoring the wastewaters in general can help controlling and eliminating some nosocomial infections. I would like to stress that antifungals we don't have many, and it's really hard to find one, and if you have a resistance, it's really hard to treat that infection. So what we intend is to help the health authorities to assess the presence of these fungi in the population and design proper health protection policies. So as these projects are, and the other Agir became first, there is a new legislation regarding wastewater. So the urban wastewater was revisited, and now it will soon be applied to other countries, and it aims to protect human health and environment from the effects of untreated urban wastewater. Regarding, I do present all its aims, but now regarding a little bit closer to what I would like to talk to you today, it aims to improve water quality through stricter water treatment and the inclusion of new pollutants to be looked at, and also advance circularity through water reuse and recovery of available resources from wastewater. And it also requires that the EU countries to collect and to treat wastewater from all urban areas with more than 1,000 inhabitants. So it will be lots or most of the wastewater treatment plants will be affected. And they should monitor health parameters, still not defined which, just mentions 2, like SARS-CoV and also antimicrobial resistance. So at the same time that the directive was about to come out, there is a joint action that started, which is called eWish, which is EU wastewater integrated surveillance for public health. And what we intend under this joint action is to support the policy priority and strengthening the European Union capacities regarding the wastewater-based surveillance. It's a project that we are just ending the second year, and it will last another year. We have already on board 26 countries, 62 participants. So we have a public health sector and also research institutes. And regarding the new wastewater legislation, what we intend is also that the activities that are being developed under this joint action to support the member states in the preparation for the future implementation of this new urban wastewater directive. There are some challenges and opportunities for the future, if not obvious. So the first one will be to decide the possible list of health determinants candidates that we want to be analyzed and monitored under the wastewater-based surveillance, because we could look for everything, but there's no money to. And so we need to prioritize the most relevant ones, especially regarding the public health authorities. We need to give it a purpose, not just looking because it would be cool to look for. We need to establish harmonized and standard methods, so we all talk about the same thing. We are sure that the signals we detect, they are real, and then we take actions upon it. We need to develop new methods for new emerging pathogens that will come along, be prepared for the next pandemic. And regarding molecular methods, which will be the preferred ones because they are fast and sensitive for wastewater surveillance, we need to know and be aware that we need reference materials and also to ensure quality assurance between all of us. So some take-home messages I would like to share with you is that in a One Health context, wastewater-based surveillance does help to identify zoonotic pathogens circulating across species. We can use it to track down antimicrobial resistance shared between humans and livestock, and also being within the environment with the selective pressure that we'll put there. For example, when we discharge the wastewater treatment plant's effluent, if it's not treated enough, then we'll contribute for selection of more resistant species. And the goal for the future is to be able to integrate the data that we are obtaining already during this wastewater surveillance, the clinical surveillance, the veterinary, and the environmental monitoring, so that we can help the decision makers gain a more complete understanding of the disease dynamics, and so they can respond accordingly. Of course, I would like to acknowledge my team and also the projects that we have been working on. And thank you.
Thank you very much, Elisabeth, for this very nice summary of the issues about wastewater management. And do we have some question in the audience? You? I have a first one. And first, I want to thank you to mention the fungi, because most of the time when we talk about resistant bacteria, virus, and so on, we don't hear so much about the fungi. I was just wondering, so you choose Aspergillus fumigatus and Candida auris, while the most widespread and difficult to manage fungi is Candida albicans right now?
Not the most widespread. This particularly because they have more resistant mechanisms
Yes, so why did not you choose, well, include it in your survey, and can you survey?
Candida albicans?
Yes.
Okay.
And can you survey different subtype of albicans, because there are lots of genetic variation, so what is the granulometry of?
At first, we do have some results regarding Candida. We also look for it, but we expected it to be present like all the time. And so we were not sure what information we could retrieve from it. But now we would like to revisit that that species and also consider it and explore it a little bit more, because now we are aware that we can look at it in a different way and take it into account. So thank you for your comment.
Okay, thank you very much. So is there any question? Yes, please.
My name is Magali. I'm from TU Dresden. Thank you for your talk. And my question is regarding the new legislations and the new methods that will come. And the surveillance to work, or the way what it was for COVID, or you need to know what you are looking for. And in this standardization of methods, do you think that metagenomics should be part of them? Because if you don't know what you are looking for, it does not matter, or if you use qPCR or culture-based, because you are always look for the same stuff.
Thank you for your question. It's really a relevant one. Yes, especially if we are looking for the next new emergence, the new pathogen, we will only have that information using metagenomics. So metagenomics will be there for sure as one of the methods, especially to look for new pathogens, new antimicrobial resistance, because like you said, the others will allow us to be aware of the spreading that is going on in the community, but if you are looking for something new, you need to use other kind of strategies.
Okay, so if there is no question, oh yeah, one question, sorry, please. Hey, can you pass the mic, please?
Hello, my name is Sotirios. I'm from the University of Thessaly. Just a brief question that may be somehow associated to this. You studied both, let's say, with qPCR-based approaches and culture-based approaches, your Candida.
Sorry, can you?
Okay, during your study, did you evaluate the presence of Candida with both qPCR-based, culture-independent approaches and culture-based approaches?
Yes.
Did you find a strong association between the two methods?
In the case of Candida, it's really hard to have it cultured. So the positive cases that we had were detected only by qPCR, which was specific for Candida auris, not the other species. It was specific. But we also have really very low loads, so it's even more difficult to have it cultured, since it does not usually grow well, and if it's a very low load, it's even harder. Yeah.
Okay, thank you very much.
Thank you.
Okay, thank you very much for the nice question, and thank you again for your very nice presentation. Okay, obviously wastewater management is a very important issue, so we'll have a second presentation by Dr. Konrad von Orde, who should be here. Is he? Oh, okay. He's online. Thank you very much. So he's a head of service from Foodborne Pathogen Sciences in Belgium, and he will also give us a report about the SARS-CoV-2 wastewater surveillance in Belgium. Please.
Okay, good afternoon. I hope you can hear me.
Yes, should be okay.
Okay. Okay, thank you. So, yes, thank you for the introduction already. I'm Koenraad van Goorde, and as said, I'm partner in the Belgian National Wastewater Surveillance, and I'm glad that I can tell you a little bit more about our activities in this ONE Bridge conference, and I'd like to thank the organizing members for this opportunity to tell you a little bit more about what we are doing. So I hope everything will work fine. So before I will start my presentation, I'd like to ask a question to all of you with respect to wastewater, and I will try to start it, and I invite everyone to either scan the QR code or to join the link at feedbio.com and fill in the code. And the question, it's a very simple question. What words come to mind when you think about wastewater surveillance? So I will give you a few moments to answer that question. And I hope people will manage to join the question.
Okay, apparently the participant cannot write.
They cannot write?
Yes.
Okay.
No. Voting was stopped.
Okay.
This is what is written.
Is that right for you? We have a problem, yes.
Okay.
It is saying voting was stopped.
I see it also. And no, it should be open.
Yes, now it's okay.
Okay, good. Okay, I see already a lot of answers. If I see the screen isn't changing anymore, then I will close it, but I will give a little bit more time. Very diverse answers also. Good. I will, for those who are still trying to answer, I will close it anyway, and we will continue. So good, yes. So basically, as you can see, it's very diverse. And I'm quite sure that I already asked this question in previous conferences, and the answer evolves with time. And if I would have asked this question I would say 6 years ago, and then I'm talking about the end of 2019, just before the COVID pandemic, I think the answer would definitely have been completely different. When I asked this question in 2023, one of the main words popping up was COVID, SARS-CoV-2, normal, because if we talked about wastewater surveillance, that was one of the major things that was investigated in wastewater. But as you see, time has been moving on, and also I would say, the interest of wastewater and the use of wastewater has shifted. And now I'm going back to my presentation. Okay, this works fine. Good. So, but if we go back to 2020, wastewater surveillance, it already existed. But I dare to say that wastewater surveillance was not really a real surveillance. And with a real surveillance, I mean a surveillance with a certain regularity, routine analysis that are not limited in time or just in space. Those kind of studies were very limited. There were some studies, and in most cases, they were in the past linked to drug use, the consumption of alcohol and nicotine, exposure to metals, exposure to pesticides, also some studies on microorganisms, mainly, for example, polio, because if we know about one microorganism that was really important to monitor with wastewater, it was poliovirus, but it's changed a lot. Also for Belgium, in the beginning, or I would say before 2020, it was mainly linked to drugs, alcohol, but also a few studies, one about polio and one about salmonella isolates. If we go into literature, and I did a quick search just last week, well, of course you see, and I went back about 10 years ago to 2014, you see the number of publications on wastewater is limited. But then 2020 came with the SARS-CoV-2 pandemic, and you see a huge increase in the number of research papers that are linked to wastewater surveillance or monitoring, and clearly the increase was linked to the subject of COVID or the SARS-CoV-2 virus. We also see that, well, we are now a few years later, that the importance, I would say, of the coronavirus is gradually decreasing. On the other hand, the number of research on wastewater still remains very high. And this is, as I said already, the focus on the use of wastewater is gradually shifting. Not anymore COVID, but many more other things. And I saw amongst the words also AMR appearing. Indeed, AMR is one of the things that gains a lot of attention nowadays. Now, I was asked to tell a little bit on Belgian wastewater surveillance, what we did, how we did it, and how it evolved from just COVID surveillance to a tool that is now being used for much more. But I will just take you back a little bit in time, back into 2020, and it might be a coincidence, but in January 2020, Cienzano, we published a feasibility study for polio, because in Belgium at that moment, we didn't have an official declared polio-free status, and this because we didn't have any environmental surveillance in Belgium. On the other hand, in Belgium, there is the presence of a polio essential facility, meaning there is presence of infectious poliovirus material, and so we actually needed environmental surveillance. So at that moment, also the pandemic arrived, and so when we picked up the first signals that coronavirus was present in the feces of patients and that it could be found in wastewater, we started to think if it could be an option to also or to start looking for the coronavirus in the wastewater on a national scale. And so in September 2020, we got a green light from our public health officials that we could start with the wastewater surveillance. Briefly, it was 2 times a week. We analyzed the samples of 42 stations. We did it together with 2 partners, University of Antwerp and eBiome. And we had a coverage more or less about 45 to 50% of the Belgian population. Only Brussels, we could cover 100% of the Brussels population with 2 stations. So, but as the virus was quite active, we also were quite active. So very quickly, we shifted our PCR-based detection to a quantification of the virus, and we used the pepper mild mottled virus as a fecal indicator for correction factors. In 2021, we worked together with our colleagues from epidemiology on how can we explain the data, the results that we see in the wastewater to the people. So how can we go beyond those numbers of viruses in wastewater? What does it tell us? And so our colleagues from epidemiology, they developed 3 indicators, high circulation, fast increase, and increasing trend to help people understand what was ongoing. And more or less at the same time, we published our first dashboards, so also to allow people to monitor more or less in real time, because the data were uploaded twice a week with more or less 2 days delay compared to the analysis of the wastewater samples itself, but was quite in real time to monitor the evolution of the virus. And then also in that same year, we were also able to secure some funding from the European Commission to help us in the development of assays for variants, because as you probably can remember, the coronavirus was quite active in that a lot of different variants were seen after its appearance, and it was really not that easy always to track those variants. So with some funding from the Commission, that helped a lot. And then in 2022, we had our first publication on the surveillance. Many other publications followed on that. And then to go further, we also received from the International Association for National Public Health Institutes a kind of recognition of the work that we have been doing on the surveillance at a national scale. And then also a little bit later, in 2000, well, I would say the end of 2022, beginning of 2023, a lot was ongoing about the potential use of the wastewater from airplanes to try to track the import of COVID-19 variants into the countries. So this work that was done by colleagues from the University of Leuven, we from Cienzano, we decided not to go onto that pathway because at the end, first of all, it was not really easy to have access to the airplanes, but also at a given moment, you need to decide which airplanes, from where they are coming, so quite complicated. So we decided to monitor the wastewater from the airport itself. So basically it captures everything that's arrived. It's more diluted, of course, but it gives a more representative picture of the things that arrive in Belgium. And at the same moment, we started also the genomic surveillance via sequencing, also again to monitor the variants, because with the PCR-based method, we were always like lagging one step behind. And so using sequencing, we could more or less sequence in real time also. To continue, the more we were using the surveillance data, our data were also integrated in a weekly report where all data on acute respiratory infections were gathered. So actually, data from the wastewater surveillance were also considered very useful and were also integrated into that report. We shifted from RT-qPCR to absolute quantification via digital PCR, and that's also the method now that we are using for all other kinds of microbiological parameters that we are using for wastewater analysis, but more on that later. And in October 23, we had a major upgrade of our dashboard, making it much more interactive for the users and also much more informative. Also, from September 23, our data were used by the risk assessment group. These are the people that use the data for assessing potential risks, and they will give recommendations to the public health managers who make then the decisions. So it was really for us, nice to know that our data were also taken into consideration for the risk assessment. And then also, which was a nice event, is that our work was picked up internationally, amongst others, by Bill Gates, who by his foundation is very active in the area of water sanitation and hygiene. So he visited our lab, and yeah, so it was a nice moment. And then in January 24, we extended our surveillance with influenza and RSV. Only then also in January 24, we started with routine polio surveillance. We also started with a wastewater repository. The only thing, I would say negative thing, was that due to a decrease in funding, we also had to decrease the coverage. So instead of 42 stations, we were only analyzing 30 stations, and this on a weekly basis. So not twice a week, but only once a week. We started to participate in the EU-WISH joint action. I think many of you are familiar with. And then by the end of 2025, so I'm talking about this winter season, we are now also quantifying influenza and RSV, whereas when we started, it was just a detection. So this brings me to quite, I would say, a short summary of why are we doing wastewater surveillance in Belgium. So, well, it's clear that it's a very informative method that gives a representative picture about what is circulating. It's also non-invasive, and people that are not sick or I would say independent of the testing, because not everyone is getting tested for a certain virus or parameter, wastewater might pick it up, and it could be used for early detection possibilities. In addition, it is complementing existing surveillance systems, and it can potentially fill certain gaps. I will give an example about that later on. And very interesting, although, well, for Belgium already, is that basically the results of the wastewater surveillance are integrated in the risk assessment, which is then used for risk management purposes. And so because I would say till 2024, we were mainly focusing on the SARS coronavirus, but between 24 and 26 now, we have been doing continuously routine surveillance, the continuously development of new assays for new parameters, and we are doing some ad hoc surveillance assays also. Now, it's clear that wastewater really fits within the boundaries of a One Health dimension. And if I'm looking at Belgium, what is being done or what has been done, it's very diverse. And I give here a few examples, but wastewater goes beyond the things that you can see here. Wastewater, it's not waste, it's a huge information, it's a huge source of information. And so in Belgium, in green now, I'm not going too much again on that, but that's what we have been doing so far. But there are ongoing research is ongoing on many other things beyond microorganisms. It's biomarkers, drugs, metals, also antimicrobial resistance. So it goes much further than that. Now, I was asked also to tell a little bit more about, okay, now in Belgium you have established wastewater surveillance, but how or what can you do more than just the routine pandemic surveillance? What else can you do? And I will try to explain a little bit on the possibilities of wastewater beyond that by 3-4 examples. And the first example is monkeypox. Now, in January 2024, so almost 2 years ago, we picked up some, I would say, concerning signals from the Democratic Republic of Congo that for an ongoing monkeypox outbreak. And so we decided here in Belgium to start working on the development of an assay to start thinking about, okay, imagine that there could be an import from the Democratic Republic of Congo to Belgium. Where should we monitor the wastewater? And this all in a context of preparedness. And so to develop the assays, we did a pilot study. And for that pilot study, we used wastewater samples from a 2022 monkeypox outbreak. And in green, you see the number of human cases in Belgium diagnosed with monkeypox. And so from that period, we sampled some wastewater samples, and we were, at the end, quite successful because some samples turned out also to be positive. Why do I say quite successful? Because, well, we do think, and it has been acknowledged by other people, that the monkeypox virus is not really stable for storage. So already you lose a little bit of signal. So we were quite confident that if we would have fresh material, we would find the monkeypox virus. So, but that's a little bit the start. So a few months later, the news becomes even more disturbing because there are two different clades circulating, of which one is a new clade 1b that is more virulent, and is also the first time that clade 1 is associated with sexual transmission. So what's quite concerning also the fact that there is a direct connection between the DRC and Belgium, also between some of the neighboring countries of the DRC. So there is, or there was, an actual risk of import of the virus into Belgium. So at that moment, we were contacted by the risk assessment group if we could develop a method and if we could start monitoring wastewater for monkeypox. And so in May 2024, based on the work we had been doing starting in January, we had an assay that was developed, validated. We actually could start analyzing wastewater when our public health authorities asked to do it. Now, just to explain a little bit the figure on the right. So on top, you see again the number of cases in 2022, the outbreak we had then. you see very few cases in 2023, and then also in 2024 you see a few cases, but much lower than the outbreak we had in 2022. And so, to make a long story short, during that period that we were monitoring in Belgium, from week 24 to more or less week 48, we analyzed using two different methods based on the liquid phase and the solid phase. At the end, we did not find any monkeypox in the wastewater samples that we analyzed. But again, as a short remark, is that the number of human cases were very low, not even attending 5 cases. So it would have been very difficult anyway to find the monkeypox in the wastewater. But in the context of preparedness, we have a tool that is ready, and if we are contacted by our public health authorities and they ask us to start analyzing, we can start. And this is the case now in November 2025. Again, member states in the European Union are saying that the Mpox clade 1b is circulating, for example, also in the Netherlands. And so we started again screening our wastewater samples for monkeypox, whether or not we can find an introduction or circulating among the Belgian population. And we are now using the nanotrap method for that. A second case is polio. It's just to describe the fact that now that we have a wastewater system in place, it's easy to scale up or to adjust something that is in place. And at the end of 2024, beginning 2025, several European member states, they communicated that the vaccine-derived poliovirus 2 was detected in their wastewater, and the fact that it was also found in Germany close to the Belgian border, it was decided to add one additional wastewater treatment plant into the routine surveillance. And in the meantime, we also increased the sensitivity of our methods. Important questions that at that moment were raised were that imagine that a human case would be found in an area that is not covered by the wastewater surveillance. Should we include the nearest wastewater treatment plant? A second question, imagine that there would be an outbreak. Okay, what are we going to do? Will we add additional wastewater treatment plants? And will we increase the frequency? These are questions that, of course, need to be taken into account from an epidemiological point of perspective. But even if the question would be yes to all those questions, it would not really be a problem because we have a system in place that can really scale up, that can move forward if needed from a public health perspective. And then the last case is measles. Belgium is since 2020 certified for an elimination of measles. Still, we have within the European Union the third highest notification rate of measles. So that could be that there is some gap in our surveillance, and we have been thinking of using wastewater as an additional complementary tool for measles monitoring. And then, especially from a perspective, if we do not find any measles in the environment, that it is an indication that there is no measles circulating among the Belgian population. So a little bit a similar approach as we do for polio. And so we recently started with that in the mid of 2025. And what we see is that, well, first of all, we can easily detect the measles virus in wastewater, that they mirror what is seen with clinical surveillance. And now we are thinking again to extend it to increase the coverage of the population. Again, something that is only possible because you already have a system in place that allows you to adapt accordingly. Now, to finish, I would like to show our third update of our dashboards. So I've already talked in the beginning when I was explaining a little bit on the history, we had 2 dashboards. In the meantime, there's a third dashboard. And for the people that are interested, you have the QR code on the left if you want to visit it. But it's a very, I would say, informative, of course, but people can play with it, and you see, and I'm going to try it with the marker. Yes, you see the different areas that are monitored or the treatment plants, so you can click on the different regions, and then you will see on the right the corresponding dynamics of the virus. You can also see how the levels are being interpreted. So if there is a high circulation or very high, and these data, again, are being used also by our public health institutes. Maybe also very interesting, well, I think something that we can be very proud of. Since the beginning of the surveillance, we have been analyzing almost 130,000 samples. And these are only the samples that have been documented for the surveillance. I'm not talking about, I think I'm talking too long. Oh. Yes, I saw something with the connection. I hope I'm still connected.
Yeah, everything's okay.
Okay, sorry, I got a message like my internet, a slow connection. Sorry, I'm almost finished. So I would say have a visit of our dashboard, but then again, to finish completely, we in our Public Health Institute, we have an open data policy also, which means that all our data from our national surveillance is actually accessible to everyone. And so people can use the data to develop their own tools. And one example, you have the link here on top, www.eotsolutions.be, Internet of Things. Basically, those are people that are using our data to develop a kind of alternative dashboard or barometer that is used by hospitals or can be used by hospitals or nursing homes so that people easily can see, oh, there is a high circulation of a virus, maybe we should be a little bit more careful. There's also here in the context of, I would say, maybe a little bit preparedness, awareness among the population, just to show how data can be used on different levels. That was the last example, so I just need to thank all of the people at Cienzano that are involved in some of the work that I showed, because it's much more. We work on AMR, we work on vector-borne diseases also, but it's another story. And on the right are stakeholders and public health actors that are also involved or that are being consulted in the work that we are doing. And thank you for your attention.
Okay, thank you very much, Conrad, for the very nice presentation and very complete data set. So do we have some question from the audience first? Oh, yes, one question here, Yannick.
Hello. Oh yeah, we can hear me.
Yes.
That's a very naive question, because if I understood correctly, when you want to study viruses, you need to do PCR first. Is that correct?
Well, we use mainly PCR, yes.
Just my question is, if you want to maybe not miss any new viruses, how you can proceed? Maybe new method of sequencing or something like that?
Yes, well, it's a good remark. That's also what I briefly said. So basically, in the beginning of our work, we were only using PCR, and we were always like one leg behind because the virus mutated quite quickly. So there was a new variant, and if we wanted really to quantify that new variant, we needed first to develop the new assay, and we were always a little bit behind. So with the clinical surveillance, they were sequencing immediately, so they already knew immediately when there was a new variant. We we were a little bit behind on that level. So that's why we started to sequence the wastewater itself also. When we saw that there was the virus started to circulate again, then we started to sequence to immediately capture the different variants that were present in a sample. So we did both. We did PCR to have an idea about the quantification, but we also sequenced.
Thank you.
Okay, I have another question from Klein. Oh, one here, please. Yes.
My name is Apostolos Veizis. I'm a medical doctor and working for Intersos. Thank you very much for the presentation. Just in order to understand a bit the experience of Belgium before I talk about my country. So when you do the analysis of whatever you found on the wastewater, be that drugs, alcohol, measles, COVID, etc., in addition to doing your analysis, etc., how far this information it is feeding the government authorities and the response, and how this is being presented to the people? Because, I'm sorry for being long, but in my country it's just media reporting, we saw an increase of drugs in this level, and then what, what does it mean? Thank you.
Yes, well, about the drugs, that I cannot tell much about. How do I, well, I'm a microbiology lab, so all the data that I showed is viral, microbiological, so the drug analysis, alcohol, is being done by another lab, but they worked, there is some European project that allows them to analyze some samples on an annual basis, and that allows to see some trends in use of certain drugs. Now, with the wastewater surveillance that is in place, they also have access to much more samples. So they have been doing much more analysis the last couple of years, generating much more information. So what I do know is that those data are really picked up much more than any microbiological data. As soon as they publish something about drugs, all newspapers, they all jump on it because they want to know what's happening. And so the first thing that happens with those data is a kind of sensibilization. And it's also important for, I would say, well, public health, but maybe also some other instances will use those data to discover new trends, maybe new drugs that are starting to circulate. So that's info that they can use for themselves to be aware about something new that is happening. Actual actions that are taken, that I do not know, because it's not my area of expertise. When I'm talking about the SARS-CoV-2 virus and influenza and RSV, the data that we produce, imagine that we would see now the next couple of weeks a huge increase in the number of influenza circulating. Those data will be taken together with other surveillance data, and it will be used by the people from the risk assessment group who will say, okay, we are now in a period of high circulation. We will contact the general practitioners that they need to be aware of the fact that there is much more virus circulating. We will inform the hospitals that there might be need for much more beds in some care units. So it's really used to inform people that they should then be more aware of certain things. And you never know, but imagine that something really, that it would become very problematic. I think maybe some other measures could be implemented then again, but let's hope we are not going into that direction anymore. I hope it's more or less a question, an answer to your question.
Okay, thank you.
We have a question from the Zoom audience, if possible. From Maria Himonopoulou, she's asking, why wasn't wastewater surveillance for coronavirus conducted in 100% of Belgium's territory during the pandemic? Why wasn't wastewater surveillance for coronavirus conducted in 100% of Belgium's territory during the pandemic?
Yes, that's a good question. Well, there are two answers to that question. The first answer is that with the more or less 50%, we were able to cover that with 42 stations. If we would want to go to 60%, that would mean that we would need to add 100 stations more. It's not linear, but the higher the percentage of coverage you need, the more and more stations you need to cover, because you then need smaller wastewater treatment plants only covering like a few thousand of people. So it would become very difficult, and linked to that is a budgetary element. Of course, we could be able to do it, but then it would cost much, much more than it costs now.
Okay, thank you very much. So no question anymore online, no from the floor. So I thank you very much, Conrad, for your very nice presentation and for the answers.
Thank you.
And I'll thank again all the speakers for this session, and we'll have a coffee break now. And maybe Zoe, can you tell us more about what's happening after?
After, we have a presentation of the ONE Bridge workshops about what happened and the outcomes of these workshops. And then there's going to be a roundtable entitled Migration and One Health, Interconnected Challenges, and a workshop When One Health Meets Our Ethics. So now we have a coffee break. Thank you very much. Goodbye. Thank you, Mr. Van Hoorde.
Hello, everybody. I would like to welcome you and thank you for your presence here. In the first part of our sections, representatives from the ONE Bridge workshops that took place in the previous 2 months will present and will talk about the topics developed and the outcomes. We will start with the Technical University of Dresden. And please, Uli Klamber, Dr. Uli Klamber, senior researcher in the Department of Hydrology, will present the corresponding workshop.
All right, thank you. So I'm happy to present what we did in Dresden during our workshop that we organized, which had the title Making Environmental AMR Surveillance Fit for Purpose, Data Integration and the Ecology of Resistance. So why did we do that? I think we heard today already in 1 or 2 talks about the problem of antimicrobial resistance, which is already connected to 4.7 million deaths a year, and it's predicted to get a lot worse with time if we don't do something about it. So we definitely need surveillance to know what's going on, because we can only fight what we actually know to be there. At the same time, some people might disagree, but we definitely can't survey everything. So surveillance should really be targeted at something, and if we target it, it should be targeted at risks. However, that might not be so easy in the environment. So if we think about clinical or veterinary surveillance, we deal mainly with these short-term risks, which is basically worsened patient health outcomes and treatment failure. So to do that, in the clinic, it makes a lot of sense to look for the abundance of pathogens or look at resistance patterns of the pathogens so we know how can we treat them, what do we need to do to basically avoid that a patient continues to be sick, dies, and so on. So basically, if we think on the pathway from clinical veterinary surveillance towards identifying risk, it's rather a straight road. And if we know the abundance of pathogens, the diversity of them, and the diversity and abundance of resistance, we can basically estimate what is the risk of this infection and what's the risk of the connected antimicrobial resistance in the pathogen. However, which it might be very appropriate for us being located in Greece, if we think about environmental surveillance and we want to identify risk, we reach a labyrinth. What is in this labyrinth? There's actually a lot more than when we talk about clinical or veterinary surveillance. So there might be abundance, diversity hosts, just like in the clinic, but there's also other things like the invasion and persistence of resistant pathogens from the human sphere into the environment. Do they stay there? There is competition, selection. Do resistant bacteria do better? Do they worse? And how do they compete with the naturally occurring bacteria? There is horizontal gene transfer and mobility of resistant genes that might undergo several different switches of hosts. There is host shifts, as the emergence of new resistance, where basically environmental bacteria have already some resistant genes, and they suddenly move towards clinically relevant organisms. And there is obviously, to go to the proper risk, exposure to the human sphere, the veterinary sphere, and afterwards colonization, and by that potential future treatment failure. So if we deal with long-term risk and complex communities in the environment, what can we actually do? Well, we can, for example, like we heard today, with wastewater influence surveillance or effluent surveillance, look at community carriage of AMR and AMR pathogens. We can, for example, if we do bathing water surveillance, look at the exposure of humans to AMR pathogens through the environment. We can also investigate how does AMR spread in the environment by looking at persistence, look for selection pressures, reveal hosts and potential future hosts. We can identify the genetic mobile elements that are involved in transferring them into the human sphere. And then we can look at
what do we find in the environment as newly emergent resistant genes that have not made it yet towards us, but they probably get enriched already. So if we do that, we can gain actionable insight for regulation and mitigation efforts to limit AMR impacts. But it's probably not so easy. However, we've recently made quite a few important policy advances, at least if we think about what we already heard about the wastewater new regulation by the EU. That, for example, says in relation to AMR, we should have environmental monitoring, so we should collect and analyze wastewater from all wastewater treatment plants that serve more than 100,000 people and track the prevalence of AMR in there. It also says we should have a public health integration, so we should use this data to inform public health decisions and support surveillance systems. We then have in there the need till 2030 to introduce a fourth treatment step to decrease micropollutant removal. Oh, that was already part of there. 2045, not 2030. And then we need integrated water management by requiring that urban wastewater management plans for larger agglomerations, so these wastewater treatment plants serve in more than 100,000 citizens, that they selectively encourage a holistic approach to controlling pollutants, including those related to AMR. However, there's still a lot of questions around this topic. What are the right methods? How to integrate it? Which mechanisms do we really want to survey? What is going on with regards to selective compounds? How do we control those? And so on. And this is where the Dresden workshop started. Basically based on, okay, what is currently there and how can we make it better to gain these actionable insights? So for that, we brought together quite a few experts, including from German research institutions, international research institutions, practitioners, for example, from wastewater management of Dresden, and some German governmental agencies like the Environment Agency and the Federal Institute for Material Testing and Research. So in Dresden, we had around 30 people and around 50 online, representing people from a total of 12 countries, which was not too bad as like a critical mass to get thinking together. We had scientific sessions, for example, on selection pressures, the general ecology of AMR, reservoirs, mobility mechanisms. We had a policy session on the integration into governance, environmental threshold concentrations, data platforms, how do we bring all that together? We had expert roundtable regarding which targets might be good, what actually is needed to make data actionable, how to implement it. And we had some breakout group work where we identified gaps, priorities, and implementation pathways. So I just want to present a few of the things that came out of the Dresden workshop as results. For example, the surveillance toolbox. So one important thing to actually be able to use this data and reuse this data is that we have standardized surveillance protocols, and most importantly, that they are similar-ish and comparable across the different EU nations. Or if we talk about Germany, for example, across the federal states, which is still a big problem. So if we think about the current main toolbox that we use, it's mainly consisting of culturing certain bugs selectively and with and without antibiotics. We have qPCR to look for certain genes, and we've got metagenomics to look more broadly at the entire community and what's in there. However, that might not be enough to gain actionable insight. Is it really enough just to know what is there and how often is it there? And all of these methods also come with clear strengths and weaknesses. For example, qPCR is very sensitive, so is culturing if you know what you're looking for. Metagenomics is not. But at the same time, if you want information on the genetic background of anything, you need to go for metagenomics because the other methods don't really give you that. And so it's a good question of like, so what is the right thing to choose? And one of the things we discussed in Dresden was, for example, modular workflows that give you very clear goals that you want to achieve, then move in towards a first method. That might be, for example, if we want to quantify total AMR load, let's first culture some fecal indicator to look, is there at all any human-made pollution in there? If it's above a threshold, you might want to go for resistant E. coli, or you want to move to qPCR of certain target genes. Again, if you reach a certain threshold, you might want to go to the chemistry to look for selective concentrations. Afterwards, move to either large cell qPCR or metagenomes. And so depending on what is actually the goal of your surveillance, you might want to use a different path with standardized protocols. At the same time, we also were thinking of, okay, what is missing? So how do we extend the current toolbox? And in science, we already use quite a few novel tools. For example, things about integrating mobility and risk assessment. So I think in the clinic or veterinary sphere, it doesn't really matter too much if an antibiotic resistant gene is mobile, because we treat the pathogen that currently has it. But in the environment, stuff moves around between the different spheres before it goes back to the humans. So bacteria are able to undergo horizontal gene transfer and share the resistance genes. And very often things that were enriched in humans or food animals are not able to survive in, for example, soil or aquatic environments, but they for sure can leave behind their genetic load. So for the environment, the host might not matter too much. It might rather be mobility that is a main risk determinant. And so we're thinking about, and we used part of the preparation of the workshop also to refine something where we currently wrote an opinion piece on, find the QR code down here, that in addition to the current environmental AMR toolbox, which I already told you, gives you abundances, pathogen abundance, community composition, there is a lot of advanced methods that have been developed in at least the scientific sphere of environmental AMR surveillance. For example, contact-based analysis of metagenomics, where you assemble genomes back together and look, okay, where is the AMR actually encoded? We have ddPCR linkage analysis. ddPCR, you can multiplex and look for, do plasmid genes occur together with antibiotic-resistant genes? You have ARG amplicon sequencing to actually look for, is it the same variant that is shared across different environments, or is it the same gene, but different variants of it, so you can source track. You can use long-read sequencing, high-C metagenomics to look for plasmid associations, for example. And by that, you get not only ARG-MG abundance, but you get genomic contextualization, quantitative ARG-MG linkage to be able to move towards risk. At the same time, and that is true for everything, we have far improving databases on resistant genes, on mobile genetic elements, on reference genomes, human animal pathogens, but also on mobile determinants like which plasmids have really big host ranges that can transfer, which ones have very high transfer rates, what are the costs of being resistant and hosting such a plasmid. And I think if we combine all of these, we can move towards quantitative microbial risk assessment. The first models that integrate mobility and abundances and pathogens are already out there. They definitely need refinement. And what they especially need is more data to be able to actually parameterize them. But the methods are all there. And I think if we generate more data and move towards this whole thing, we can actually get refined risk models. We can go to good decision making where risks can actually be identified and lead to policy invention and regulation. Then finally, we talked also about regulating pharmaceuticals. So there's threshold concentrations to limit the selection for AMR. And we currently work together already with the German Environmental Agency on this. And basically one of the big questions is, how do we decide what is an acceptable concentration of an antibiotic in the environment? And so a lot of you might have seen this graph, which basically shows growth rates of a susceptible strain and its resistant counterpart that is exactly the same, except that it's resistant to an antibiotic across an antibiotic gradient. So you basically have the susceptible strain dying, the resistant strain survives, and probably dies somewhere far later at a far higher MIC. However, usually resistance, as I said before, comes with a slight cost in growth rate. So in the absence of antibiotics, the susceptible strain does better and resistance doesn't spread. So what we really want to know is, how do we ensure that we can identify this spot where for the first time the resistant strain grows better than the susceptible one? And so one of the things we talked about was this new ecology-based approach to predict no effect concentrations. So far, what people have done mostly is they looked at the EUCAS database, looked for the lowest MIC that's known, and just randomly put a factor 10 as safety on that and said, okay, that's probably good enough to go for minimal selective concentrations, calling that then the predicted no effect concentration, and that's what they use. So we now propose that rather than doing that, we should again take the lowest MIC, but we should multiply it with the likelihood of a certain cost of resistance to appear. So we propose that basically the cost of resistance, if you multiply this with the MIC, we get exactly this concentration, which is mainly because this here is a logarithmic scale. Otherwise, they would just be two lines that intersect with each other, and based on basic 9th grade math, you know that that is probably true if the lines stay as linear as possible. So we created quite a data set of different bacteria with different resistances. We looked for if we measure the minimal selective concentration in competition compared to if we just calculated based on knowing what's the cost of resistance and what's the MIC of the susceptible strain. And you can see here that that concept worked very well. And based on likelihood of cost distributions, we are then able to put this in here. And we propose that PNEC-RES for 95% of the cases should be MIC lowest times 1 divided by 250 rather than 1 by 10, which is based on that only 5% of resistant genes have a lower cost than 0.4% or 1 divided by 250. So, making environmental AMR surveillance fit for purpose. This was not everything that we did at the workshop. We had a few other things, for example, how to include biocides, for example, as non-antibiotic compounds into surveillance and ecology, and some other parts. But I think these were three of the highlight things that we talked about there. So what do we need to do to make environmental AMR surveillance fit for purpose? And when I talk about environmental AMR surveillance, I talk not only about wastewater surveillance, but about surveillance also of surface waters, soils, and so on. So we need to first define and regulate safe environmental limits of AMR, antibiotics, and co-selective agents, which is probably the easiest part of it because it's mostly based on numbers and chemistry. We need to set up standardized surveillance protocols and targets to generate meaningful environmental AMR data, because only with sufficient data available can we be evaluating risk and make the data meaningful. We need to evaluate, improve, and develop surveillance methods, continue to develop new ones, and define which targets are actually the most meaningful for environmental AMR surveillance to yield actionable insights, but also to keep it financially feasible. So if we have one target that can actually give the same information as 10 others, it's probably not worth doing all 11. And then we need to integrate the data into quantitative risk assessment models to evaluate environmental AMR risks in the absence of direct measures, such as, for example, treatment failure. So finally, all this data from wastewater surveillance should be integrated into databases to inform public health decisions and make it interoperable with data from the other One Health compartments, where such databases quite regularly exist in different EU countries already. With that said, that's at least most of the people that attended the workshop who are currently going on with the group work. Thank you. Is there any questions for the Dresden workshop?
Thank you, Dr. Klumper. Is there any question? I hope setting limits, we will tackle the AMR. So we will continue with the next presentation. Thank you. Thank you very much. From East Global, from Barcelona, two researchers, Marina Taurus and Carol Delgado will present the outcomes from the workshop in Barcelona. Thank you.
Thank you very much. Hi, everyone. We are going to introduce it on behalf of Sara Soto, who couldn't attend to the workshop or the conference here today, so we are glad that you invited us instead. The workshop that we organized in Barcelona happened just a couple of weeks ago, and it was titled like this: Human Public Health and Antimicrobial Resistance, What Do We Need to Know? Just to start a bit, it's a bit of a different presentation, but we wanted to introduce also who are we and why did we join and why did we organize this workshop. We come from IS Global, as we were introduced. It's a research center based on global health, very targeting the human global health. And for it is organized by several research programs that include climate and urban pollution, environmental and health, environment and health over the life course, malaria and other neglected parasitic diseases, maternal, child and reproductive health, and where we belong is the program on global viral and bacterial infections led by Sara Soto. That is focused on these research lines on new drug discovery, and one of the most innovative ones is the one that she's carrying on gold particles. How can it help to distribute or target, to make the antibiotics target better bacterial microorganisms? Also on wastewater surveillance on treatment plants around the city of Barcelona. As well, the group is also leading research lines on new tools for diagnosis and prevention of major viral and bacterial infections, such as the AMR Detect tool, to understand immune response and the microbiota action against these infections, viral and bacterial, and to develop and assess strategies to deliver or proven effective interventions. I forgot that animation part. Within this program, there's the Antimicrobial Resistance Initiative, which is directed by Professor Jordi Vila and coordinated by Elisabet Giral and myself. And we are focused on all this research evidence created in the lab to translate it into policy and global suggestions, and sometimes they are more successful than others, but as well on actions for training and dissemination among citizens, and also participatory research, so we can feed back the laboratory research carried out by the group of Sara Soto. And just to describe as well a bit of the actions that we carry out are mainly targeting these main key factors that are considered that are more relevant behind the AMR problematic today. So we tackle the overprescription, and we work a lot with healthcare professionals on their training and their involvement on governmental programs that actually Carol will talk to you later, because we discussed them in the workshop, as well as on antibiotic dispensing without prescription to try to prevent that, which is still it's still done in Spain, like in a lot of southern European countries, to also prevent self-medication and to improve the knowledge on antibiotics and infections, as well as to foster the discovery of new treatments. Moving on to the workshop that was titled, as again, Human Public Health and Antimicrobial Resistance, What Do We Need to Know? It was held in November, some of you were there, and it was held in Barcelona. And now Carol will help me out on introducing you. What did we discuss?
Okay. In this part, I presented you one abstract where we follow within this program. I'm beginning with one welcome for the Dr. Sara Soto, who provides some context for public health and scenarios on antimicrobial resistance. And the session one specifically focuses on the general context on One Health in public health scenarios, and specifically on projects that focus on this topic, such as the project Rambla, as well as citizen participation, and the project, for example, ANR Educare.
It's more dynamic like this, if we do it like this. The second session was more focused on the current measures that are in Catalonia being implemented to combat AMR, although the word combat, we are trying to avoid it because it's quite warish style, but to fight it. On one hand, we had a representative of Hospital del Mar, one of the biggest hospitals in our city, that was sharing with us the measures in hospitals around the city for infection prevention and control and stewardship of AMR, as well as on intermediate care as well from the Catalan Health Consortium, and also the AMR measures in primary care that was presented by the director of the program on antibiotic optimization optimal use, sorry.
Okay, in the section 3, discussing the European Commission, their type on the One Health concept, which focuses on the multisectorial and interdisciplinary collaboration to address global challenges such as antimicrobial resistance, and the crucial aspect for the success of the One Health, which is cooperation at all levels, from the local to the global.
The last session of that day was targeting more the role of public health departments in AMR, and it was in a roundtable dynamic or setup, and it involved from academia to the Drug Agency of Spain, so more governmental representatives to discuss actually what can be done or what can be improved in this collaboration between research and governmental agencies, finalizing with the closing and final remarks.
Okay. And then day 2, we look at that one overview of all activities in the plan, and the plan is the national plan against antibiotic resistance in Spain. On the One Health and their impact science plan have been founded in 2014, and the one relevant point was highlighted is in difficult of changing legislation so that the doses purchased at the pharmacy are equivalent to the doses prescribed by the general practitioner. And for example, commonly in Spain, the prescribed treatment is usually, for example, 3 tablets for 7 days, and in total is 21 tablets. But currently in Spain, boxes of the 20 tablets are sold, and which means that these 2 boxes must be prescribed instead for 1 due of the missing date. Dose.
Which actually, maybe if there's no questions from your side, I have one for you later, but first we finish this. When it comes to the key points out of it, well, one that it's not written here is that the attendance we were all quite profiled on the same field of work, mainly research, so unfortunately we didn't bring patient associations or governmental agencies, we couldn't attract them. But we did reach this key point among ourselves that, as you can read, the current situation regarding multi-drug resistant microorganism and population characteristics need the implementation of actual functional antimicrobial stewardship and infection control and prevention measures.
And the second key point, it is needed to have access to new antibiotics.
And innovative, yeah. And then that a successful national action plan to avoid antimicrobial resistance or to reduce it has to have the following characteristics: the One Health approach, to include surveillance, as we mentioned, and stakeholder representatives as we need since the very beginning, and to participate in EU and global initiatives such as this one with measurable and achievable objectives, and to include communication and training for non-experts as well. This is also a picture of our workshop, which some of you can recognize yourself, sorry, and thank you.
Thank you.
Thank you very much for your presentation. Is there any question?
Well, if not, I have one, which is a bit, but because we were wondering, in Spain, as Carol explained, when it is prescribed the antibiotic treatment, right? So they give us this prescription, you need 20 pills, but then we have to buy 2 boxes for it, so there's no dosing from the pharmaceutical, from the pharmacist. Is that in your country's case as well? Like you have to buy way more pills than what you need, what are prescribed, or actually are they dosed as the prescription is required?
I'm from Mexico, and Mexico is the same case. So you always have way more than you need.
Yeah.
And then this is when it happens that, then you get sick again, or people get sick again and say, I have some leftovers.
Yeah.
or they are like, it's not well regulated. And then some doctors are like, or people say he's not a good doctor because it does not prescribe antibiotics.
Yeah. Which is, making some time in the meantime. It is a big deal with the pharmaceuticals to deal with these industries as well, to shorten as well the treatment, because of course then the profit is shortened as well, which is one of the barriers we are finding.
Yeah, I think, thank you very much for the presentation and the antibiotic resistance. I think for my country, that's Greece, one of the biggest problems, we're the champions in Europe, so, respect to us, was that until recently, the antibiotics could be purchased in any pharmacy without any prescription. So that was also a problem of the use of antibiotics, and people just will go and buy whatever they were thinking about. So this created, of course, this chain of stuff.
How recent?
It's a few months.
Really?
Yes. So the idea behind is that out of this discussion regarding the general concern that we need new antibiotics, also let me add one also small element. I used to work for an organization called Médecins Sans Frontières, Médicos Sin Fronteras. So the reason that today, for example, we don't have new antibiotics is because there is no investment on research and development. I mean, theoretically, we need to work in parallel systems. And of course, the price and what the companies present as a high cost for research and development, it's not true. 83% of the money that pharmaceutical companies get to do research and development is public money. And at the end of the story, the pharmaceutical companies, they take this as a monopoly of 20 years, and they sell it as their own product with very high prices. So I think overall there is a process in the middle, has to involve the process of transparency of the use of antibiotic, education about it, etc., etc. So there is a whole process, and it's not enough just having new antibiotics, because if we don't use them properly, or if we don't identify the process on the way, then we'll be coming back again in a month from now, we need new antibiotic. That's not the solution. So I think we need to combine all those efforts with the elements that we discussed before. Thank you.
I can just describe the case in Norway. It's very strict. So you get exactly the number of tablets that you need. And they also change the packaging all the time so that it's not possible for people to copy. So it's the complete opposite.
Yeah. Well, great example. Yeah.
I can also share some insights from Denmark. They are extremely strict. I think it's one of the most strict countries to get any antibiotics at all. I think you really need to be in desperate need of antibiotics so that you can get such a prescription. I haven't had a personal experience, so I cannot tell if there are a specific number of pills, but I'm pretty confident that there should be, because I think it's shared by the Scandinavian countries in general.
Yeah, now we're missing Portugal.
Yeah, I can share my experience in Portugal. We usually, for the antibiotics that are classified as antibiotics, and I'll explain what I mean, you get exactly the number of pills that you need to take. So usually it's 8 days. Only in the case of more resistant infections that you need to take 10 days, then you don't have the right amount of pills. And in that case, you could have leftovers. Most people are already used to go then to the pharmacy and just give away the remaining. We do recycle the medications. And when I say about what is classified as antibiotics, because there is a compound that is not classified as antibiotic, but is used for urinary infections, and that you have a high number of pills that you are not going to take. So you have a lot of them in-house. Yeah.
Well, thank you very much. Yeah, we go.
Okay, thank you very much. We will proceed with the next presenters from University of Lille. Yannick Rosser is the next presenter.
screen one. Share screen one. Okay.
Sorry. So I will try just to highlight some of the outputs from Lille, and to introduce us quickly Lille, where it is exactly, and if you want to visit as well. So that's a good occasion to talk about my city. So there is beers, there is cheese, lot of cheese, many cheese. And Lille is a pretty city as well, so it's not only about science, but the place where the science happens, let's say. Now I will talk about my unit as well, my research. And because Catherine and me talk a lot about sugars, because we are in part of the Unité de glycobiologie structurale et fonctionnelle, so the units specialize in the glycobiology, and we are trying to add that complex degree of complexity in One Health questions, which is quite so far unusual, let's say. So we are 13 teams at the moment, 69 researchers with different non-permanent staffs. And we are working on different models as well, on humans, of course, and different animals too. So why carbohydrates and glycans in general are important is because they envelop all living organisms. So from bacteria, so it's not only on the host, but as well on bacteria, even on virus. On plants, there are a lot of polysaccharides on plants too. And during host, this is what I'm interested more, between host and pathogens interaction, there are sugars in both sides. And if we look at different type of glycosylation as well, I describe, and Catherine too, the glycosylation we call the mucin type, but there are a lot of different type of glycosylation, like protoglycans. Glycoproteins are not only O-glycosylated, but as well N-glycosylated. And it's something very important because if you have one mutation, you can have some genetical disorders. You can find as well some glycolipids, and some of them are very complex in different type of tissues. In animals, you can find different type of glycolipids too. And some glycolipids, for example, are involved in host pathogen interactions. So, and if we look at the surface of cells, you have what we call glycocalyx. That's a quite old word. You probably heard that when you were studying. Not for me so accurate, but still used. But that's summing pretty much all glycans and glycostructures at the top, at the surface of cells. So, of course, you can imagine the type of glycostructures interacting with the different type of pathogens, bacterial or virus, etc. So about the venue, we did that in Université of Lille, and more precisely what a school called Polytech Lille. It's not exactly in Lille, but very, very close, called Villeneuve d'Ascq, but it's easier to say Lille, I think. And it was in that big room here, you can recognize Jan talking about viruses and about the programs we just try to follow. It was something quite broad, involving different models of zoonoses and infection of different type of viruses. We started the section with a good example of zoonoses with Jan. I forgot the R again, sorry. Between oysters and noroviruses, and how glycans can mediate the interaction between oysters and the virus themselves. And we had two speakers from Thailand, so we did that remotely from, not remotely, from Thailand by Zoom, sorry. They discuss about something quite important for Thailand, the prawns, because there is a huge epidemic with involving different type of viruses who are killing all the prawns. At the beginning, the prawns were the first product of exportation from Thailand, but not anymore because they are all dying, the prawns. So they are trying to fight and to discover new mechanism therapy to kill the viruses or to avoid them to proliferate rather. And we are collaborating with them to, of course, regarding the glycans and how the glycans from the shrimp are recognized by the different type of viruses. Catherine talked about pretty much of what she presented earlier today. We had one speaker to talk about the flea, so a perfect zoonotic case because it involved rats, as you know that, and through the flea and how the bacteria Yersinia are infecting humans. It's still occurring now, not in Europe or very few cases, but most likely in Madagascar. There are still a lot of cases happening. We had a speaker more relating to what's been discussed by Huli, for example, on the environment reservoir of antibiotic-resistant bacteria. Then I talk about my chicken story. And we tried for the last three speakers to highlight some new therapies. And then the main output was to discuss about what we can do next about the antibiotic resistances. So Ruben talked about new strategy to develop new antibiotics and in combination with all treatments to increase the efficiency, of the antibiotics. Laurie talked about new vaccines. So then to fight antibiotics, one of the alternatives is to find new vaccines when possible. And Marie Titekat, so she is a clinician, and she presents many things about bacteriophages, who rely to some of one of the speeches this morning. It was, in that case, directly on humans, but I think there are a lot of similarities of what you talked earlier. So many pictures to show, and that's it for me, just to sum up some of them. So now we are still discussing with Jan, Catherine, and some other speakers to try to write a paper with some recommendation, case of study through the zoonoses, and most likely new strategy to fight antibiotic resistance. Yeah, if you have any questions, you don't have to.
No question. Thank you very much for the presentation. Too many researches, too much effort. For an external observer like me, there is a question arose during this session. The AMR problem is well established, at least the last 15 years. Even in this area, in this room, some years ago, this problem was discussed. Does any of the representatives give me an answer about if there is a trend of declining the problem or increasing the problem, or is it in a steady state? That's the question for the public.
If there is a trend of decreasing the antibiotic resistance, as far as I know, it depends on the pathogens because and as well in the timeline we are talking about. If it's about 20 years ago, for example, one bacteria I'm used to it is Acinetobacter baumannii, was not known at all, and it's not a real pathogenic ones because there are no weapons, let's say, like Pseudomonas aeruginosa, for example. But now it's becoming a huge threat because of the antibiotic resistances. They are very good at gathering new genes to fight antibiotics. So that's something still who needs a lot of effort. And one statistics, some people are not probably okay with that data, but it's still a consensus, I think, is that in 2050, we will need more treatment to fight bacteria than cancers, and there are at the moment more products developed to fight cancers by company because it makes much more money than developing new antibiotics. So yeah, that's something. The trend is still getting higher in global, I would say. Unless if someone wants to say something else, I'm not with on the authority with only for that big question, I would say.
Yes, thank you. Is there any other opinion about that? Anybody wants to say something about my question?
Yeah, I mainly agree with Yannick that it's very dependent on how you want to look at it. If we look at total, global, all bugs, yes, it's still increasing. If we look at certain countries that have put in a lot of effort already in minimizing it, at least if we look clinically and veterinarily of the bugs that matter the most for health outcome or economic outcome, we see declining trends. For example, Norway is a great example, but also some other countries, it's going down quite a bit. And you can see positives of the efforts. But you have to very dedicatedly discern the data of what is currently the target and is there new emerging pathogens that suddenly start popping up and so on. So it's not as easy to answer based on where you are, what actually is the outcome you want to target, because there's probably no one-size-fits-all solution to the problem.
Thank you. Thank you very much for your. Thanks all. We will continue at 5:30 for the next section. If there is any comment or any question, okay. So we'll continue in a few minutes. I'd like to thank you for your presence here to our roundtable about migration and the One Health. It's a very hot topic. Our first presenter is Dr. Apostolos Veizis. He's Executive Director of IDESOS Hellas. IDESOS Hellas is a humanitarian organization with very active in Greece about the refugees, about the moving populations. Dr. Veizis, could you please make an introductory statement? Thank you.
Thank you, Professor, for the introduction, first of all. And I would like to thank everybody today that I was very happy to hear about all your workshop and research and the outcomes of the One Health, especially trying to connect many elements together, from zoonosis to animals to humans, etc., etc. I think on the other side, if we want to talk about One Health, it's an important part, one little element, one human. And I think the important part is humanity, because what we don't see currently internationally taking place, it's talking about one humanity. And unfortunately, we had to talk about people on the move, being migrant refugees and asylum seekers. I liked also the second part, and I would like to thank Professor Vandarakis for One Bridge. So I think it's good to have bridges, because bridges connect. For example, Patras has a very nice bridge, and bridges connect people. While walls and fences are trying to keep people aside, but you cannot keep people aside, neither you can keep diseases with fences and walls. You need to do management of that. And I'm saying that because today, living in a European context, and I think being a European project, I will add only 2 or 3 small elements. Today we talk about mass migration. I have just to share with you that currently at the moment, from 1st of January 2025 until now, 143,000 people tried irregularly to cross Europe. So if 143,000 humans, children, women, and men, it's a mass migration for a continent of 700 million people, that's one. Secondly, while I'm talking to you at the moment, just to mention to you that we had 2 shipwrecks taking place in the Aegean Sea. 17 people, and most of them minors, dead in Crete, and today 1 12-year boy. And this, of course, is adding a list of about 80,000 humans who lost their life on the attempt to try to move in order to look for protection and in order to look for even for a better life. What we achieved as international community, we achieved to increase the number of people on the move. So every year, we talk about millions and millions and millions. So we are very successful in that. But then when it comes to the management, another element that was just voted, the ministers of Home Affairs agreed to detain migrants 2.5 years, from 18 months to 2.5 years in detention facilities. And of course, thanks to Trumpism is coming with big waves in European Union. They decided even to raid houses where migrants and refugees are living. Now, because I would like to leave the floor to very important people who are really, first of all, my mentors. Secondly, people that are on the front line and people that probably can give a bit of a hope on this dark environment. But I think we need to remind ourselves something that Virchow, because some of you did mention about Virchow before, about ill health. Ill health comes because of social inequalities. And if we want to see what are these social inequalities, we can see the social inequalities and we will talk about them. So before that, I would like to, I have the pleasure, first of all, to have here with us Professor Bernadette Kumar. If I read her CV and her work, then we will not have the time to do the roundtable. But I have just to mention that she came from Norway, and I think it's an honor, first of all, having her here. And as a president of a global society of many things, of migration, of health, of ethnicity, etc., a global society which is trying to include many elements together. She can bring an experience on how interconnecting partnership, how connection between countries and bring challenges and experience. So Bernadette, the floor is yours, and thank you very much for that.
Thank you very much, Apple. First of all, I'd like to thank the organizers and, of course, also my good friend, Dr. Professor Vantakris, who I've worked with before. It's a real pleasure to be in Greece and a pleasure to be with all of you and all those who are also attending online. So basically, what I'm going to do is, I'm not a historian. And I know that Apo has given you some statistics already, and I won't bore you. So I'm going to try and keep you engaged with the story, because of course, these are all stories about people and about our lives. And I'm going to start with the past. And very often, we tend immediately to look at everything that's going wrong. So I'm going to talk a little bit about the past, about the present, and maybe future, and then the other panelists, I'm sure, will draw into the discussion that we're going to have. So if we look at the past, and if we look at human existence and human health, because we're all concerned about health, and the previous session also talked about that, we're actually living longer. We have decreased mortality. We have made tremendous success when it comes to technology, management of diseases, etc., etc. And I think we shouldn't forget the past gains that we've had. And sometimes we only tend to focus on the negative. We also know that in the past, and it's a glorious past, that many of the great civilizations, many settlements, all came out of migration. People moved, and they moved together, and they lived that. And we've often forgotten that migration was something that was positive. It was bringing people together, and they were flourishing societies. And even if we look at the recent past, and we look at across the Atlantic, we look at Europe, we look at Australia, many countries, they've come of migration. But suddenly, migration has got only a very negative stamp to it. And that is very, very unfortunate in the present situation. One of the reasons that we have an issue with that, and if we look in the past again, we see that there was a lot of conflict. It's not as if war is unknown to human beings. War has been with us as long as we've existed. And yet we see today, the present, and in the past, we learned from the lessons of war, and we learned about peace and reconciliation, and we still learn about peace and reconciliation. And today we have over 61 armed conflicts across the world. And this means it is one of the largest determinants of forced displacement. People have to move. We also know this has meant, in today's present, that lots of countries are spending a lot of their funding on military expenditure. It went down 50 years ago. Now it's up to 2.3%. And we don't have unlimited resources, so which means that resources for health are limited, and they have becoming even more limited. And of course, we know that this also involves research. So the current situation is a world where it's precarious, it's uncertain. And in this uncertain, precarious world, we really have no other option but to work together, because we're human. And human beings are connected, and our interconnectedness was very clear during the COVID pandemic. So if you thought being not connected was an option, it's not an option at all. It's no longer optional. So that's my first point, and really looking at the past and looking at the present today and looking at the future, we have to do that. It's important to connect, and we can connect in many different ways. And one of the things that we really have to look at is how we connect. And we can connect and how we connect and why we need to connect. So let's look at first why we need to connect. When we look at, and just talking about all the bugs are the same, right? We can say, regardless of where you are, bugs don't know borders. But it is a reality that many of the challenges that we face today, they're common. And the form might be different. The approaches might be different. The extent might be different. But the challenges are very similar. And because these challenges are very similar across the globe when it comes to health, we do have a common platform. And that's why we agree on this common platform, which is looking at the Sustainable Development Goals. And we all agree, we set these goals for us. So we have some common challenges, we've agreed on common platforms. And how are we going to do that if we don't have partnerships, if we don't work across countries? So that's my second argument, to say that we need to work on these common challenges with using the common platform. However, there is a disconnect, and this is my favorite, if you read The Tale of Two Cities, it's all about working in silos, which is why I was going to, Apple already said it, but the bridges are important, because we can't really continue to work in the silos of academia, policymakers, civil society, the private sector, industry. All of this is all interconnected. And we have to make sure that it's not operating in these silos. And it's very easy to say, okay, we have to, tear down the silos. Nobody wants to tear their silo down. So the only way we can do that is by bridges, connect them. We also have silos within the disciplines, and all of us know that. We have the natural sciences, we have the social sciences, we have clinical science. And again, here we don't speak the same language. So very often we need to be connected, and for that we need the interdisciplinarity. So we need the sectors to connect and for that we need the partnerships between the sectors. We need the disciplines to connect and have some kind of common knowledge base that they can. So this disconnect has to be dealt with. And the disconnect is the only way to deal with the complexity, because these are very complex issues. It's no longer A plus B equals to C, and my apologies for oversimplifying to all the virologists and microbiologists in the room. It was easy with the tuberculosis, where you said, okay, this is the bacilli, we know. Now there's a web of causation. And we see in the case of migration health and the health of migrants, it's many things that are acting together. Why they moved could be a number of reasons. Climate change, conflict, labor, all of these things are coming together. So it's very complex. And in order to deal with these complex or wicked problems, we need to partner. We need to work across. And we need to learn from one another. And for that, we need to be able to connect the dots. And how do we do that? So we need to generate knowledge. And many of us sitting in the room here know that generating knowledge is important. And for research, sometimes it's not possible to conduct research, or sometimes it's possible, but it might be a wastage or duplication if you do it only in one particular country, which is not to say that we don't need research at the national level. We do. But we also need to share that both in the regional level as well as at the global level. And I'm sure, if Santino joins us, then he will be talking a little bit about the global health research agenda. So I will not talk about that, but that's a kind of partnership. We also need to share experiences, because research is not only knowledge. Knowledge is also lived experience. And the lived experience is often not seen as knowledge, which I think is very unfortunate, because that comes from civil society, it comes from the people themselves. And we always say, nothing about me without me. And if we don't have that, then we don't have a really good understanding. So we need that, and we need to share these lessons learned across. And I will, end with a few examples. And for example, the research collaboration that we have is of the Lancet migration, where we have collaboration across, which is transforming. The main purpose is to transform evidence into action through this global collaboration. And we also have a collaboration at the European level. So you have the Lancet Migration European Hub, you have the Latin American Hub, and soon, hopefully, the North American Hub also in the offing. So you have those. You have the big, large EU projects. You have UFO, which is the European Association of Public Health. So there's global collaboration. There's the Global Health Research Agenda by WHO. And then you have joint actions. And I would like to share the experience of a joint action, JAHI, which was Joint Action for Inequity, where we had 27 countries working together across with partnerships, and actually Greece was one of them. And one of the things we learned was that it was not only important to look at what each country was doing, but also to see that what could we learn better from each other. And in the course it's not about copy paste. You can't take one thing, even within a country, the regions are different. That was one of the learning. And it's different at the local level. But what we can do is that we can actually, through the experience and the lessons learned, try to develop and adapt for the future. And I think that's a very important thing for collaboration. And JAHI was a very good example of that. And it was really through a process of 3 years working together across these countries that really helped us to come up with some common lessons learned. And I'm just going to share those lessons learned from JAHI with you because they were very important. The first thing was user involvement. Without having people involved in the process, and this is where the lived experiences, the narratives are very important, it's very hard to know what will work because it becomes very theoretical. The second thing, which I think is an upstream factor which often gets ignored because everyone is concerned with their own arena work, is the whole issue of leadership, governance, and dialogues. We need the champions. We need people to front the issues. We need to have the dialogue, and this is an example of dialogue at different levels with different groups of people across the sectors and discuss some of these challenges and issues. And also we need to keep making these adjustments. It's not like, okay, now we've done the project, we have a very nice project summary, the EU is very satisfied, and we can all go home. Doesn't work that way, because we need to have a continuation of that dialogue to meet and say, okay, where are we now? Did that really work? We thought it would work. And so we need to keep the momentum. And that is a very important thing. And I think I will stop here, otherwise I can continue talking. So we can come back to the discussion. Thank you so much.
Thank you, Dr. Bernadette. Thank you for your presentation. The next speaker is Dr. Santino Severoni. He's a director of the Health and Migration Programs at WHO headquarters. And the topic is building migrant and refugee inclusive health systems in a changing climate. He is an online speech, so we can connect.
For having me today. I'm really sorry for not being able to be in person in Athens. I would love it. I love your country. I love Athens. But tomorrow we have the global school starting, so I had to stay here in Geneva. Looking forward for the next meeting in Athens to join in person, Apostolos. I put together a few slides, and this, although we didn't coordinate with Bernadette, I could consider the second chapter of the introduction she made. When we go to the public health issues related to migration, it's a complex business because we need to navigate political sensitiveness, we need to navigate disinformation, we need to navigate narrative or anecdotal stories. So within the health sector, within WHO, we started to work on this area not long ago, and here at Porter only in 2020. And our objective has been so far try to depoliticize the narrative by investing into producing stronger evidence and facts in order to offer Ministry of Health and health sector in general solid argument to manage the situation. So I prepared few slides. It's not a presentation, but just few flash to give you an idea of what are the complexity and challenges when we look at the intersection between health and migration. If then we extend this intersection also to One Health, another sector, you can imagine those are additional multipliers that we need to take into consideration. In my presentation, in my few slides, in my speech, I will focus on the intersection between migration and the implication for the health of these people and the public health. Please just tell me if you can see my presentation.
Yes, Santino. You can make it full screen if you want.
I just requested. Thank you. Can you?
Affirmative.
Hold on, I need to probably change the setting because I see too many slides there. So when we look at we start to look at the health sector in general from a global perspective, and then we start to conduct carotage to see how the health system behave in relation to this topic, we realize that three big areas that were the challenges or the issues relevant for the health, for the public health. First of all, the health outcome for this population because of the migratory process and because of the risk factor in many cases impacting on the health of these people. In fact, we like to say that to be a migrant might be play like a determinant of health in many cases, but not always. Indeed, the lifestyle, the life course, the exposure to risk factors, which is something which is affecting anybody, regardless of the migratory status, even resident population, is a very important issue. The second element is the limited or problematic of challenging access to health system, either because of barriers, mostly related to the rights and entitlements that country might be selectively utilizing to control the spending on health, and also before there was reference to the economic dimension, the economic narrative attached to provision of services, but also the challenges related to navigating an health system while you are not from the country where you're going to utilize the health system. We need to remind to ourselves that the concept of health system is relatively old concept in public health, and usually we define health system or an efficient health system, the health system which is able to provide answers to the need of the population they're supposed to serve. But if, as an approach, we use to standardize the population only with us, without taking into consideration a demographic change, then we start to have some dysfunctioning. Then the availability of evidence. Again, we invested with a research agenda because this is really a huge gap in terms of understanding how things are functioning, and data availability. When we look at the migration, often, and I refer in these slides to the allusion to the economic dimension, is a reality. Every time we are talking about health sector reform, or we are talking about the national budget, the budget for the health sector is always a very hot issue. And we cannot expect, we cannot be so idealist, so naive, thinking that bringing in the discussion of expanding the access to services doesn't have an health implication. But it's also important that we are reading the numbers in a more correct way. The narrative is that we are confronting with a mass movement of population. Yeah, there was a growing of the international migrants, as you can see from those data. But in reality, globally, we are talking about 0.4 increase, so relatively small increase, a sort of steady phenomenon of migration. What politics, what policymakers, they need to focus that the demographic variation, demographic change we are meeting because of the effect of globalization or other factors, socioeconomic, or even instability in many cases, they are determining a variation of demography. And we need to have an health system which is able to be flexible, resilient, to adjust to this new context. A big issue is represented by the labor migration. Those are ILO data, and in my view are extremely interesting because we are talking about people which are, in most of the case, regularly present in a country. They are working, they're sending kids to school, they are paying taxes, and their taxes, they are also financing the health sector. So there are people contributing to the economy and to the budget of the country for the provision of services. What is interesting that the official data that ILO shared are indicating a bit more than the half of the overall quota of international migrants. So what is the message here? What is the interpretation? That we are having a problem of registration, that the data we are referring to, they are not accurate, not because of mistake, but because of the definitions and legal framework we are utilizing to define the group of population, so that in many cases, the quota of labor migrants means people regularly present into the country, contributing to the health system or health insurance system, are the majority of people on the move. So basically, we need to also fuse the idea that this is only a spending for them, but there is also a contribution. And when we look at the health sector should not be a business where we are investing to have an income, but should be a control-loose business where we are investing, the product is not money, but the health of the population. And again, the situation is different when we are looking at the displaced quota of this population, because in this case, we are having people moving en masse because of very tremendous or powerful push or pull factors, usually conflict, climate changes, a number of situations that can force people to move. Once more, those are people in search of life saving, so it's important for us to understand where the movement are going, and we're going to discover that the countries more in need of financial cooperation, support, and share of responsibility are countries with a low or middle income capacity, not the high income countries. But again, we are talking about a dual type of issue. Life-saving interventions, mostly implemented through emergency or humanitarian response that can be also quite long in terms of time. Or we are talking about, again, the first issue, access to health system, because also displaced population might become refugees or staying in conditions of chronic displacement for decades. We are having cases of 50 years living as a refugees or even as a apoli. And this posing the most important issues of access to health system. In this group of population, we see also a very interesting phenomenon where the humanitarian assistance should be devoted in supporting this population also with an eye of equity, because in many cases, this population tend to be received in communities in low income set. And it's important that we are not generating also for the benefit of the operation of supporting the most in need, that we keep in mind the issue of equity with the resident population where those group of people are often hosted. The real problem that politicians and policymakers, they cannot manage for a number of reasons, particularly because there is a very blaring picture, is the irregular migration. The irregular migration because it's out of control, because it's coming totally interesting country in totally unexpected way, even if there are progress and initiative to get prepared. But mostly this is the problem because it's the just political justification for government to say we don't want migration. But what is it? In reality, it's the smaller quota of people, and a big mistake would be to penalize or to have scapegoats the migrants But here we are talking about trafficking. We are talking about inter-country smuggling of human beings with all the consequences and repercussions. And here we are talking about probably worldwide of 30, 45 million. But again, those are data very, how to say, very approximative, very proxy, because we don't have enough information to size this population. And we don't have also sufficient information to define trends and to really assist countries to have a proper planning and to manage eventual public health implications. The challenge of the millennium, and we are still debating and there is no clarity, while we know very well how climate changes are changing the pattern of diseases, particularly vector-borne diseases. This is very important for the One Health issues. And we know very well how heat waves or catastrophe generated by climate changes can impact human beings. We know very little about the intersection between health, migration, and climate changes. And again, from our study, from our work, the main challenge is represented by inter-country cooperation, collaboration, which is not partnership, but it's really sharing resources across border, really looking at innovative modality of shaping our health systems in order to fill gaps, in order to serve populations that are forced to move. An interesting element in the discussion related to climate change is that when we are talking about migration in the context of climate change, migration is perceived as a positive thing. And that's something probably where we need to work on. It's a window of opportunity because migration is recognized by the climate change community as an adaptative phenomenon. And in fact, you might be coming across media news that Australia is already granting full access and nationality and hospitality to entire population from small island in the Pacific. We try to monitor and look at the changes because changes are taking place. I don't want to project the idea that is a catastrophe. It's always the usual negative narrative about the topic. In reality, I can really tell you precisely also within the time frame when health sectors start to understand that there's a role. And we observe a boost in terms of this understanding during COVID, where the intersectorial collaboration became clear as an essential element for success. And we are monitoring the situation. We are preparing the second world report also to see how we move from 5 years ago when we produced the first report, which show us the element which I mentioned before, the poor health outcomes, the data, lack of evidence, and lack of access to health system. And we asked two countries to provide us information on very specific questions in order to start to detect trends and changes. And it's very interesting to see that we send a request of data information to all our member states under 94, and 93 countries responded. Might be look a small amount, but it's not. Usually those kind of operations, they receive very little sympathy from member states in sharing data. In this case, we found a quite surprising amount of countries providing data that show progresses. So what we see in terms of concrete situation, what are the findings? And I'm just giving you a very quick snapshot because I don't want to preempt the content of the report that will be released in February. Well, there are changes taking place. This is a reality. The world of today, despite the political narrative, despite what we see on media, despite the toxic narrative around migration, is changing. And the health sector is getting more and more ownership and understanding and leadership in addressing the public health implication of population movement. In many of the countries which provide data to us, the inclusion of refugees and migrants into national policies is nowadays a reality. And this might look simple, but it's not, because up to yesterday, if I can say like that, the reference to migrant refugees was as a vulnerable population, which is the worst way to do our job, because first of all, not always are vulnerable, and second, because addressing them as a vulnerable in general, you not address anybody. So considering the specificity of this group, considering the legal framework, considering the peculiarity of the process of accessing the health sector, it's very important that we are very precise with the definitions. And then the intersectorial collaboration, it's really growing. So sectors understand that sector goes out and meet with the Ministry of Interior, Ministry of Labor in order to come up with a rational approach at the national level. Remain extremely concerning the gap of inclusion of refugees and migrants into emergency policies. Still, this is considered, is forgotten. We have seen even during COVID with the access to vaccines, with a COVAX instrument. Countries, they don't tend to assess the health system capacity, so they don't do a verification. Is the service I'm having, what I set up in order to serve my population, are really functioning, are really granting access? Again, data and informed, capable healthcare workforce. And going to conclude by showing you, this is a living dashboard, because we are tracking practices at country level. You can see also on a map which countries are really engaging, what they are doing. So we thought important to facilitate sharing of practices among countries. And it's interesting to see that we are receiving spontaneous submission of good practices, or countries willing to share what they are doing in terms of including migrants and refugees into the health sector. Incredible is to see that countries, regardless of their own income capacity, economic capacity, they decided politically to expand the health insurance coverage to millions of migrants or refugees. The case of Uganda, the case of Colombia, South Africa is also moving in this direction. So it's extremely interesting realities. We are operating in a volatile world. We are operating probably in a world that tomorrow will be totally different from the world where we started to work. I'm convinced that what we are going through is not a shock, is not an emergency, it's not a transition. I'm convinced we are not going to go back to previous reality. So we need to be super smart, super prepared to imagine how we want to have our future, particularly in relation to the health sector, in order to have health sectors and health system able to address the needs of our population. And I just put here a few data to give you an idea of the changes. UNHCR is cutting 40% of the budget, and the health sector is one of the areas more affected. UNHCR used to cash support access to services in many countries. IOM cutting 30% of the budget. If this is going to affect the personal health screening of millions of migrants moving for work across the country, means that public health becoming myope, losing sight in terms of detecting TB, HIV, or other health conditions, diabetes or chronic conditions. WHO is cutting 30%. I'm here today after one, probably the most difficult year in my entire career of WHO, where everything was under discussion, but we managed, we succeed to preserve this area as a priority. The Director General decided this approach, and we are still here, and we consider extremely important for the interest of the organization. But certainly, as I'm speaking, also working internally in the office to reimagine how we want to operate, what we can do, how we can better serve our member states in a changing world. And I want to conclude with a provocation. We tend to look at the health sector a little bit in a static form. This is the way how we provide service, this is the way our health system is structured. In reality, we are in a moment of transition, change worldwide, that we need to start to look into innovation. We need to be brave to look at better functioning solutions for the health sector. And I like very much the work that is going on in collaboration with the London School and the researcher. The idea of a cosmopolitan health system means really switching from an idea that health sector, health system need to be focused within the border to his own country population only, but the idea that health sector need to be also able to cooperate across border. We have seen with COVID how important is cooperation, sharing of information, sharing of resources. The same is true for all challenges, including demographic change challenges related to migration, because we have seen that country by country, single country solution is not functioning, it's not efficient. Also, in terms of support that today country can count on is very minimal. $23 per person per year is what the international community is able to allocate. So why not to look across border, to have a health system which are more prone to inter-country cooperation by sharing resources, not only financial, but human resources, services, particularly in those areas where migration is a consolidated phenomenon. Imagine the issue of climate change. We are having area in Africa where the migration in certain area, it's a seasonal and recurrent phenomenon since long time. Integrating health networks across the board. So really we need to pool our resources, we need to align across the border in order to boost cooperation across border between health sector and networks. Imagine services like tertiary care services, which are too expensive for a small country or country with low incomes, while it's affordable for a neighboring country with better economic conditions. And make sure that policies, they are reflecting the real population which we want to target with that policy. We are still not good. We are still referring to people that we want to serve in a very generic and specific manner. So it's very important that we are referring specifically, and we are including this in all national policies or international policies. And then health is a choice. The health system that country want to give to their own population is a choice, it's a political choice. So we need to be ready to make an investment. And this is not an investment, as I proved you before with the labor migrant quote, against people which will not return anything. It's an investment against the population living in our country, which might have a demographic adjustment because of the time we are living. Thank you.
Thank you very much, Dr. Severoni. Is there any question? Okay. Thank you very much. You wanna...
No, I think as we'll complete the panel, if there are questions we have a bit of time for discussion, so question will come up, Santino, so we'll better continue.
Okay. Okay. Okay, so next speaker, I think is in person, Dr. Kevin Bozoberg, preventing and managing non-communicable diseases across the life course in migrant and refugees communities. So Dr. Kevin Bozoberg is a director of the Department of Population Medicine and Health Services Research at Bedford University and head of the section of Health Equity Studies and Migration at Heidelberg University Hospital. So is online also, eh? Okay. So we can connect.
Thank you, and good afternoon, good evening. Pleasure to be here. Thank you to the colleagues of University of Patras for inviting me to join this panel with the precious colleagues Bernadette, Santino, and Timur. And thank you to the preceding speakers for laying the ground for my contribution, which will focus now more on non-communicable diseases, and especially in the context of One Health, which is a very important topic. There is a strong tendency to look at infectious diseases, emerging and re-emerging diseases, infectious diseases, and also on antimicrobial resistance, which is all very important and big challenges for public health in societies. But it is equally important to also have in mind the non-communicable disease perspective. And I brought a presentation that I will now share with you. I hope you can see my screen.
We can see it very well, Kevin.
Excellent, thank you, Apo. And my focus will be along the lines that Santino nicely has put, more on the health system and policy perspectives, and trying to bring to light the relevance on preventing and managing NCD. So as we just heard, we have complex migration journeys, but they face actually stationary health systems that are set and built up across over decades and centuries to serve stationary populations. So what you see here are the complex migration trajectories of irregular migration to the right, and on the left, migration flows of regular migration, international migration, that show us patterns. And what we can see for regular migration is that the majority of migration actually takes place within the continents. And especially in Europe, the dominant narrative is very much looking at the smaller proportion of irregular migration. And in this situation, it is really key to consider, if we talk about managing and preventing NCDs among migrants and refugees, that the majority of migration, also in Europe, but across the world, happens within the region. So more than 70% of international migrants in Europe are from Europe, from European Union countries, or from the continent. And the remainder is actually coming from other continents, intercontinental migration, and especially driven by the majority of conflicts that we heard in Bernadette's presentation, pushing and fueling forced migration across the globe. So the risk factors for non-communicable diseases are actually the same that are for resident populations. We have health behavior, we have lifestyle and diet, we have social determinants that shape the risk of acquiring or developing non-communicable diseases in the country of origin. And these are also shaped by the strengths or weaknesses of the health system in diagnosing and preventing non-communicable diseases by healthcare interventions. But what is specific now, when people are on the move, there are migrant-specific risks that are exacerbated or are created by the migration context, by lack or poor access to preventive health services, by migrant-specific risks and exposures that may be depending on the labor environment, on the workforce environment, on the housing environment. And these actually interact with the risk factors that we know from resident and non-migrant populations, and especially the situations in transit and in the resettlement countries affect the migrant-specific risk to develop non-communicable diseases. So if we talk about the burden of NCDs, I deliberately did not bring figures and numbers to bore you with that, but also because the evidence is quite patchy. So there are no global estimates that try to validly estimate the burden of non-communicable diseases among migrants, refugees, and displaced persons, because these actually depend on the country composition of migrants. So we always need to keep in mind that these are socially constructed categories, right? And that there is potentially not even a globally valid estimate out there, because the composition is certainly dynamic, it is amenable to change, of temporal and spatial variation depending on where the migrants come from, in which context they live, and especially what risk factors they bring with them. So you can take the burden in country of origin as an orientation, but we all know that migrants are actually a selective population, and these selection effects that work into multiple directions. So if we have countries that are sending countries for high skilled migration, we may have better educated, better situated migrant or societal population groups migrating. We may also have the difference or more low skilled labor migration, which may come along with higher risks for NCDs. So this may lead to over and underestimation. And as I said, the interaction with a specific context can increase or reduce the specific risk. So it's really hard to come along with, a global estimate on different NCDs. However, those regional and rather national estimates or studies that exist underline that the non-communicable disease burden is non-negligible, and it is key to consider them, especially related to cardiovascular diseases, to diabetes, cancer, respiratory diseases, and also often overlooked, especially among children, actually, in neurological conditions. So preventing and managing non-communicable diseases is key, taken now here from a global evidence on review performed by the World Health Organization, where the key message is that all the measures we have to prevent and manage NCDs in resident populations are equally important across the migration cycle to ensure a continuity of care in these populations. And let me dive a bit deeper into the notion of continuity of care, well knowing that it is very oftenly invoked in fora, in policy fora, but also in scientific fora. So what does it actually mean? We have three dimensions of continuity of care that are key. The first is relational continuity, which means having and developing a trusting and longitudinal relationship with a healthcare professional. The second one is informational continuity, which means that healthcare professionals have and require all information on their patient and the medical history on hand when needed. The third element is management continuity, which means that if care is provided by multiple healthcare professionals, their approach is consistent with that of others and is congruent with the patient's needs. So these are the three dimensions of continuity that are key. And to be very frank, this achieving continuity among NCD care is even a challenge in national health systems for non-migrant and resident populations, but especially in mobile populations, we have breaks in continuity related to displacement, relocation, transfers, and the geographic distances. We have information losses, which then lead to repeat assessments across borders, but also within countries when people move, and to multiple forms of redundance in diagnostics or treatment. And in terms of management continuity, we have varying treatment regimes. You all know, all practitioners know how challenging it is even to implement evidence-informed guidelines into daily routines in the health systems. But these are exacerbated also across countries when we have multiple forms of drug availability and multiple different varying ways of treating or diagnosing specific conditions. So what can we do? There are multiple entry points to enhance continuity of care, ranging from the individual level to the system level. And I would say there are multiple interventions out there that try to address also the individual level. These are certainly very important. But for us as health professionals, I think the biggest value and the biggest leverages are actually to focus on the provider side and on the healthcare organizational part, because this is where we have the biggest reach and the biggest influence that can be implemented by health professionals. And to give you an overview of evidence-informed solutions or models of service delivery that enhance continuity of care, these are care management models, including network-based deployment of care management models that try to serve regionally and spatially defined populations, integrated care models that try to build up one-stop shops, trying to reduce the geographic but also the disciplinary divides in making diagnostics and treatments on these complex conditions. Community-based outreach models, including mobile clinics or deployment of allied health professionals, non-medical health professionals, non-physicians like refugee health nursing. These are models very well evaluated, especially in Australia and in Anglo-Saxon world, or midwifery-led models that try to deploy preventive services for maternal and child health. So these are very well evaluated, including also step care models that are mostly evaluated from mental health, but from the logic very well also suited to address the specific needs of migrant and refugee populations. And the evidence of the effectiveness of these models mostly comes from resident populations, but also from resident populations that are considered to be vulnerable, like living in disadvantaged areas or having other forms of disadvantage. But there is also evaluations among displaced populations and in complex emergencies. So the cross-cutting elements for these are listed here. Like in all these models, we need interpreting and cultural mediation services. We should ideally involve migrants and displaced health workers and set up, as for resident populations, these teams to consist of multi-professional teams and social workers. Now, a frequently asked question is, should we set up vertical and special programs for migrant or refugees, or should we try to integrate them in existing services? My answer to this question is usually this is a false dichotomy. It's not an either nor, and we need to move diagonal. What does this mean? It means that we need special services for special needs, but at the end, these special services, even if they are set up in a parallel stream of the health system, require some form of integration over time and linkage to regular parts of the health system. On the other hand, we need horizontal elements, which means that for equal needs, we need to integrate people within the existing structures without reinventing the wheels. But if we do this, we need to ensure that these are tailored. This means that the equal needs take care of the special requirements, for example, by providing language mediation. So both streams need to converge. It is not an either or. Now, some cross-cutting challenges and key considerations for decision makers in these areas, and these are specific challenges, are to train the personnel to work in these models. As also pointed out, we may need specific financing and reimbursement models to make these systems work for populations on the move. The referral and re-referral approaches are very challenging and require specific mandates and responsibilities set to either organizations or individuals to take care and reduce fragmentation and reduce breaks in the continuity of care, especially related to a One Health approach. I would say intersectoral cooperation. If you think about industry, labor migration, different workforce settings, agricultural settings, it is key to cooperate with these actors, but we don't really see that this is happening. So this is really a challenge. And especially if we talk about cross-border care, we may have some regulatory frameworks that allow this, but we see that this is not happening in reality and in practice to an extent that would really develop a population-level impact. So in conclusion, NCD prevention and management is key for migrant and refugee health, and the burden varies by composition of migrant populations and contexts. And in this situation, it is key to understand what is the burden on the ground in the specific context, and to ensure that continuity is realized with multiple levels of intervention that ideally also include the workforce and organizational level to not only intervene at the individual level, which may not develop the same sustained solutions and may not lead to what we actually need, namely that health systems adapt to the mobility of the world and to increasing mobile populations and their health needs. And with this, I would like to stop and look forward for the discussion and to your questions.
Thank you, Keivan, very much for for the analysis on the non-communicable diseases, prevention, management, and continuity of care. I think in order to get the whole picture, I would propose that we get the last but not the least, the expert of ECDC, Taimur Nouri, talking about HIV and migrants in Europe. So after Taimur, then we can open the discussion with all of us for questions and suggestions. Taimur, the floor is yours.
Thank you so much, Apple. Let me just share my screen. Do you see my screen?
Yes, we do.
And do you see this mouse?
Yes, it's okay now.
Good, perfect. Okay, so thank you so much, Apple. So you've heard 3 excellent speakers looking at the big picture, quite visionary talks. Mine is going to be much more focused on a specific disease, HIV among migrant populations. It's very data-driven, so quite complementary to what you've already heard. So at ECDC, we've worked quite a lot on migrant health over the years, developing screening guidelines, etc. But for this talk specifically, I'm zooming in on HIV specifically, which is my area of competence, and we have some important work in terms of migrant health going forward. But let me just start. So some of the data I'm presenting is not published yet, so please be careful with posting anything on social media. So I have no conflicts of interest to declare. Just trying to get rid of something on my screen. Okay, it doesn't disappear. Okay, so this is the content of my talk. So I'll start with giving you an overview of the HIV epidemiology in Europe. Quite detailed data, which is very important actually, because most of the diseases we collect, we can't do this detailed analysis that we can do with HIV because countries don't have this disaggregated data. But so we're quite fortunate when it comes to HIV and specifically TB as well. Those 2 disease areas, we have quite good data, but not so much in other areas. I'll show you some of the inequities in the provision of prevention services, specifically looking at pre-exposure prophylaxis, testing and treatment, and trying just give you an overview of whether or not Europe are going to meet these targets specifically for migrants. So let's start with some epidemiology. So we estimate that roughly 2.1 million people were living with HIV in the wider WHO European region at the end of 2024. 40% of these diagnosed cases occur in the western parts of the region, the countries in green, 3% in the center, and 57% in the east. So the true burden of HIV really is in the eastern parts of the region. It's actually the only region in the world where HIV is actually increasing. The incidence, new infections is only increasing in Europe, and that's very much due to the situation in Eastern Europe. In terms of new diagnosis, that we just published data from 2024, roughly 106,000 people were diagnosed with HIV in 2024, 25% those in the West, 10% in the center, and 66% again in the East. You see the rates per 100,000 populations between the 3 subregions, and really much, much higher in the eastern parts of the region. In terms of known routes of transmission, very important when developing programs, quite similar for West and Center, where roughly 45% of diagnosed cases occur among men who have sex with men, followed by heterosexual contact, both men and women, and only 4% of diagnosed cases among injecting drug users. And in the East, very different. So the main mode of transmission is heterosexual, followed by injecting drug use, and only 4% among men who have sex with men. Now, because of very high levels of stigma in the eastern parts of the region, we believe that this proportion is probably much higher. But because of stigma, you don't come out as being gay, and therefore you are misclassified. But this is official statistics we do get. But what is very important is the issue of migration in our epidemiology, where 6 out of 10 people who are diagnosed with HIV in the western parts of the region are born abroad. So that's our definition of migration. You're born outside the country in which you reported. That drops to 15% in the center and only 2% in the east. So in terms of prevention, testing, treatment programs, we have to address this issue of migration in the West in order to address all these gaps that we're finding, right? So to reach the goals, we need to focus our primary prevention programs on people who are born abroad. So if you look at the EU, EEA countries, this shows you the proportion of HIV diagnosis among migrants. So overall, 56% of all diagnosed cases occur among migrants, as I said. The countries on top here, Iceland, Malta, Norway, etc., 80 to 90% of all their diagnosed cases in 2024 were among migrants. To the EU, where less proportion occur. The
You use this 8 minutes for those great people to ask questions or comments or challenge. And it's an opportunity that we cannot get every day. I think there is 1, 2, 3. From Portugal.
Hi, thank you. Thank you so much for all your presentations. This is indeed an important topic that we should talk about. Regarding the migrants and the refugees, I think we have an issue that goes both ways, maybe, because they are arriving in difficult conditions, but some of them also isolate themselves. So how can we overcome this situation so that we can integrate them into our systems? Of course, they are important, especially considering that our birth rates are really low usually, and so we need the migrants to increase our natality rates. So, because I see that they also segregate themselves sometimes. How can we overcome this?
Bernadette?
Thank you very much. Very important question. I think that it's a very important question because it's not about assimilation. And I think it's about recognizing diversity, which means including people as they are, because often it seems like it's a linear spectrum that everybody has to become the same. And what we need really is now we have some research coming up saying that, and personally, I think the word integration always causes a problem because integration sometimes gets mixed up with assimilation, but it's inclusion. And you have to look at the common arenas for inclusion. And the common arenas for inclusion, particularly in the European setting is, for example, work and education. And I think those are the arenas that, and of course, health is a common arena as well. But when migrants come and when they're trying to understand how society functions, it's where we need that. We need models where one can work so that people don't get isolated. And I think it's in the initial period, because once they get isolated, then it's very difficult to bring them back. So it's a very simplistic, but we have a very big study in Norway looking at that. So I can tell you about that in the break.
If I add just 1 second on that, the question is that when we have the discussion about issues of access to health, and we talk about health for all and one health and one bridge and all this stuff, I think these are two different things to discuss about the economical side or what is going on with our society. And I'm saying that because what is an important part is that, already Santino did mention the cost. And I mean, the two main issues that many countries have been brought on the table is that, the provision of healthcare, starting from primary healthcare to specific medical conditions, it has a cost. Okay, let's agree on that. Now, on the other side, when European community was discussing in the past the issue of undocumented migrants, because we did not face at that time the issue of a so-called refugee crisis, they had a study of Fundamental Rights Agency having 3 countries, that was Greece, Sweden, and Germany, where it was found out that the idea of exclusion from the healthcare system of access and for provision of services, it is increasing the cost of the healthcare systems. By including people from the beginning on the healthcare system, of course with the objective not for life, but the objective of integrating the society, it is less costly for the system itself. And I'm just giving an example. I have blood pressure and my cost of treatment is 30 euros a month. If I maintain my treatment and I have a physical normal life, my cost to the system is 360 euros. But if I don't have a treatment, I will have tomorrow a heart attack or have a stroke and I have to go to the hospital. So I think the important part at the moment is that when we discuss one health, one bridge, and everything, you cannot keep aside migrant refugees, whatever groups. First of all, I don't like putting tags on people. I think health for all, it is for everybody. So I think we need to work on that direction. And then other elements regarding how much is for the economy or for the public, etc., we let the economists to analyze that part. As medical staff, we need to talk about what means access to health. Professor Vandarakis, I think you had the floor.
Well, first of all, I would like to thank Apostolos for organizing this, because it's a really, really nice subject about One Health and combining with migrants and the migrants issue. I'm not very, how can I say, I think that it will be really difficult to adapt One Health issues in the migrants. And because even now, One Health, the health literacy, the terms, the terminology of One Health has not been adapted in the community in general yet. So think you need to adapt that to migrants as well. So I would like to ask you, how feasible you think that can be adapted, or the One Health issue in the migrants in each country, especially that there are countries that they have not faced the One Health issue in the major population, not only, of course, migrants. So I'm not very optimistic, at least in the near future, I mean. So how feasible you believe that can be done? And because we have speakers from European organizations, I would like to hear their opinion on that.
I think Santino and Keivan and Taimur, if they can take 2-3 minutes to give as an example concrete. But definitely I can tell you that as societies, the so-called refugee crisis showed also many weaknesses that we had, any crisis, including COVID and other stuff like that. So I think it is possible to be done. I understand your diagnosis, but the three experts that are on the other side, they maybe have a better point to say on that.
Thank you. I'm happy to start. And although from the public health point of view, I share the position of the gentleman in the latest intervention, I like to allow me to be a bit provocative at the end of the day, so we can keep discussing after the meeting. Personally, I believe that we're supposed to not to give any space to the risk of stigmatizing this population. The link with One Health project very strongly the idea of transiting health issues from an ecosystem to another. And migrants too often are seen as a carrier of communicable diseases, or a cost because unhealthy or abusing the utilization of our health systems. From the evidence we are having, from the study we are conducting, the main issues remain the fact that there is no adequate inclusion and equality or equity in accessible, but also instruments to ensure that a diverse population, a diverse demography can access to services. So the issue is not so much to link with other public health topic, but really the issue is to reduce the gaps that we are having nowadays in terms of navigating our health system. So my provocation is, less we see migrants as a population that is connecting ecosystem in increasing risk of ill health or public health issues, I think better it is for the sector.
Keivan?
Yeah, I think in terms of One Health, if we understand it, as Santino said, the connection between ecosystems, like animal health, human health, and the connection. And I think there is actually no way to not consider migrants in that sense, because if we replace the term migrants but by human beings in new environments, or mobile human beings, then ultimately they are a core part of the One Health concept. And I think it then becomes crucial to understand how we understand these socially categorized people, either only as risks, or if we find other ways to embrace human mobility in the One Health concept. And I think this will be crucial. And if these notions are to hold, I think there is no way to include them no other way. Yeah.
Teimur, on your experience on HIV, maybe you want something, or you are covered from your colleagues?
No, I think they've answered the question. I just think that it depends what the goal is in what we're trying to do. The goal for public health for me is to improve in general the public health, the health of all. In our case, in the EU here, which I represent, the health of all. And the second objective for me is equity in health. And I think this is where migrants come in. It's not only about migrants, it's all vulnerable populations we're talking about. We just need that angle. Just as we need a One Health approach, we need a migrant health approach. And they shouldn't be against each other. They should be integrated in whatever we do. So whatever subject we're talking about when it comes to health, we need to look at inequities, because we see huge inequities. Whatever we're looking at, we're going to see this inequity angle, if it's migrants or if it's homeless people or if it's transgender people. And I think the goal of a One Health approach, or as Apo said in the very beginning, is a One Human approach, is to address this issue. So not a great answer, but I agree with my previous speakers.
I think we have the last question, except people they want to stay more, but I think I'm sure that the rest are too busy. So there's the last question, I think, from...
Okay, thank you very much for this amazing and insightful presentation. Actually, I have worked in a voluntary basis with migrant communities within the Red Cross. And one problem I really faced during that time was that we as team saw these environmental and One Health challenges evolving rapidly within these communities. And yet the decisions were made from the top down, like from the respective agencies. From your experience, how could you think that this policy flow could be reversed, or do you have an experience would you like to share in this within the practical perspective of that? Thank you.
Yeah, I will let the lady of the panel to answer it.
Thank you for raising this really important question. One of the things that we see is that participatory approaches are misunderstood. And there's been a lot of tokenism. So I completely agree with you. There's a lot of tokenism and there's a lot of hierarchy. And usually people, migrants or any other underserved population, are usually consulted at the end to rubber stamp everything, instead of having approaches where they're involved from the start, because if they were, then the solutions that would be found would be better and more feasible. So I think this is what, there's a discussion about whether we should use the term stakeholders or not, but let's say all the actors involved should be included from the start of the process and not late. Also, it should not be consultative, because what happens is it's very consultative, which means there's no decision-making power. So in the decision-making power, representation is needed, because until you get representation in decision-making, in the decision-making forums, change is not going to happen.
I would like just to continue and to add one little small point on that. Just to mention that what Keivan said before about what way of approach we want. We want a horizontal or vertical, or we want a combination of that. I have to tell you that the experience of this country that we are being present is we have been specialists on making the life of people, physical and mental health, vulnerable as possible. So we have been deciding, for example, to exclude everybody from healthcare system, or to make it on a temporary basis. Like, a pregnant woman, if she loses the access to asylum, she has to stop her pregnancy because it's her fault. So that's what reality is about. So I never understood, for example, and coming to your point, the cruelty that we're building up. Because at the moment we're expecting the EU pact to start in June. And this means that people will be detained, more and more detention, increasing detention. And we know that the detention have been having systematic problems on physical and mental health. We've been documenting it. There have been reports and reports and reports. So I think we had enough of information, and I think we're creating more and more difficulties, including the people who are healthcare providers. They don't know what guidelines that they need to follow and all this stuff. So I think that the things can be simple and complicated, depends what we want to put on board. I'm not saying it's easy, I'm not saying it's difficult. I think there are a lot of positive experience. So one of them, for example, when we worked with Santino together in 2016, we vaccinated for less of the 2 months, 30,000 kids. all around Greece. So I think it's possible. It's a matter of political will, it's a matter of coordination, and it's a matter of rhetoric at the same time. Because I think at the moment, the rhetoric is that these people are inviters, so they're inviting us. So it's a bit of a point on that we need to take in consideration. If there's no other question, I think probably I would like to thank, first of all, the people that are online. Secondly, to thank all of you for being with us on this session. And again, thank you, Professor Vandarakis, for having us, and we really enjoyed the session. And have a nice evening to everybody.
I would like to say that now we've reached the end of day 1 of our conference. So we will go on tomorrow at 9 o'clock with day 2 and antimicrobial resistance. You can find outside light dinner. Thank you very much.